Well, hello. Good afternoon or evening, perhaps morning, wherever you might be tuning in. Welcome to Divestream 903 of the remotely operated vehicle SuBastian. This is the second dive stream for this expedition, which is part 3, really, of designing the future. So you'll notice that SuBastian's got quite a few new instruments on board, and we're going to be using those. If you tuned in yesterday, you saw some of them already in use. There's other instruments that aren't really visible, but hopefully we can learn more about those through the cruise. And right now we are looking at this little Tino and hoping to pass it through some lasers. But I will be bringing up the mission control audio in a little bit. If you tuned in yesterday, you know it was a bit nuts. So just hold on. And if you have questions, slap those in the chat. Yesterday we had plenty of scientists answering questions. All manner of different languages spoken in here as well. So hopefully we can get to all of your questions. And I'm going to hand it on over to Karen Osborne. Maybe a little intro, but then you'll be hearing mission control as a whole. So enjoy the dive.
Hey everybody, this is Karen Osborne. I'm an invertebrate zoologist from the Smithsonian National Museum of Natural History. And we are here to do midwater outside of Brazil. We're a little bit north. If you were to go straight out from the coast of Brazil, we're a little bit north of Salvador. And we are here to look at midwater invertebrates. We have a lot of different ways to look at them. One of the special ways that we've brought with us is this deep PIV, the camera that we're lining up right now. So this arm that you see sticking out in front of you is a camera and a laser sheet. And what we want to do is pass this animal through the laser sheet. And it's essentially like a little CT scan for the animal. So we can reconstruct the 3D structures, internal and out. It works great on transparent animals. You can get a lot of the internal details on them. Today we'll talk about what we're doing as we go. We are headed today down to about 1,200 meters. Famous last words, we're going to head down and then work our way up. But of course we will stop often when we see cool things like this. This is Thalassocala Sea. It's a lobe 18-4. And we're going to try to deep PIV it. Hopefully today we can give you a few narration in a few different languages, Spanish, Portuguese, maybe some Hindi, some Mandarin, some Arabic, German, Dutch. What do you guys want to hear? Juliana, do you want to, can you hear me? Yes. Okay. If you want, I can pause and let you catch up.
No, I can just wait you at the same time.
Oh, I don't think I can hear both at the same time. Oh, but then it's like too much.
Slightly less exposure still.
I'll do shorter chunks.
Just introduce yourself and say that I'm probably.
This would be another chance when people
So on point today, we have one of our other scientists, Julieta Cebular, is here. She is Argentinian. She will translate into Spanish periodically a lot of the dive.
Some exposure settings on our end. Okay. That was a little bright. You could see that the more reflective parts where kind of, we call it blooming. They just appear larger than they are.
No, it doesn't look like, it looks like in the CPD file, it doesn't show up in the real-time data feed that comes in.
That was excellent flying on to get that little scan right away. You had it ready for us right outside the screen, right outside the laser sheet.
Yeah.
Yeah, it's good.
Heading degrees, I believe, is the direction the vehicle is pointed, no?
Sorry, what?
Yeah.
Someone in the chat is asking what the heading degrees is. That's the direction the
Yeah.
vehicle is facing, correct? The 360 degrees around the vehicle, the heading is in that measurement of degrees, the direction that it's heading. And I'm going to guess 0 is north? Yeah, 0
Yeah.
is north.
Yeah.
Just
I do have
checking
to
with
call
Sunny,
out-
who's driving today.
In the Twitch, we just got an emote I've never seen. I love it. It's little skateboarding crustaceans. So I'm ready for that. That's good. Sorry, I had to call that out. It's good.
That's
It's good.
awesome.
Okay, get ready to turn off.
Yeah, maybe like
Turn off. Yeah.
Oh, it's on the deucer earlier.
Nice.
Yeah.
That was beautiful. So you're seeing a
Push it straight
cross section
back.
of the animal as it goes through the laser. That is nice. Looks so thick on the scan and in person when you have this guy in the lab. Yeah, definitely. Yep.
there's a spectrum. I think we're getting
I mean, you don't need to say whether it's a success or not. You can just say we collected data. That's all. That's all that we're worried
Some of
about
the
on it.
scans are like,
So you want the time code here, not just we did it.
oh, okay.
So the time code you can get up there on the bottom of that.
I would like to.
That's the feed video.
Oh, you activate it, then you're going to press A for annotate.
Yeah, there's 4 digits.
Chair off?
Or 5.
Yeah, there you go.
Yeah.
You write in what kind of animal it is. And
Yeah,
then
you're
most
welcome to
recently
translate
things-
anytime.
Did you focus? Did you go off the same setting?
So just do time, yeah, like that. Time code when we start, and then we know that this is a section, and they can go back and get it all.
I can see it.
LLX
It's larger than it should be. Yeah, so part of this relates to laser, maybe the size of part of it.
Oh, 1L.
I'm tempted to play with it.
This is the hardest. Yeah, ELL.
Trying to
I don't want to get A,
speed of the vehicle constant by the time it gets to the
We're coming up.
So they want to give
Oh, there it
a little bit of runway.
How deep are we going today, Karen? How deep are we going today?
1200.
1200.
Okay, so that gives you the label. And can you just click there once? Double click. I've already captured the team once. If you can press click to link the label, and then just pick the other side. That way we don't have to shout it. Just click it again. Okay, now it's linked. And then you get out with a little cross. Not that one, yeah. So normally we'll just capture.
Hola, buenas tardes a todos. Estamos acá en la segunda inmersión del ROV a bordo del Falkor 2. Mi nombre es Julieta. Bueno, les voy a estar contando un poco de lo que vamos a hacer hoy. Así como les conté ayer y como el público se renueva, hoy en esta expedición somos varios investigadores de diferentes países y de diferentes instituciones que estamos estudiando el zooplancton del Atlántico Sur, más o menos a la región a la altura del norte de Brasil. Y bueno, como pueden ver, ahora estamos yendo atrás de un tenóforo que pertenece a la familia de los alfilón de los nidarios, pariente de las aguas vivas. El ROV fue equipado con un nuevos tipos de cámaras, cámara iris. Son diferentes cámaras que pueden escanear los animales y tomar fotos de desde diferentes ángulos y diferentes lugares para poder crear una imagen 3D. Y eso es lo que estamos haciendo en este momento. So when we're scanning these things, it's really important to try to get the animal as stable as possible. And then what we want to do is just scan it at a really even speed, bring it through there slowly so we get lots of high resolution data on each layer as it moves through the laser, which is an incredibly difficult thing to accomplish with moving animals, currents, and a giant vehicle. But the pilots here are pretty awesome at doing this. Quien está conduciendo la inmersión actualmente es Karen Osborne, que es la coordinadora científica embarcada. Y yo voy a estar traduciendo un poco de lo que ella está explicando y de lo que estamos haciendo para los que están del otro lado de la pantalla. La idea es que el tenóforo esté lo más estable posible y lo más quietito para que puedan escanearlo bien y que las imágenes sean nítidas. También lo que hacen estas cámaras es medir la corriente alrededor de los movimientos para poder estudiar mejor cómo son los movimientos que hacen abajo del agua. Do you want it further out in the laser? Or further out in the...
I mean, yeah, you have more space. This panel to the right of the camera. That's a pretty good balance, I think, where you see what you need. Yeah.
So we turn off the lights, most of the lights of the vehicle when we are running it through the scan because that makes the data a little bit cleaner. You don't have as much other stuff in your view. It just isolates it down to the laser. Como les contaba, a bordo de la expedición hay personas de diferentes países, entonces la idea va a ser que durante la inmersión del ROV haya traducción en distintos idiomas. Hay gente, somos varios de Brasil, que pertenecemos tanto a la Universidad de San Pablo como a la Universidad Federal de Río Grande en el sur de Brasil. Silvina y yo y el profesor Renato Nagata estamos representando a la FURGI. El profesor Marcelo Melo y Marcos están representando a la USPI y después hay varios investigadores de Japón, India, Estados Unidos, Países Bajos. Entonces, a lo largo del día van a estar escuchando las traducciones en diferentes idiomas para poder alcanzar y poder transmitir el conocimiento a diferentes lados.
We have people here from 13 different universities and institutions spread out across United States, Australia, Japan, South America. It's a big group. It makes it really fun to all collaborate and work together and learn about each different person's research tools that they bring and different ways we can look at these animals and learn about them.
Does it come out darker for something or nothing inside?
Cualquier duda que tengan pueden escribirla en el chat y tanto yo como cualquier otro de los investigadores que está a bordo del barco, que obviamente son expertos en el área, pueden responder.
good if I zoom in on your side?
Yeah.
Is it better if it goes in here and stays, or is it better if it passes through and you kind of
I mean, ideally have like a constant speed, so pass
passing
through.
through?
So kind of hovering outside of the sheet until the animal's kind of lined up right, and then like accelerating before it hits, so that you're like constant speed through.
yeah.
So oftentimes what the pilots do is like they'll push on the throttle, they'll push forward, and then sort of coast through it.
Right.
But they also don't want to go too far after that, because then you spend time lining up.
Right.
So it's like this really narrow band. And you know, we're asking them to move this 8-ton vehicle with millimeter precision.
You mean this thing right in front of
You're
the camera?
demanding, aren't you?
Yeah.
Not at all.
Yeah, it just covers that vertical height of the. Yeah, so it's a little more forgiving, just a bit to the right, then of course, it's easier to get it outside of the pool.
Guys ever use the laser sheet to measure particles in the water?
We're not really focused on
No.
Actualmente estamos en aguas que están afuera de la zona económica exclusiva de Brasil, a la altura de pasando el talud continental. Y el objetivo de esta expedición es estudiar el zooplancton, todos los invertebrados que están presentes en la columna de agua desde los 200 hasta los 1200 metros de profundidad.
The objectives of the cruise are a few fold. So we're hoping to describe a lot of the midwater diversity. Most of what's out here is unknown, undescribed. Oftentimes there's a couple of species described and they're assumed to have global distributions, and then when we go in and look in detail in different locations, we find out there's actually a bunch of different species that look fairly similar. We can tell them apart oftentimes with morphology, certainly with genetics. And so what we're doing out here is seeing what's here. We're exploring a new area. We haven't been down here before, using this technology, and we can image the animals. A lot of the animals are too delicate to collect, or too big to collect, and so we can explore them with the video, with DPIV, with some of the other instruments we have on the vehicle to collect data on their morphology in situ. And then we are also collecting animals, a few in the samplers on the vehicle, and also with some specially designed midwater tucker nets that we can collect animals and bring them on board. And what we want to do is build reference libraries that actually include all of these animals, so that when people are out there working with eDNA, instead of just getting a no match back when they try to figure out what their sequences are in their eDNA samples, they actually get an animal back, because we've come out here, identified the animals, sequenced them, publish those sequences to public databases like GenBank, so that they can be comparable to eDNA data sets. It's really important to get those animals in there, so they can be referenced. And we're actually sequencing on board. We're also sequencing full genomes of a few animals, and the first one happened yesterday on a new species of jelly. So that will get a genome and it will get described. We will do that with several animals. So we're interested in lots of different animal groups down here. We've put together this team from 13 different institutions that have experts on copepods, experts on larvaceans, experts on high-barred amphipods, experts on worms, experts on jellies, experts on mollusks. So we've got as broad a group as possible to come out here and look at these things, because we kind of never know what we're going to find when something really interesting or unusual is going to show up. But everything out here is interesting, and much of it hasn't really been assessed for how much diversity is really there. So that is one of the objectives of this cruise, is to see what's out here.
Como contaba Karen, todo en inglés, imagino que algunos podrán entender y otros no. El crucero, en este crucero hay investigadores de muchos lugares con diferentes especializaciones. Tenemos especialistas en crustáceos, en peces, en medusas. Tenemos geneticistas a bordo y estamos comenzando a secuenciar genomas completos de algunos organismos. Tienen todo el equipamiento para poder hacer estas tareas a bordo. Realmente es increíble todo lo que tienen. Entonces la idea es, entre otras cosas, poder, como no se sabe mucho de esta región y de estas profundidades, poder conocer más en profundidad lo que tenemos en la columna de agua, en estas aguas del Atlántico Sur. Y por eso este crucero, organizado principalmente por el Instituto Smithsonian en Estados Unidos, trabaja hace 30 años en estos ambientes y se reunieron una serie de especialistas para poder estudiar mejor toda la biodiversidad del zooplancton. No, Viviana, todavía me parece que no no cerraron los votos para esa categoría. Creo que todavía está abierto.
The impression of the Lassicanosi is that it's a fairly shallow water animal. They're completely holopelagic, which means they don't associate with the bottom at all. We usually see them up here in pretty shallow water. Right now we are at 460 meters. Which some people don't consider particularly shallow, I suppose. I've been working on deep sea for about 20 years now, and so 400 meters is the upper water column to me. But they are certainly deep. But we see these up shallower than this as well, but not all the way to the west.
Like this animal, at least when you're flying next to it, it's not falling apart. But things like
Nosotros estamos
larvation
usando
houses.
el ROV para muestrear, sino también en los horarios donde el ROV no está operando. Se hacen muestreos con red de plancton y con red Tucker, que permite el muestreo desde los 200 hasta 1200 metros de profundidad. Y una vez que salen las redes del agua, todos empezamos a separar los animales de interés de cada especialista. La gente de San Pablo, que está trabajando con peces, está montando la colección del museo. Los demás investigadores que trabajan con copépodos, con camarones, diferentes tipos de moluscos y cefalópodos, hacen una serie de investigaciones con imágenes para poder crear poder crear las diferentes, poder crear imágenes en 3D de estos bichos. Y tanto Silvina como yo estamos tomando muestras para isótopos estables para poder estudiar la red trófica del lugar. Sí, la corriente Malvinas no llega hasta acá. La corriente Malvinas llega más o menos hasta el sur de Brasil. No, sí, hasta el sur de Brasil. Acá lo que predomina es la corriente de Brasil, superficialmente, pero como ya estamos a más profundidad, lo que están predominando son masas de agua profundas que por lo general son mucho más frías. Como pueden ver en la pantalla, en la parte de abajo, en la izquierda, pueden ver la profundidad, que actualmente es 460 metros, y la temperatura está a 7.01 grados.
Lots of those guys around. I don't see them. They must be more tropical because we don't see those so often up north. Some lights were
First things up, but
Need more light?
a little.
Lights on.
Another one. Seems a little stiffer. Might be another smaller philosophy. Yep.
Okay?
We want to scan some giant larvaceans. We did
Como
not
les
see any
contaba,
yesterday.
actualmente algunos investigadores que trabajan con microrobótica pudieron conectar unas nuevas cámaras que crearon al ROV, que son una serie de cámaras que toman fotos de diferentes ángulos y permite recrear imágenes tridimensionales de los organismos. No solo toma las medidas y permite ver el volumen, sino que también puede medir las corrientes que pasan por los tentáculos y pueden estudiar mejor los movimientos.
SD skin.
Maybe a
Oh,
bit
it's over here.
more forward.
But the aft camera. So try aft camera.
So grab it, keep it down.
There you go.
Possibly a little bit more forward as we descend.
That's
5 centimeters.
Viviana es el
It's
tenóforo
very
que estábamos
incipient.
viendo en pantalla. Estamos diciendo acá por cucaracha que puede tener menos de 5 centímetros más o menos.
Oh, there's a tail of something. And a little calicopher in there. Let's see if we can find the head.
Ahora parece que estamos viendo
Might be a ghost tail.
un filamento de alguien o la cola de alguien
Active.
y estamos intentando buscar de quién es.
I had that done. It's a ghost tail. What's that?
I had that done earlier as a copepod.
Copepod?
Huh?
I don't think so.
Yeah.
Okay. But this right here is
This is
Perfect view
probably a
from the lower
ghost
portion.
tail. Or what's left of ghost tail. Pretty ghosty.
Oh, Karen está diciendo que posiblemente sea la cola perdida de alguien que la dejó en el camino.
Alright, carry on.
Más a la noche, a lo largo de la inversión del ROV, vamos a tener diferentes charlas o diferentes traducciones. El profesor Renato Nagata va a estar traduciendo al portugués y obviamente va a poder explicarles mucho mejor de estos organismos de los que yo les podría explicar. Tanto Silvina como yo trabajamos con mamíferos marinos y bueno, nuestra actividad a bordo actualmente es colectar muestras de los diferentes bichos que estamos juntando para hacer análisis de isótopos estables. El profesor Renato Nagata de la FURGI es especialista en medusas, entonces obviamente va a poder contarles mucho más de estos organismos que son muy copados.
No, although right now there's everything's deep. Right, so that's, we're gonna try to, everything should be spread out, so we're gonna try to get down, get an idea of what everything is. We just wanna see what's in the water column, and then we have time to take our time and explore down. Given our proclivity to stopping often, it makes it hard to make a lot of depth.
No, actualmente no se colecta audio, no se colectan datos para bioacústica. Quién sabe, en algún futuro alguien desarrolle algún equipo para poder adaptarle al ROV y no solo a medida que toman imágenes de los distintos organismos, pueden tomar datos para bioacústica.
One tries to pretend nothing's going on, and the other one definitely wants to make sure you know about everything.
Pero imágenes de mamíferos marinos es bastante difícil con el ROV porque necesitaría ser un equipo muchísimo más grande.
Oh yes. Still in there. Middle hanging down. Nice galaxy.
Aparentemente lo que estamos viendo ahora es un sifonóforo. También es pariente de las medusas.
It's got a lot of bracts, the transparent parts that you see above the kind of main stem.
Lo tengo acá Renato al lado mío y me dice que es más parecido con las carabelas portuguesas que quizás con un agua viva, porque pertenece a otro grupo.
So the yellow bits are the tentacles and the nematocysts and a fish just about
Renato,
got
si
caught,
quieres
but
decir
got
algo
away
en
and took a
portugués.
tentacle with it. Oh, you can see nicely, you can see the head, which is hanging straight down there.
Boa tarde, pessoal que está nos acompanhando. Bom, o que eu acho mais incrível sobre esses animais, vamos passar para o português aqui um pouquinho, é como que um organismo que não tem cérebro, né, consegue se movimentar nos oceanos e se posicionar de uma forma como esse animal se posiciona, né? Ele faz uma espiral completa. Então ele tem ali na parte de baixo, que vocês podem ver, os propulsores, né? Que são como águas vivas que realizam a locomoção, que pulsam. Eventualmente vocês vão ver esses propulsores, que são chamados de nectóforos, pulsando mas é incrível como ele consegue adquirir esse formato em espiral que é super eficiente para ele escanear e capturar partículas de água, partículas na água, né? Partículas de alimento na água. Então ele está numa posição de pesca ali, né? De captura de alimento, em que ele otimiza muito o espaço que ele pode ocupar para reter o máximo de animais, né, e se alimentar. Então é um organismo que é colonial, né? Então cada parte dele seria um indivíduo diferente que atua de uma forma integrada, né, para o funcionamento da colônia.
Como Renato estaba contando en portugués, estos organismos son coloniales y cada uno de ellos cumple una función. Principalmente los que estamos viendo ayudan más a la locomoción. Lo interesante que él contaba es cómo estos organismos se ubican en el agua y cómo coordinan entre todos ellos para poder moverse y poder alimentarse. Sí, Viviana, a estas profundidades se pueden encontrar algunos mamíferos, como por ejemplo cachalotes. Pero bueno, el objetivo ahora de este trabajo es estudiar todos los invertebrados zooplancton. Actualmente estamos más o menos en las aguas internacionales, pasando la zona económica exclusiva de Brasil, a la altura de Salvador, más o menos.
Wow.
Están pudiendo hacer
That is nice.
un montón de imágenes con el láser. Están todos súper contentos
Look at those
acá, está todo el mundo prestando atención a las pantallas.
Those are the protective
La verdad
bits
que
you see
es
on top of
súper
the-
lindo, es
of the stream.
increíble. Tienen de todo acá adentro para poder hacer estos estudios. La verdad que es una oportunidad increíble de poder estar acá.
Because you press record, right?
Beautiful. All right.
Do that on the second dive.
Back out through the.
Come
get
back
the
to
hand
here.
back.
Wow. Deben estar ahí tomando imágenes increíbles. La verdad que las cámaras que tienen
All right. We'll let him be.
son
Beautiful.
muy potentes.
Very nice flying.
Y bueno, imagino que al terminar la expedición van a poder publicar cómo quedaron.
Nice.
Thank you.
Very nice.
Está todo el mundo celebrando acá que pudieron tomar las imágenes.
What do you want to collect? The head? In a D?
It's a new family. It would be
No,
great
el
to
láser
have
no les hace nada,
important things
solo
that
ayuda a
are
escanear
before too,
It's
los
pretty
organismos.
so bits
You
of
could
the
put the
tail.
head in a D.
Bits of the tail would be more useful than the
Yeah.
head.
So you want to suck up a bit from the back end.
Suck it up from the back end, yeah.
Which section? Which jaw do you want
More
to?
value to
It's fine. We're on 5. We're on 5 right now.
If any of the pieces get come off the colonies,
it's a bit like a CT scan. Yes, you're gonna translate it.
Yes.
So it's like a CT imaging scan where every frame of the video becomes an imaging frame, but we're only seeing like a slice of the animal at a time. So by scanning the entire vehicle with the instrument back and forth, we're really just doing one big pass through. You get these slice by slice by slice. It's almost if you were like cutting the animal in many tiny little slices, and then later on the computer will put those back together. It doesn't harm the animal at all.
Acá me estaban explicando cómo funciona el escáner para poder contarles. La idea es tomar imágenes de diferentes partes del organismo. Es como si lo cortaran con una cortadora de fiambre. Entonces va tomando diferentes secciones, imágenes de diferentes secciones a lo largo del individuo, lo que le permite después reconstruir todo el tamaño en su totalidad. Y no, ese láser no los lastima de forma ninguna. Por suerte, los chicos que trabajan con estas imágenes y con estos láser y que crearon estas cámaras están acá en el estudio y pueden responder todas las dudas que ustedes tengan.
Might be necessary with this guy.
Yeah.
So we're going to try to suck up the tail. This is a new family of Bisonex cyclonophores.
Which will ride on the end of the tail, plus we drop them through growth. Plus you'll get some of the larger tails.
Claro, sería como hacer una tomografía.
Well, it's a lot smaller now, though.
You might get the whole thing.
Yeah, you might
Lo
get the whole
van a
thing.
fusionar. Sí. Lo van a
Oh!
Obviamente estaban todos acá celebrando.
Get in the canister.
SS5, right?
So it's just, yeah, and just move forward. Yep, exactly. That'll just create a really
Five,
gentle
SS5,
suction. It'll
thanks.
push it back into the...
think.
It's 5.
Suction? Okay.
Pero primero
That
pasan
was
también por otra serie de
18,
cámaras
17.
que les toman diferentes fotos para poder estudiarlos en su totalidad. Así que estamos todos esperando.
We could. It's harder to see it. Especially.
Yeah.
We're watching the bucket to see when it comes in.
To the bucket that we collected in,
Bring it inside.
the canister that we collected in, there is a shadow cam, which,
It's going through.
it's in. So it's passing, right now it's
Está
passing through the
pasando, podemos
chamber
ver
on its way, and as it goes
que
through
está
that
entrando
chamber,
en el colector.
basically take pictures of the shadow that it creates, and you can see really fine detail in that. Did you see if the head came in?
Some soup.
Can't.
There it is. Yeah, there it is.
All right.
They're no longer connected. Here's the ectosome. So the part that looked like a tail is sort of like asteroids eating cracks.
Let it settle a bit, and then we can change
A medida que
the
fue
buckets.
entrando, como es un organismo colonial, fue separándose en cada uno de los pequeños individuos. Y bueno, ahora podemos ver que está todo adentro del colector. Así que están todos muy contentos.
So moving to buckets.
You feel that on your tongue.
Wow, you don't get hardly any flow when you move across. It's probably because it's a tiny house. We can always see like big swirls and move. Super. All right, let's go down.
Can we get to 1200?
That was 100% where it's stopping for. And we've made great progress today. We're at 560 almost. Great progress.
Bueno, esta
Almost
primera
halfway.
tentativa
I mean,
fue
we're
un
only
suceso,
an hour.
así que ahora vamos a
An hour
seguir
into the
buscando
dive. That
cosas.
is really record time for us.
Basically free fall.
I mean, this is not benthic, right? We don't bomb through and not look at any of these cool things.
No, if we were a piece of marine snow,
If we're a piece,
rocket right now.
we would be a rocket, exactly. It can take years to get to the bottom. Fed on multiple times, in multiple ways. It was a little fish, probably Luriglossus or something. All right, so a little bit of forward as we go down. You need to get settled or something. Okay.
Actualmente estamos en aguas internacionales, fuera de la zona económica exclusiva de Brasil, a 570 metros de profundidad más o menos, a la altura del Salvador, Brasil.
Little fish over the side, but we don't stuff the fish. We don't stuff it. Okay, we do. Oh, it's not a fish. It's a piece of siphonophore, probably. Oh, look at this guy. Some type of period, siphonophore.
Rosacea.
Rosacea.
Rosacea, that is...
Probably.
Es un sifonóforo.
Aí a gente consegue ver os...
It did not disintegrate, but it did
Yesterday. We didn't catch
come
any
into
today.
pieces.
Vamos trocar um pouquinho para o português. Agora, um encontro com outro sifonóforo, um animal colonial, como foi comentado anteriormente. E agora sim nós podemos ver os propulsores, né, que são os nectóforos ali. Então a gente consegue ver, são dois que estão pulsando e são esses nectóforos que realizam a locomoção do animal. E a parte posterior, que são aqueles pontos amarelados, é a parte da colônia que é responsável pela alimentação do animal, né? Então são vários indivíduos que trabalham juntos, mas que têm uma morfologia diferente.
I'll fly the set, you take cyan, do whatever you need to do with the
So now you guys are seeing another piece of equipment. This is called
Just answering Mariana Silveria Piccolo, it's like having a CT scan. Maybe, yeah, it's in numerous images that are done that are unite and they do a three-dimensional image of the whole animal.
It was probably an orcaplurid. This is not it. This is still a prey. It's the one we're looking at. But you could put in there that the orcaplurid was free swimming. No house.
Como nos contó Renato un poquito, que obviamente es nuestro especialista, eso que estábamos viendo antes era otro sifonóforo, también primo de lo que del otro sifonóforo que estábamos viendo antes, otra especie. También son animales coloniales.
So the piece of equipment you see that we're bringing out to set up now is called Iris.
Respondiéndole a Fede, todavía no
In other
cerró la
words,
votación, creo que los resultados van a estar, si no me equivoco, a partir de la semana que viene. Y sí, todos esperan que ganen.
So it has a single large sensor in the back, and then instead of in between the lens and the sensor are 2,400 individual lenses. So each one of those lenses focuses at a different depth. And when you post-process that video that comes in, you can reconstruct the three-dimensional structure in a cube about 14 centimeters. About 14 centimeter cube. To get actual measurements, which is a challenge in the midwater.
But you'll see when we
La batatita era un pepino de mar, como aprendimos en la otra live con la gente de Mar del Plata. Ahora vamos a tener que darle nombre a estos bichos de media agua.
So we have lights. The red that you see is the lights. There are little arms out so that we can really light the object. Oh, look at that spear. That almost looks like a big butt. Probably not. Probably some type of single-celled organism. What you're looking at.
That's awesome.
That's the sweet spot.
Yeah, that's the sweet spot.
I think we got it here.
I think we're blasting it.
What's that?
We got it.
So we're not
Yeah.
seeing the
Exactly.
De nuevo con el escáner están intentando sacar varias fotos de este sifonóforo para poder reconstruir la imagen tridimensional.
So the colors tell you how far away the pieces are. If the warm colors are closer, the cooler colors are further away. When it looks like you're looking at it through a compound eye, which is a little bit of what you're doing, that means it's too far away for it to measure. It's a
Sí, creo
pretty
que va a ser
small
difícil
area
poder
to get it
captar
set up
una
and to be
ballena
able to see.
con el ROV desde acá, solo si
Oh,
pasa así
Halotraphes
muy
over to the
cerca.
right.
Pero bueno, ustedes pueden ir pensando diferentes nombres para los organismos que vemos acá en la columna de agua.
You probably won't look at the exterior of the but I think that was fine.
Is it recording?
Yost, you're recording those. You're recording those, right?
Sorry, the one thing on top, it was like, oh.
I know that is.
So it doesn't auto detect or something when
It
there's something in the
can.
thing?
Yeah, it
Okay.
can. I don't know.
Okay. Oh, that would be super cool. So it only
Thank you to the Sasakawa Foundation
for that. Excellent. Indeed. Sasakawa Foundation provided funding for all the participants of this cruise and also to develop this instrument further. So
Yes.
Okay, Yost. All right, we'll take that. Next up is
Okay, you could take the side camera. Yeah.
Before the U.
Principalmente estudiar terópodos, que son algunos tipos de moluscos, copépodos, camarones,
So what comes off of this camera is
pulpos,
really high,
calamares, así como peces también.
really high data volume for that camera because of all the different depths that it's focusing at. It's a lot of data and hard to store. And so one of the things that they're developing with the funding from Ocean Shot is to be able to have it automatically detect when it records. So as soon as something shows up in that picture, it records and then it stops as soon as everything's outside of that square. Because as you can imagine, as you're flying around trying to get this moving animal and the moving sub and the currents, trying to get them all lined up perfectly, could potentially be recording for a very long time, trying to get that sweet spot, getting the animal into the sweet spot. So one of the things they're trying to do to make this more viable is target the bits they record.
Trabajando en una universidad brasilera en el sur de Brasil, que es la Universidad Federal de Río Grande. El profesor Renato, que está acá conmigo, él es brasilero, nativo, y estará haciendo las traducciones en portugués. Ayer también tuvimos traducción para hindú, porque tenemos algunos investigadores que son de India.
Like doing a scan of this. There's a nice little lobate passing by, little photo bombing of the DPID, but we're focused on the prey, on the rosacea. And yeah, we're hoping to bring a lot of different languages today. We have Spanish, we have Portuguese, we could do some Hindi, German, Dutch, Swiss, German, Mandarin, Arabic.
Sí, sí, está traducido por inglés.
Okay, killing lights, that?
So we kill the lights when we do DPIV to just minimize the background images and really focus on the laser shoot as the animal passes through. It gives us essentially a CT scan of a slice of the animal. So this is a real challenge for our pilot. They need to line the animal up, and they need to kind of they love it. The pilots love it. They're not tense at all when we're asking them for this. We ask them to line it up just outside the laser and then just kind of have it glide through slowly and evenly through the laser sheet.
Estos animales que pertenecen al filo Cnidaria, tanto aguas vivas y tenóforos, pueden preguntar, pueden escribir en el chat que tenemos acá nuestro especialista brasilero que puede responder y yo traduzco para el español.
To help them in their work here, we have a bunch of different cameras that give them different views so that you can try to coordinate in your head where the animal is. I'm looking at it from a couple of different directions. We have one that's looking down the laser light, that top instrument that you see pipe clamped on the top is a camera that's looking straight down the laser line so they can see when it's getting close. We have one that's our main camera is just looking straight at the side of the laser sheet to collect the data. And we have a couple other cameras up on the sub to kind of give different perspectives from the side and above. And then on this cruise, we also have a really wide angle stereo camera that is mounted. I'm not sure if it's mounted today, but that gives us a really wide angle view for when you see the really big animals.
So, my thing
There's the lobate.
multiple things coming in multiple ports. Is it
another possibility.
Think about what these guys are doing. The animal is moving, the water is moving, and current, the vehicle is moving, and the vehicle moves water around with it. And pilot has to keep in mind all of these different things, so that it's really important to have a really great 3D spatial map in their head.
A little bit if you're comfortable.
Yeah.
It is quite a challenge to fly this vehicle and do this really precise stuff in the midwater.
Voy a responder para Mona. En esta inmersión no vamos a llegar hasta el fondo. No vamos a estar en las aguas bentónicas. Solo vamos a estar analizando y colectando muestras en la columna de agua desde los 200 hasta los 1200 metros de profundidad.
So the animals that we're interested in here are holopelagic, which means they spend their entire lives up in the water column. They don't spend any time associated with the seabed or the walls of the canyons or anything. They spend their entire life up, drifting, swimming, floating in the midwater. We call them holopelagic. We also see a lot of things up here that are spend a part of their life up here. Maybe their reproductive stage comes up into the water column, or their larvae come up to the water column. But most of the diversity that we're interested in are these larger animals that spend their entire lives up here.
Is the order of this important to this? So the second one should be CTD.
Until right before it gets to the sheet between them.
No, I haven't seen it. Pretty okay.
This could be
every time I see a cow, it's just not do anything, I do stuff over there, that goes up and down.
Indeed. Yay for international collaborations. It's really fun to work with a diverse team of researchers.
Mandamos saludos desde acá y tal vez más tarde la escuches así hablando.
Once I have it there, line it up behind the window.
But why do they have such bright spots at so with along the tail? That's not
The
terribly
stomach and the gonads are highly reflective.
and they're really super close together, almost in the same
Yeah, they
structure
have-
on these guys, or do they spread out
These
at
are
some
the
point?
chlamydia,
Yeah,
and they're very large, meaty, rounded bracts which keep each of the chlamydia
Keeps them
separate.
apart. Okay.
And then the stem in between is very highly extensible.
Yeah.
It can become real like a long piece of fishing line.
okay.
And each of the chlamydia.
And then they look like Christmas lights spread out
Yes,
with the
exactly.
the bracts
Yeah.
and then the stomachs.
in prayer, when it's contracted to this amount, it'd look more
Solid.
spirally
Yeah.
and solid. Yeah.
It's cool.
Where's it going?
So on this animal, this is a Calicofferan siphonophore. That means it has two swimming bells up on the top,
Is he still
and then it has a little
Yes.
string tail that connects all the different individual pieces. Each piece has a stomach, gonads, and stinging cells, and also bracts. Bracts are gelatinous, kind of like they're protective pieces. And on this one, when this tail is shortened and contracted like this, those bracts keep the gonads and the stomachs together, which is why you get this kind of string of Christmas lights feature where they look very separate from each other, each of the pieces. Some of the other siphonophores are more continuous, more spread out, or more even along the tail.
Just keeps on swimming, huh?
This is what I'm heading against.
It takes a little breathe on.
Just like Freya, I think we only have like one's for at least one sort of having some
Lo que están intentando hacer ahora es poder tener este sifonóforo lo más estable y quietito posible para poder escanearlo y poder tomar todas las imágenes posibles para tener mejor resolución. Están diciendo acá que pudieron tomar algunas fotos, pero no completamente. Así que están intentando tomar más. Vamos a estar más o menos por la misma zona, son 10 estaciones en el nordeste de acá de las aguas internacionales y terminaríamos más en el borde de Brasil. Y una vez que termina la expedición, el barco vuelve a Fortaleza, donde desembarcamos. Son en total 10 estaciones en donde vamos a ir colectando tanto con el ROV y con las redes Tucker y red de plancton a diferentes profundidades para poder aumentar los conocimientos que ya se saben y los que no se saben también, porque es un área que no es muy explorada, sobre los invertebrados de la columna de agua.
The animal we're working on now is Rosacea. It is a preyed sarconophore. Trying to get a slow scan. He's giving us challenges because he's swimming quite a bit.
Looks like reality. It's not. The kids are watching it. This is my kids. They're watching at home too.
Oh, really?
River and Sage. Yeah. Cool.
It's a four.
How old are they?
Just say hi to them.
Oh, hey River. Hey Sage.
How old are they?
They're 4 and 7.
7. Okay.
Sí, tanto Silvina como yo nos formamos en universidades de Argentina. Silvina es de la Universidad Nacional de La Plata y yo estudié en la Universidad Nacional de Mar del Plata, y las dos nos mudamos para Brasil en diferentes momentos para hacer tanto la maestría, doctorado, y actualmente Sil es profesora en la FURGI. Y obviamente, claro, que le agradecemos a nuestras universidades públicas de Argentina. Tal vez Silvina se esté queriendo morir en el laboratorio viendo los comentarios. Sí, es todo mérito, así como obviamente tuvimos una gran educación y obviamente la resiliencia de quien sale del país en algún momento para poder continuar con sus estudios. Actualmente en el barco somos 5 investigadores de universidades brasileras. El profesor Renato Nagata, especialista en nidarios, o sea, en medusas, sifonóforos, tenóforos. Tenemos 2 investigadores de la Universidad de San Pablo que estudian taxonomía de peces. Y Silvina y yo trabajamos con mamíferos marinos, que no tiene nada que ver con lo que sea en este crucero, pero actualmente, como trabajamos también con estudios de dieta, estamos tomando muestras para análisis de isótopos estables, lo que nos permite estudiar la cadena trófica.
Fix as it goes down the tube. We have a pollinated light, chamber with camera collimated light on one end of the chamber and a camera on the other end of the chamber. And as the animal goes through, you get a shadowgraph or a picture of the shadow of the animal. It has very high resolution, so we can see a lot of the details that we can't see with
Bueno,
our
como pudieron
laser
ver,
sheets.
estaban intentando
And
también
it will be imaged as it passes
atrapar
through the
ese
chamber.
sifónóforo. Me parece que ahora está pasando por las cámaras para terminar de escanearlo en el interior del ROV. Y en algún momento vamos a ver cómo cae en el cubículo.
So it's still on.
Okay, send the jar.
In the jar,
No.
oh, in the tube, okay. Like, wow, that was super fast.
No, gracias a ustedes que están del otro lado escuchando.
Down the tube, very nice. Fine, very nice.
Lo,
Cara, certain point at the hose I goes into the sections and got loose, so it wasn't given full suction. The second half of the
Okay, that would explain why some of the ones have lots of stuff.
but now it's taped
All right,
secured.
super. There's always something.
This one.
Oh, sí, ahí llegó en diferentes pedazos, pero llegó.
Number 6, yep.
Un suceso.
Fragile. Nectophore is intact. The two bells are still together. It's good to see. Sorry.
Una parte del quedó intacto y después la
Yeah.
parte colonial se separó en dos, pero bueno, eso no es ningún problema. Ahora vamos a seguir moviéndonos para buscar otras cosas.
Laying on the
Vamos a ir hasta los 1200 metros de profundidad, así que esperamos tener una inmersión de 12 horas y a lo largo de estas 12 horas supongo que iremos cambiando de traductores.
PIV'd it and then sampled it. And then same thing with the sky rosaceae. Yep. Iris,
That's the
PIC.
galaxy Sapanophore.
Yep. Rosacea is the little.
Another jelly.
It's a prayed siphonophore. Yeah, it has two twin bells of Tom. Elastic galaxy is a lobate ctenophore. Super.
What's the
We're headed to 1200.
Cereboma.
Getting close to 600.
right up front?
Oh, look at that. Cereboma.
Vertical silver top right.
Bony fish, cartilage fish?
Yeah.
Bony fish, yeah. CER.
Okay. Ahora estábamos cerca de los 600 metros de profundidad y lo que habíamos visto era una larva de un pez. Oh, se escapó.
Not one of picture taken. Oh, he's out there still. He's stuck a little ways away, but that's okay. We can keep going.
También si tienen dudas sobre ecología y taxonomía de peces, tenemos actualmente acá en el estudio a Marcus, que es de la USP y trabaja con peces.
You want to switch? Or? And then I'll relieve you. Just ping me. I'm gonna get a cup of coffee and sit in the back. Oh, something on the left.
A low bait. Pretty beaten up. We can leave it.
Oh, ahí pasó un bebé de medusa.
Looks like Salicera by the way it's swimming. Little jelly. We can go on.
Marcos es Marcos dos Reis Jr. y está haciendo su doctorado en la Universidad de San Pablo. Entonces, cualquier pregunta que tengan sobre peces, él puede responder, va a responder en portugués, yo puedo traducir para español y así seguiremos a lo largo del día. Esperemos también tener la participación de Manu, que trabaja con fitoplancton y hará las traducciones para hindú.
We're going to 1200 meters today. And probably the individual parts of the Cephalosauria came apart on the suction, and that's just due to the shear of the suction moving through the tube. Most cephalophores are described from their bits and pieces, unfortunately not connected to each other. That little fish that just went by was probably cyclothone or bristlemouth.
El director de Marcos es el profesor Marcelo Melo, también de la USP.
Yep. Cyclothone is reputed to be the most abundant vertebrate on the planet. Oh,
what was that?
off to the left.
Something big.
Something big.
Looked a bit like another galaxy size.
Like another.
Could have been one of those big larvation noses. But I think we drove by it too fast. Thought it was foul. Yeah. The wind put them in fluid with water from.
See something
I always find it interesting to think about that fluid mechanics in the ocean are make so much sense to us, but fluid mechanics in air are the same, just different viscosity, right? So you can translate those things in your head pretty easily. Maybe a little bit more forward as we go down.
Picture would be just you done.
This is from us earlier.
Ah, here.
Yeah, the frame grabs. I'm just looking through. Nice.
Is there somebody else studying how this animal can do this the spiral like this position? Is someone studying that?
Yeah, I mean, we see it. It's almost the same. It's the same in many cephalophores.
Yeah, in many.
They do this special shape
But you believe they have a way to sense that they are in that position? Is there someone studying that?
I don't know. It would be probably Jack.
Yeah, something like that.
Oh yeah,
Right?
Oh no.
that is a pretty substantial house. If we could scan this, there is definitely somebody home. You could see the tail and they're pumping. It's a really odd little house. Super weird little house. Lobes sticking out. Different directions. So let's get some video of this first, and then it would be awesome to keep the idea.
Larvation is a tunicate, just like salps.
Yost, you're gonna want to DPIB this, yeah?
Yes, please.
Ahora lo que estamos viendo en pantalla es
So
una larva de
one thing that the
tunicado
DPIB
y la
It's not
idea
very
es
big.
también escanearlo y poder conectarlo con el ROV.
It's big enough to scan. It's not huge, but yeah, we're setting up. In process.
Do formato de um girino. Do sapo. Larva de sapo.
Mhm.
corpo dele é só essa parte.
It's a fine mucus mesh.
Uma cauda que bate ali. Ele secreta
Otherwise, you're the snot. Snot.
uma casa de
Tiene como forma de un renacuajo, pero lo que es realmente el organismo es muy chiquitito y él secreta su casa alrededor y va creciendo. Esta casa es hecha de mucus. Y una vez que está llena de partículas que lo ayudan a alimentarse, la descarta y crea una nueva.
What's that?
Acá me dice Renato por cucaracha que se llama oicopleura.
It doesn't matter because this is what we're recording.
Capaz le voy a pasar el micrófono a Renato para que les explique un poco mejor.
Hola a todos amigos de Argentina. Eu não sei falar espanhol, vou falar em português porque acredito que são duas línguas que podem ser entendidas se falamos despacio. Então, é um animal que produz uma casa de muco e esse muco agrega partículas e esse animal engole, come as partículas e quando essa casa se queda llena de partículas, o animal sale de la casa e produz outra. Então, é um processo que ajuda na coagulação de partículas nos oceanos e essas partículas que ficam aderidas à casa de muco descem para o fundo dos oceanos. E assim, a oicopleura ajuda num processo que é o afundamento do carbono, um carbono da superfície que vai para o fundo dos oceanos. Então, é um animal que tem um papel fundamental no sequestro do carbono atmosférico e no armazenamento do carbono no fundo dos oceanos.
So this foundation is probably like a floor, but it's got a slightly different house than what we typically see for the standard ones. It's slightly different group. The animal is shaped like a little tadpole. It sits inside this filter that it basically sneezes out and then pumps up. If you put the mucus that house is made of in a scanning electron microscope, you can see the fine mesh and measure the size of the mesh. They have different size meshes in different parts of the house. And we use that mesh in the house to pump water through the house to filter out just the right size of the
Não dá para escutar.
the shapes of the houses and the way that the water, the way that the water can flow through seems to be specific to different house designs for different groups. And one of the things that this DPIB is amazing at doing, that we do in the lab.
Sabe o que eu tava pensando? A gente combinar, nós combinar de todo dia 6 horas da tarde ou 7 horas, um horário bom, direção diferente, dominar aqui, ficar falando só em português ou espanhol.
Pode ser, quer trocar aí?
Porque daí a gente estabelece e divulga, entendeu? Daí a gente combina a turma aqui de 6 e pouco ou 7 horas.
Ficar serrivômeno aí, que legal. Eles vão serrivômeno no dia.
Só espanhol.
What we do with these houses is after they're particles, those particles are aggregated and everyone leaves its house, which some groups where we've measured, they build several houses.
Acá el profesor Renato y el profesor Marcelo van a combinar un horario durante las inmersiones del ROV para poder explicar todo sobre la ecología y la biología de los organismos pelágicos. Tal vez en algún momento, en alguna hora, como sería las 6 de la tarde, 7 de la noche, en donde como estos organismos son migratorios a lo largo de la columna de agua, tal vez en estos horarios es más fácil de tener más colectas y más visualizaciones. Y ahí poder explicar mejor.
So libations, what we're seeing is an adult. This is an adult. I don't know what, I don't actually know. I'm not the person to ask though, what the larvae of libations look like. But the ones we see and the ones we see are adults. And once they've aggregated all of those particles on the house and they leave the house, when they're not pumping the water
Hola,
through
Sancas
it, the
Zad.
filter
Los
collapses
larváceos
and sinks
no son
very
larvas,
quickly down to
son
the
adultos.
bottom of the ocean.
Pero
This is an iframe inside of the what you told you, and then if you click on the left tag, you get what's actually inside the iframe. But you don't need to ever close this window because you could be looking up and capturing that. Surely
lo nombre es larváceos porque asemelham-se a larvas, pero son de un grupo que se llama Larvácea y es muy acerca de los vertebrados. Son cordados como nosotros, pero son invertebrados. Y lo producen el muco todo el tiempo. Cuando le queda su casa se queda llena, salen de la casa y producen otra casa. Y la casa vieja se afunda para el fondo do océano. Portuñol.
Acá lo que Renato nos estaba contando, como bien todos pudieron entender, es que estos larváceos es el nombre del grupo y ellos secretan este mucus a lo largo de toda su vida, que los ayuda a captar partículas que les sirven para alimentarse. Una vez que este mucus está lleno de partículas, descartan esa casita, ese mucus les sirve como casa y producen otro nuevo y limpio.
Yeah, moving on.
So line that up. Look at this camera here. And then we zoom in on the side region right there. So, and then also you can see it sometimes on this monitor. Like when you're doing that with PIV. You have the red line of the laser, so you'll see it. And then just get the sweet spot. So if you have it in the middle of this monitor, and then when you're looking for so you can also line it up with this.
They mentioned popcorn in the feeds. Totally made me. The what? We've seen several Rizeria. Not the big fluffy one. We saw a couple. They were small, up shallow. Thank you for the popcorn.
That goes into the try getting get taken, you don't need to give it all subject. So it comes in. Stuff like and stuff. And what the people you can for the time.
Really wants to come on.
Yeah, when we're flying, she wants to come on.
What is that? Yeah.
And then she will
Take over. Thank you, Sunny. That was awesome. Take two. Take two. You're in the hot seat. Yeah.
whether we could like everyday house people like presenting games maybe important.
Yeah, you can talk to them anytime you like in Portuguese.
Yeah, but nobody
Oh, to tell them like regularly.
Yeah, regularly.
Sí, Sara, estamos
There's some
con pochoclo
predictability sometimes that
acá en el
it's not-
estudio.
Yeah, we can't guarantee we'll be in the water at
Yeah,
5
usually.
o'clock every
You
day.
can say
We should be
usually.
though. You can say we're aiming to be. Mhm.
You
Yeah.
were hearing us?
No, it's great.
Great.
Oh my goodness. That's my glory moment.
Just nice and slow.
Yeah,
My
little
friends were
forward.
saying to me that
Not too
now
slow.
today
We're trying to make
have
it
some fun.
down to 1200. We'll do most of our hunting. With beautiful things to video on the way back up, but enough forward that we can see what's going on. Can we maybe bring the camera? Or are we zoomed? No.
I had thought that there
And we might need a bit more light.
far away on the right.
And then just slower. That's good. Yeah. No,
Or we
that's
can
nice.
switch and do uppers.
No, this is nice because we're headed down, so it lights it up before we get there. We still get nice contrast on the top.
Yeah, I thought
That's good. So enough forward that we can see fresh water. We don't get this stuff streaming past the screen this way. We're kind of more coming, more, 80 screens coming into it.
Bueno, a las 7 horas por acá, por-
And just exactly driving in the snowstorm with enough speed that we can still stop if we see something with enough warning. Obviously, the little tiny stuff that we see at the last second, we won't be able to stop on. But
a las 7 horas por acá, por el canal del streaming, el profesor Renato y el profesor Marcelo van a estar conduciendo la live. Así que todas las preguntas que tengan las van a poder hacer en el chat y ellos obviamente van a responder. Ambos dos son brasileros, pero se manejan bastante bien con el español.
they do. I think my shift is over sometime soon.
Worms are
maybe in like an hour.
Ketignaths?
1 more hour.
we're 3 hour shifts today.
Oh. There's something nice. See this sparkly thing here? We could try to put that in a piece.
Oh, what's that?
It's a foraminifera.
Oh, foram. This is a good one. Please be good
Nice.
video.
That's our area, right?
Yeah.
Let's go.
Bueno, lo que estamos viendo en pantalla ahora es un pequeño foraminífero.
No, we're gonna try to put it in a D-sampler.
Gentler.
Van a intentar captarlo. Manu, que está conduciendo uno de los programas, está pidiendo para que sean gentiles porque después quiere escanearlo en su propia cámara.
Marcelo, do you use Squidle? Any of you use Squidle?
I have not.
The button is broken. I cannot hit
My wild guess. Are you clicking here somewhere?
Oh, that one's.
No, I'm your sister.
That's red flags. Oh,
No, I put the red flag because it was just a test. This is all good. I can go in, I can complete this.
It's right up front of the jar right now.
Arm is.
Okay, so it was doing all of these clicks while I was.
Yeah. All right,
Okay,
I'm on the
I think
outer
he had
jar.
said it.
Sorry.
Okay, so now I need to annotate.
The one by
So
the-
go annotate.
Yep.
I click here.
Kind of bumped up.
I got that. Now I go in.
think it's in.
It looks like it's in.
Close. You have close. You got it? No. Not that
No,
one.
wrong
We
one.
got it.
Oh, parece que
The other jets.
lo capturaron.
Wait, it moved a little.
No, todavía no.
It was a fabulous catch.
Para quien estaba preguntando,
starting.
los foraminíferos son protistas parecidos con las amebas. Forman parte del plancton también. Construyen unas pequeñas conchas alrededor de ellos de carbonato de calcio, como lo hacen algunos caracoles o mejillones.
The gentle nose. Very, very gentle.
Alright, next time we'll aim for the other one.
Only the very slow.
Estamos intentando ver si lo capturaron o no. Parece que no. Es bastante difícil porque son súper delicados.
Yeah, we'll try this one.
Okay. Alright, so kind of just one D today. This is one, that's two.
Number one, okay.
Well, that was beautiful though.
Karen, what's
We
the
can
word?
do it.
Did we get it?
Doable.
No. T sampler malfunction.
Let's make a little note of that. So what's going on?
That was very nice flying.
I was really excited.
Those guys are hard to see. Yeah, that was awesome.
Karen está diciendo acá que estos organismos son muy difíciles de ver, como pudieron ver en la cámara, casi imperceptible. Entonces Manu tuvo el reojo para poder identificarlo.
Did you see something?
I thought I saw something like shiny briefly, but it
Okay, this is the deepest we've been disturbed.
It is.
It's too much.
I think it's okay. Because we're only going to stop on big things, so we should have enough warning. The direction is perfect, right? We don't get that swirl in the thing. You can see just enough forward that you can tell you're moving down, but you get fresh water all the time to look at instead of
Yeah, that one I
it
was
washing
going down
around.
too much.
This is perfect. So what are you controlling versus what is the other pilot controlling?
I am.
You're flying, obviously.
Yeah, and
Oh. Ooh. Old.
Karen, what do you
House,
think
maybe?
that was on the left?
I think it was an old house,
Old house,
potentially.
Manu estaba preguntando qué cree que Karen es.
There it is.
En la izquierda, ahí lo estamos viendo en el medio de la pantalla. Podría ser el resto de una casa de alguien, tal vez de un larvacio.
And it depends as far as like cameras.
Now it's working.
It's working now?
That's it.
Was it like the min max maybe?
No, no.
Oh, good. It's working. Great.
Yeah, so who was determining where the camera is doing suction?
So the suction, I think, we're basically kind of controlling it.
cameras, it depends. Sometimes, like, I'm able to move them, like, when we're trying to line it up.
Yeah.
But sometimes if I'm really struggling to study the
Yeah.
vehicle, I'll ask him to. A lot of times, like last night, when I was co-piloting, I would do the zooms and the focus.
Mm-hmm.
So it's really difficult, especially with the
It
curves
is.
and
Yeah, we
everything
usually
for
do
pilot.
we usually drive the camera and do suction and the zooming and focusing when it's everything's moving and moving in relation to each other is like really a challenge, especially when you put new people in the seat and stuff. Focus, try. Sorry, it's okay. Yeah, go ahead. We're trying to focus and go down. It's just we've seen a few really cool things, so we stopped on them and made it through iris,
Bless you guys.
DPIV, and collection on a single animal, which is excellent.
We watched a section.
Yep, the big side or the little side? The big side, yeah. Be interesting to see what the shadow cam gets from that because there's so many pieces in there at one time. It might be a little bit of a mess, but yeah. Yeah.
You know, can you read the question about the things that you really want to follow? Where do you think that high depth is?
I'm told there's a question in the
Second one up from the bottom.
okay.
Any of the bands? I think the upper, do
So
you think
we
that-
are looking at broadly at biodiversity of midwater animals out here. We have experts on many different groups. So one of the objectives is to image the large, delicate, gelatinous animals that we can't collect and bring into the lab to study their structure or to image the mucus houses that we definitely can't collect. Oh, what's this? That we definitely can't collect and bring into the lab. So we really want to focus our work on what we can do in situ without disturbing the animal too much. What is that? It's a funny little larvation house. Nope, funny little larvation house. Almost looks like a fritillarid.
House?
It is, yeah, fritillarid house probably. Pretty small one. So we have a larvation expert along. That's what we're looking at now.
Respondiendo a la pregunta de Mati, la idea principal obviamente es intentar buscar especies que todavía no se conocen en esta región, a estas profundidades. Y una de las ideas de usar estas cámaras súper potentes y ultra tecnológicas es poder sacar fotos de estos organismos que a estas profundidades quizás al ser llevados hasta la superficie no mantendrían su forma original. Entonces estos escáneres permiten tener una mejor definición y permite estudiar o descubrir partes que no se podrían ver si los organismos son sacados del ambiente. Estas cámaras fueron instaladas para poder ver los organismos en su hábitat.
Let's do it.
Try
We're
and
gonna
put it
try to
in
put it
the
in D sampler.
sampler.
Either one or two. It's your choice.
Ahora le voy a pasar la palabra a Marcelo, que va a poder explicar muchísimo mejor lo que estamos haciendo. Marcelo es profesor de la Universidad de São Paulo y es especialista en peces.
I thought I saw the animal, yeah. But I thought it was sitting still. So the head.
You saw the head?
Oi,
So
Mati,
one thing
tudo bom?
before we
Aqui é o
go
professor
to-
Marcelo.
get a quick-
Eu trabalho com peixes, eu estou aqui no cruzeiro para atrás de espécies novas, e estudar também a diversidade. E um dos objetivos nossos é justamente descobrir essa diversidade. A gente vem equipado com redes de pesca, equipamento de sequenciamento a bordo, e aí um dos nossos objetivos é justamente buscar essas espécies que ainda não são desconhecidas para a ciência, ainda não tem um nome, e aí a gente descreve elas formalmente, dá um nome para elas, e a partir de então elas ficam catalogadas E aí outras pessoas podem estudar outros aspectos dela. Oi, Fernando.
Suction or the samplers?
To use sampler. Either way, this stuff is part of it.
Me quieres romper, ¿no? ¿Quieres ser el Martín Fierro? Quiero mi momento de gloria. ¿Quieres ser el Martín Fierro?
Okay. Challenging with the small things because you're so easy to lose. Being close and
What do you think?
lost within.
No, that. That makes it better. And then
Eu tenho uma entrega. Vou deixar meu.
A gente pode revezar. A gente pode revezar. Se quiser sair um pouquinho, se quiser sair, por exemplo.
Sim, não, eu vou ficar aqui.
Fica aí, então.
Sugar, do you
you
want
can
that?
just start typing.
Sim.
So many things in there.
Snack?
You can also type in Japanese and it'll come up.
That sparkly little fish looks like an anglerfish, or not an anglerfish, a hatchetfish.
How's it going?
If it's not selected,
Exactly. So the circle.
Any
Yeah.
good
A little bit
questions?
bigger.
on the right,
Vegetarian.
Yeah.
you see.
Several
We
questions.
saw.
They ask about what you're doing here, you're discovering new species. It's a really good one.
Now if it's done, it's saved.
I think capture, I need to have like a touch screen.
Okay.
Oh, dragonfish.
Oh, is that true?
Dragonfish.
Yeah, come here in the bottom left.
That's an
Do I also need to hit save
Dragonfish?
here again?
Dragon or a viper or something. Let's take a look.
or mystery fish.
Same you?
Or viperfish?
Oh, that's look like a viperfish.
Coliotus.
Oh, pretty
Meliodas.
That's
Wow. I'm gonna delete this.
Cauliodos.
C-H-A.
Esse agora é uma espécie de peixe-dragão, família Stomide. É um cauliodos, dá pra ver o primeiro raio modificado. Ali na ponta tem uma isca que ele usa para
Fancy imaging with it.
Tell the species
Pra
to look under the eye.
capturar as presas. Esse bicho tem, essa espécie tem dentes muito grandes, ele usa para pegar as presas também.
CAL,
A little bit further down.
I think.
E muito legal que deu para ver direitinho os fotóforos também. Então, o padrão de fotóforos são esses órgãos de bioluminescentes que estão na parte ventral.
No. But if you add a comment.
Eu sou, Fernando, eu sou biólogo de formação, mas eu sou professor no curso de oceanografia da USP, na Universidade de São Paulo.
But much more interesting is top left. Round about
Então, se você está interessado na área,
a third to the left.
dá para tanto esta biologia quanto a oceanografia. E Mirante está ao vivo. Um grande abraço do amigo ictiólogo.
Sim, isso chicarita. No, lo hacemos los días.
¿Es cuando? ¿El jueves? ¿O aquí? Ahí lo vemos ahí otra vez.
Sim.
Mirante é um ictiólogo lá da Argentina.
Ah, é da Argentina?
É.
Ah, ele falou mais cedo. Disse que
Sim.
talvez ia te conhecer. Ok. Ah, viste que había una pregunta si éramos casados.
something in the center of the screen. Capture you want as close
You
as
see that dot heron?
What's that?
That one they're interested
Yeah, yeah, the one that we're zooming in on.
Wow, it's a beautiful color.
No idea.
Puede ser nada.
It's an odd little thing. Is that one of the blue sites? Dougal.
No, it could be Jim Prep. I can't see any canals or anything in it.
Is this even a jet?
Yeah, it's a genocide wonderful.
That's my win for today.
Maybe we're kind of far away from it, yeah. Yeah, okay.
Take it.
It doesn't get any better than that. I've peaked.
Possibly Jim Laprea. It's a little blue site.
Oi, Shahar.
Color is really nice.
Okay.
There we go. Nice.
It looks like a single stem unit of
Yeah.
something.
All that's left is one piece.
Hi, Shahar. There's a there's a team of researchers here from different areas. There are biologists, there are oceanographers, there are engineers, people that works in biochemistry, and so we have several different majors here, and of course marine biology as well. But for example, myself, I consider myself an ichthyologist, which is a branch of the zoology.
We've used up today so far 2 of them.
And we all got to interact to get better results. There is also one physician aboard.
How about the comments with the SOI highlights? Not sure what it is, but it looks good.
The left is
Yeah, don't.
So it doesn't leave the trace here. When I'm done, that disappears. Yes. Take it up.
So sometimes you see things from distance, and we can only know what it is when it gets really close. Fish, for example, it's very easy to get confused with shrimp or squid that pass really fast. So only when it gets close to the camera, that's when we can actually see what it is with precision. The
Well, that was quick. After telling you an hour ago, yeah, she looks horrible. So sick. That's a cute picture. She
Go to my room and cry.
She looks horribly sick, Connie. Yeah, I think that's. I think she's a goner.
5 minutes from yeah. I wish I looked that good when I was sick.
Right.
Oh, that's beautiful. So in this frame, I didn't know the photo. That's okay. Yeah.
So you won't from them, so you can hear.
It'll be a bit.
The little brown thing.
I just focused on that screen, it went much better than trying to look around.
Just on this one.
Yeah.
Yeah, because every time you, with something that tiny, you take your eyes away, then it's really hard to find it again. This is, maybe I have to tell myself, don't look away, don't look away. Don't blink, just keep focused.
Yeah, okay, so I'm not.
Keep an eye on it. Oh, that's a good
See.
Did we get back to Quintin?
So no suction, just
Yeah, just keep going forward, which is why we really can't stop on these things.
Oh,
If you want it to go in the bucket, there's no stopping for a moment.
okay.
Sorry.
Y pedí para aumentar el
It's not
audio
in the
de
bucket.
Karen
It possibly went down the tube.
para que las personas que hablan inglés puedan estar escuchando en vivo lo que están decidiendo.
Already out of my. Yes or no?
I just zoom in and see it like go.
Yeah, we're gonna see it in the bucket, but let's just go for a bit because it does take a long time to get through all the things we
Even
have
that
back
large,
there. Yes,
that
yeah. I was surprised how long it took for that. Oh, nice Tinapor, so big Tinapor there. We're driving forward, creating motion to gently move the little blue siphonophore piece back to the bucket. What's up? Gymnoprea? Is that right? Gymnoprea? Dougal?
Yeah.
I don't know. I don't
Potentially
understand your
Jim.
problem.
Dougal.
You just have to know what the spelling is.
Gymnoprea? What was the name of that one?
Possibly, yeah. Possibly,
Oh,
yeah.
okay.
Could the, one of them come? Well, Gymnoprea is not supposed to have bracts. It's not supposed to have chlamydia. That was a chlamydial unit.
Yeah.
So it's not
So it's not
currently
Gymnoprea.
described species of it, but it could be another gene, another species.
Okay.
Yeah, in the same gene.
Glad it's working.
In here, I had just written single stem. Single.
Sorry, what's up?
Like, I just written single stem units.
Yeah.
Yeah, oh, that was good. Okay, annotate.
And the blue.
And I can't type shrimp here.
Because that's kind of it's an unusual thing. There's some green ones, very few blue ones.
And that's all we can say, right? Poliokamata? No.
Almost 750. We'll keep going down.
I'm doing
Keep going
this
down?
tonight.
Yeah.
Gotta get.
So you want to go get coffee? Okay, yeah, I think I'm comfortable right now.
No sign of it in the bucket
It's
yet.
the names.
Not that I've seen.
I think it's the detail. I can do the high level.
I'll be honest,
But
I'm
the
gonna come
details
back.
matter.
Yeah.
And especially
Over there.
And it's you kind of have to catch it when it goes, right? Then it's like
Se não
up
me
to the
engano,
screen
tem
or
duas,
whatever.
três
It's very hard to
espécies
see.
de
Can we
caulinos
have a look at
listadas
this
aqui
guy?
para a região do Atlântico.
How's so how do you tell
Ah.
So we're gonna want to DPI this. So oikoplurid.
It's a larvation. I think it
larvation,
might
yes.
be a fritillaridae, but we gotta get in once.
If it looks like a hammer, like you see the body on the front and the back of the tail really clearly, then it's a fritillaria usually.
Oh, but it's in the recently annotated thing.
For fritillarids, usually their houses, the filters kind of, they always look to me like a pair of like angel wings. They're kind of curved with a little bit more space in between them, and the oiks generally look more rounded, but that's based on not having good catalogs of their houses or the animals necessarily, because we're just with DPIB able to catalog the houses because they don't make them in the lab without a fancy spinning tank. And because the animals are super delicate, so getting them in good shape so that you can actually see what's what is challenging. But Russ is one of the few world experts. He's described a lot of them. Oh, there's a rhizome.
If you do get them into a chrysal, do they build their house?
No, not necessarily. If you put them in a big tank that is slowly spinning around them, they will.
Right.
So
But Karen, there's a couple of species in Norway now that they have a culture for.
Yeah.
It's super nice.
Yep.
Oh, something on the top.
And a lot
Oh,
of the
it's
shallow guys, we're setting up for stuff, a scan. So a lot of the shallow guys will build houses in the lab, no
Yeah.
problem.
They're
But these
making like
deep
a
guys
house
are
a
hard.
day.
Look at that
beautiful
shrimp.
Annotate.
Yeah, and then the best thing to do is
This can get addictive. Okay, decapoda. Just takes a few years and that's it. This is the beauty of science, you know. Just everything can be learned. That's it. You just put in the work. I don't know why we don't tell kids that. Rather than saying you're smart and you're
Respondiendo
not.
a la pregunta de Bárbara, sí, podemos pedirle a los chicos que controlan el YouTube. Actualmente estamos en aguas internacionales, afuera de la zona económica exclusiva de Brasil, más o menos a la altura de El Salvador. Ahora están diciendo acá que estamos un poco más arriba. Salimos de El Salvador y estuvimos navegando y estamos más o menos a la altura de Alagoas, en Brasil.
Oh, that was beautiful. Scanned. You want to put that screen there? If you, this is, I mean, this was just a test case. No, you can do that now. Yeah.
There's no big ones.
So what's your camera resolution? Sorry.
It's a larvation.
Huh?
It's a larvation.
Yeah, larvation.
There's gotta be some of them.
I think it's the same one.
Something about there not being larvation confirmed. You ready?
Whoa, good house.
Well, little house, but house.
There's two. Yeah, you're right. There's another one. Like that.
like that was framed.
It's that little house is just so good.
What camera are you seeing? What camera are you seeing it look big in?
Oh, the black and white with the depth.
Oh, okay. You here to sit? You here to sit?
Yeah, I had a good sleep.
Great.
I'm all ready to go and be my normal obnoxious self. Sorry, entertaining self.
Hot mic.
We can take a vote.
Full of character.
Yeah, character. Yeah, I just wrote it.
Ahora en pantalla estamos viendo un pequeño copépodo.
Professor at University of Alaska Fairbanks. He is an expert in zooplankton in general. Has an incredibly broad knowledge of different midwater groups, and particularly interested in libations, which happens to be what we were looking at. Right? Too small?
I think it's too small.
Okay. All right. Moving on, and I will hand you over to Russ.
Where did Russ go?
I think he starts in 20 minutes, maybe.
But just kidding.
Never mind.
For most recently used ones, because you're doing the same.
We lost Russ.
Yeah, no, it does. But
And then departed.
oh, Marcelo.
Where did he go?
I don't know. Sorry.
No.
I couldn't see. No, I just saw a tiny dot. I know it has antenna, and that's all I can
Sé que
say.
están preguntando acá el nombre del copépodo, pero es súper muy difícil de saber la especie así a través de la cámara sin verlo en microscopio o haciendo análisis genéticos. Solo pudimos identificar un pequeño puntito que sabemos que es un copépodo.
Not of the larvation.
No, the larvation we don't know yet, right?
It's probably Fibrillaris.
Fibrillaris. This is nicely adjusted springs. Good for both sleeping and breaking the neck.
Right before it died yesterday was definitely one of those stories there. We were just getting ready to go to the water, but I was just waiting for like 2 minutes, and I'm pretty sure, 2 minutes, I'm pretty sure that someone said something to me. And then he said it to me again. Oh, okay. Here.
This is so
like the look I got from did you forget what I just said?
Definitely find
Out for 2 minutes.
You were in a flow state.
I was gonna say, what are we scanning? I don't see it
Just
anymore.
a little tiny guy right here.
Okay.
Just on the inside flow state.
Goes
Ah.
through so fast because it's so
You know, even just
Okay.
1 or 2
Let's-
hours of sleep feels so good.
Let's move
I
on.
don't know why.
Yeah,
It just
too small.
feels so good.
Yeah.
All right.
Nap.
Great
I was
practice,
feeling
though.
miserable. Oh, what is that?
All right.
Down below? No, down
Down
below there.
and
Yeah.
forward.
Oh, it's
Yeah, we're not gonna scan that
Was it deflated? Yeah.
Let's find something bigger.
We will continue.
To
There
the
we go.
depths
So
and
we're
beyond.
at 740. We came up a
What is
little bit.
Bud
We lost a
Light
little
doing?
ground on that one.
Yeah, to infinity and beyond. We need the inverse of that. To negative
Right.
and beyond. Oh, arrowworm.
You're not supposed to be here for 15 minutes. How about we swap now, and I will come and spell you something at
I still the plan would be.
Yeah, and we're still heading to 1200. We made pretty decent progress. I mean, we've collected 2 beautiful, awesome animals. We've done iris, DPLB, and collected one of them, and iris uncollected the other. We actually
Los larváceos, en teoría, ellos tienen reproducción externa, liberan sus gametas al agua, donde se produce la fecundación. Esa casita que ellos van construyendo de mucus es solo para alimentarse.
We're at 7.
Galaxy size, and then there's ACN 6 and 7, 6 or 5 and 6. We're in 7. We have the small mesh on those 3.
Okay. And what do you want to put in those?
Whatever. If we get clouds of hyperids again, it would be
Try
great.
that.
So part of the reason they found that one of the hoses had disconnected a bit, and so we had suction. Not a lot of suction was happening the last half of the dive yesterday, which makes it very challenging to get things down the hose past that open spot. But it's all taped up and good today, and we can totally tell the difference. Things are just like moving through really nice. Although there's a lot of hose, so it takes a while.
Yeah.
For
I
things
saw that
to
was
make
taking
it back.
Okay.
Yeah, so that's pretty much it. Work your way down and see what you can
No, de nada, y gracias a ustedes por todas las preguntas. Cualquier duda que tengan, acá a bordo hay un montón de especialistas en organismos de zooplancton y de mar profundo que podrían responder muchísimo mejor todas sus dudas. Hay investigadores de diferentes partes del mundo, entonces la idea de la dirección es que todos en algún momento puedan responder preguntas en su idioma original o su lengua madre para alcanzar todas las regiones. Minha nova vida, trabalhando na rádio, Oceano da Fúria.
Monitoradora. E se alguém tiver perguntas em português também, eu tô aqui para ajudar e poder responder também.
How's it going?
Awesome.
People liked, I think, the translation. I don't know what's going on with the chat because it pops up in English and then it switches to Spanish and then it pops back into English.
I think it's a translation.
So it's like automatically translating it?
Yeah, everything that's has this icon
Yeah.
in Spanish then it's been translated by automatic for us.
Oh, so it won't show up in Spanish. Or it shows up in Spanish for a while and then it changes.
I guess that's how long it takes.
Okay, because I was like, well, I would look up and I'd read it in Spanish, and then a few minutes later I couldn't find it again, and I'm like, where did it go? Oh, wait, there's the same question.
But we got some very good questions.
Yeah. Yeah, it is. Yeah, there's some good questions. Which is nice because, you know, you can only sit up there and talk about so many different things, right? So the questions are a good way to be like, oh, people would be interested to hear about this or that. So it's nice to have those.
Polvo ambiental.
Yeah. Cool.
Ese polvo que están viendo, o esos puntitos blancos, en teoría se conoce como nieve marina. Está formado por detritos, polvo o partes de diferentes organismos, y una vez que mueren también los organismos, parte del fitoplancton, del zooplancton o pequeños pedacitos, va precipitando a lo largo de la columna de agua y sirve de alimento para otros organismos que se encuentran en profundidades superiores y no tienen posibilidad de acceder a la luz o hacer fotosíntesis. A esas profundidades, como las que estamos ahora, más o menos cerca de 800 metros, la luz solar no llega. Entonces, la mayoría de los organismos para vivir se alimentan de estos pedacitos de otros organismos o de detritos que forman la nieve marina. Y es también una forma de reciclar el carbono dentro de los océanos.
Might be a heteropod. If you could zoom in on
Heteropod. That's a heteropod, yep. Definitely.
That was my victory in that.
Sí, es increíble cómo esta lluvia sirve para alimentar todo el resto de los organismos que están en profundidad y en el ventos también. No solo es súper linda, sino que también es bastante nutritiva y muy útil para el resto de la cadena trófica. Ahora en pantalla estamos viendo un ketognato. También a veces se los conoce como gusanos marinos, me parece, si no me equivoco. Son carnívoros principalmente.
Micronecton. Micronecton is the small fish and shrimp that are not fast enough to go against the currents, but they swim more than normal plankton.
He's moving. We're good.
It means very small, swimmy things.
We are okay. Yeah.
Estos organismos son del macronecton, me están diciendo acá. Solo cumplo mi papel de traducir.
I don't know what's happened.
Obviamente todos los organismos del zooplancton no tienen una capacidad muy grande de natación, pero los que pertenecen al macrozooplancton o al macronecton pueden moverse un poco más que, por ejemplo, los pequeños copépodos o las células del fitoplancton.
Super fast. These guys are very fast at eating. They eat lots of copepods. They're a very big predator in the midwater column. All right, we're good. We can keep moving on. Thank you. Later, dude. Hi, everybody. We have switched watch leads. Karen Osborne has done a fabulous job starting us off today. I am Heather Judkins. I'm with the University of South Florida, St. Petersburg in Florida in the U.S., and I am excited to take over for the next few hours here.
I'm taking over in the... I'm thinking of getting a new one. I'm in a plutonic relationship, but it's really shallow. Okay, squiddle me this, squiddle me that.
So, when we see something come along here, we'll hit the capture button.
We hit capture button, yeah. Well, let's do that right now.
Let's wait until there's something in the screen.
Oh, we could just say it's crap.
Sorry, am I going too late?
No, that was good. It's back up.
Oh, there's that too.
There's something.
Salvage.
It's okay. I should have hit sooner. That was it. I don't think that was it. Okay. If we can go just slow for a second forward.
Okay, ROV.
What's this guy?
Yeah.
All right.
Grab that
so
guy.
now I
There, I guess.
I have to click it first.
Click it
Sorry
first,
about that.
activate
I need
the
to give you a
window,
little more
and then
heads
click
up.
it twice in rapid
Ah,
succession
sí,
of
esos sí
the thing you want to animate.
son bien bonitinhos.
I did.
Hey, is this a larvation without his house?
Yes.
Yeah, it is.
Do you want to zoom on this?
Lo que estamos viendo ahí en pantalla es un larvacio como
It
los
is
que
very
vimos
long.
antes, nada más que
So this
sin
looks
su
like it's a
casita.
larvation without its house.
Can we get it closer to the head?
Yep,
Ya
if we can
ha
get a zoom, that'd
estado
be great, pilot.
súper llena,
Yeah,
se
he's definitely
descartó
moving.
y actualmente va a tener que empezar a construir una nueva a partir de mucus
Part of
para
his visceral mass in his
captar
body and his
partículas
head.
de detrito y
Everything's contained in
alimentarse.
his head. And then literally tail is just for propulsion.
Alt seed window over
We're
here.
trying.
It's a fritillaria.
Take your time.
Fertile area?
Yeah, the tail's broad, the head hinges back fairly far.
It's because we haven't
Definitely moving.
I haven't hit A for annotate.
Yeah. Now you're in the annotation thing. It's one of the list of animals comes up over there, you're in annotation.
Russ, are you
en su
happy with this?
mayoría, organismos
Good to move on, or would you like a
es
zoom?
decir, que no dependen de la luz solar para crear su propio alimento, como quizás células fitoplanctónicas, y permanecen flotando hacia la deriva
So,
siguiendo
continue?
las corrientes. Claro que algunos tienen una capacidad un poco mayor de movilidad, pero en su mayoría dependen de las corrientes.
Did I mistype it?
No. Oh. Literally, I'm going into the section. Oh yeah, if you see that again, yes.
Oh, okay.
But in general, I don't mean with this guy. I just mean in general. But yeah, that would have been too funny. Opportunistic.
All right. Yeah, I had trouble. I didn't realize
see him in
that was
this
a
one
close
yet.
button for the dialogue. Okay.
It's too funny. It's all good. We're good. We'll come up with some others. That was so funny. I'm like, wow. He just wants to come on board.
Yes.
Nope, we're good.
For science.
Mm-hmm. Oh, that was a larvation house.
Os peixes lanternas, eles fazem parte do microplâncton, do microzooplâncton. Eles conseguem nadar bem, eles fazem a migração vertical diária. Eles não são considerados zooplâncton por causa disso. Eles têm um tamanho já suficiente para nadar de acordo com as vontades que eles querem, né? Mas eles também sofrem alguma influência das correntes. Assim como se a gente cair na água, por exemplo, no mar, essas correntes carregam a gente, mesmo a gente não fazendo parte do plâncton. Então, eles não são considerados zooplâncton por causa do tamanho deles e essa capacidade de se movimentar no ambiente marinho.
Middle stop?
Mas é bom lembrar também que os peixes também têm a fase de larva, que aí sim fazem parte do plâncton. Então, os jovens, os peixes lanternas recém-eclodidos, eles são larva e fazem parte do plâncton.
Yeah, we're looking at a zoom. We're working on it. There you go, you guys.
Ahí tuvimos una gran explicación de Marcelo sobre los organismos que pertenecen al zooplancton. No solo también los peces, varias especies de crustáceos tienen una fase como larva que forma parte también del plancton. Ahora en pantalla estamos viendo también otro larvacio con su casita alrededor.
This is like a Mesoicoplura. It's got these kind of clubby extensions on the ends of the arches of the horse collar.
Okay. Are you guys good with that? You want more? Do you want this?
Good. It's probably not more than a couple centimeters.
Only if it's near the funnel, then just go.
If it's opportunistic funnel, sure.
If it's not, then yeah, don't worry.
How deep are we?
7.
7 to
Yep,
50. Okay,
dental
that makes
section.
sense.
If it works, it works. If not,
Because Mesoicoplura is usually below 5.
he's high up there. No, he's not. He's low.
Yeah.
Low.
Deceiving.
Okay. Yeah, he's out there too. Got it.
The tail's all too wide for Mesoicoplura.
Keith Ignath passed by.
Adoro que Fernando ya escribió en los 3 idiomas para poder alcanzar a cualquiera de los que estamos acá para responderle.
You don't have Mesoicoplura in here.
No, he's not in there. I mean,
La corriente principal que baña estas aguas, pero por lo menos las superficiales, es la corriente de Brasil. Pero a estas profundidades lo que aparecen más son masas de agua que son formadas en otras regiones del océano, que está todo interconectado y obviamente tienen otras características. A veces cargan características del lugar donde se formaron. Entonces estas aguas de acá pueden venir, por ejemplo, de más al norte o de regiones árticas. Sí, no estoy equivocada.
No, you're right. Okay.
There we go.
Way up there. Okay.
Nested backward.
Yeah, so click appendicularly the green one once, and then click it back again. Now open it up again. Open up. Okay, and go below, open up again.
Alright,
Not
that's
not
it.
information, the
Below.
Cross. Yeah, there it is.
turn off everything, there's orchids every day. Political.
Oh, the purple, okay.
If you type down a leaf, then it only gives you the branch the leaf is attached to. So you have to go up and then expand.
See, there it looks, all right, right. I'm good. I'm good to go. You guys good to go on? Yeah? We did not get it? We're good?
So
Yeah,
does
let's
that
move
actually
forward,
take
please.
it, or I need to click it?
Thank you, though.
Bueno, parece que no van a intentar
Every time
capturar
we use
ese
the suction
larvacio,
sampler,
así que
it is
vamos a
always
seguir buscando
a challenge because
otras
there's so many
cosas
things
a
that
lo
the pilots have
largo
to
de esta
account for.
profundidad. La
They need to
idea
account
de
for
hoy
the
es
currents,
llegar
they
a
need to
los
account
1200 metros de
for the
profundidad.
strength of the suction, the position of the animal and the ROV, and the ship movements. So it's always a win when we can get one of these guys in a canister. Hi.
Hi.
Oh, that's fine.
Respondendo a pergunta
Yeah,
do
we'll see
Fernando
how
de
this
massas
goes.
d'água,
I was having a good
a
time.
água superficial mais quente é a corrente do Brasil, é uma água tropical, de água bem quente, ali em torno dos 20 graus até mais ou menos 200 metros. Logo abaixo dela está a água central do Atlântico, que é uma água já mais fria, em torno dos 14 graus. Essa água se estende de uns 200 metros até mais ou menos uns 800 metros, até uns 500 metros. E aí a gente tem duas massas d'água bem geladas no fundo, três na verdade no fundo. A água intermediária antártica, que vem da Antártica, a água profunda Atlântico Norte, que já está bem próximo de 0 graus, ela fica bem rente ao fundo, e ela vem do Atlântico Norte. E por último, a água de fundo da Antártica, que é a água mais gelada, também é uma corrente de água que vem da Antártica, né, que serve como um moderador do clima também. Então essas correntes de águas muito quentes, tropicais, elas quando chegam na Antártica, elas afundam, gelam, e elas voltam ricas em nutrientes e ricas em oxigênio. E cada massa d'água é caracterizada por diferentes temperaturas, diferentes salinidades, diferentes massas de concentrações de oxigênio. Então, com um equipamento chamado CTD, que desce coletando esses dados, a gente consegue caracterizar exatamente a massa d'água em que os organismos ocorrem. E alguns organismos eles ficam em massas d'água específicas. Então, se eles forem retirados desses ambientes muito gelados, por exemplo, quando eles chegam na superfície, eles acabam morrendo, eles acabam literalmente cozinhando em massas d'água mais quentes. Mas alguns eles são mais tolerantes. Eles conseguem fazer o que a gente chama de migração vertical diária. Eles saem de águas mais fundas, vão para águas mais rasas, né? E eles conseguem tolerar essa variação de temperatura ao longo do caminho.
Yeah, the tiny white. Yeah. Is that anything of interest? Scientists in the back.
Just a piece of...
Mas vale a pena fazer isso, porque a gente restringe as pessoas de entrarem todos os horários. Tem que divulgar postagem. As pessoas vão achar que a gente só tá nesse horário. Agora quer fazer uma foto?
Ooh, yes. What are you seeing that I'm not seeing? Oh, a larvation house. Excellent. Like the larvation? Maybe.
It was really pretty. For this.
You would have loved it. Thank you. Are we zoomed out on the science camera?
Yes.
Okay, thanks.
Pode ficar falando vocês.
Nope, we're good.
Perguntaram se a coisa era casada. Se ela e a Silvina eram casadas.
Mm-mm. Nope, it was a fish. They're all cool. Great. We are at 800 meters and we're continuing our descent down to 1,200 meters today. Our dive should go until around 2 a.m. local time. We're doing a 12-hour dive today.
Obrigado pelo elogio da voz.
Bueno, esta inmersión empezó a la 1 de la tarde, más o menos, y será más o menos de 12 horas. Entonces la idea es terminar a la 1, 2 de la mañana. Bueno, por mí ya mi tiempo acá con ustedes terminó, así que los voy a dejar con los dos especialistas, el profesor Marcelo y Renato, que van a poder responder todas las dudas que ustedes tengan. Ambos dos son brasileros de la Universidad Federal de Río Grande y de la Universidad de San Pablo. Hablan en portugués, pero bueno, hablando un poquito despacio en portugués o en portuñol va a ser súper fácil de entender para los que son de habla española.
Okay. We're good on speed. Yeah, this little guy in the middle. He is a... Yeah. Oh. We could zoom. Yeah, zoom in on the little red guy. Oh, it is a crustacean. He let out of here. It was a crustacean, so I can tell you that. All right, so we can keep moving on. Fish. Yeah, that was a fish.
I'm so glad all these years of education didn't do that.
A gente também fala portunhol aqui, Fernando. Eu hablo um pouquinho de portunhol.
Stop. Little guy up there. Yep, just above. Yep. Guys, middle of the screen. Russ, it's a Tina 4. I was gonna say Dougal just stepped out the door. We're gonna zoom in on this for a second. Yeah, that'd be great. Oh.
Yeah, so Sidipin.
Sidipin. Is this something we want to collect?
It won't survive anything but a suction, about a D sampler, so
So you think?
Kind of early to commit a D-sampler.
Okay, so no then. If we can just stay on it for a minute to get a couple of
try and get a
good
nice zoom in on
video,
it.
that'd be great. Oh, do you want to iris it?
Yeah, why don't we, yes, should we put it in iris?
Yeah, if we could get some zoom video, get a good eye on it. They won't do well if we try to collect them.
If we desample it and we come across something else that we want, we can always dump it.
We can always dump it, yeah.
So,
I'm okay with whatever you guys decide.
I want to make sure, let's hold on to it.
Did you want to scan it first?
I'm having a hard time deducing size.
It's kind of square in cross section.
It's pretty small.
Yeah, let's just collect it in the beach.
Try to desample it?
Yeah.
Okay.
Then if you don't want it.
Yikes. Sorry, team. All right, so we would like to get a video for a little bit, to get some good zoom video on this. And then if we can get it into one of the desamplers, that would be great. These guys are really fragile, so they don't do well going through the suction sampler. So we can attempt a desampler event.
Esse é um quitenóforo, esse é um bicho muito frágil. Então ele é bastante difícil de coletar. Ele geralmente, mesmo com esses coletores, são bastante delicados. Eles não resistem e acabam explodindo. É muito legal que dá para ver os cílios, né? Uma característica dos quitenóforos. Eles têm essas redes de cílios que ficam vibrando. E é isso que faz com que eles se locomovam.
On the list before.
Eduardo, o Renato ele é paranaense e está na FUG. E eu sou brasiliense e estou na USP.
Yeah, if you don't mind, because again, if we come up with something spectacular. D4 center?
Yeah.
All right, and that one is Mike's.
Or it goes to the left.
To the right to close it. Yeah. So if we're going for the center, ready. Yep. You have it?
Ele é sinistra, a mecânica da. Sinistra.
He's in. Yeah, close it. We got him. Good news is.
Ah, pegamos ele, conseguimos fazer a captura. Tá dentro da garrafa agora. Tá todo mundo comemorando aqui porque esse bicho realmente é muito difícil de capturar.
That was the Sidipin.
The corner's like immediately in the way. There he is.
Ah, olha lá dentro.
Nice job.
E aí dá pra ter uma ideia de tamanho também, que ele é muito pequenininho.
He's tiny.
Like 2 centimeters.
This is about a 2 centimeter size.
Ele deve ter uns 2 centímetros no máximo.
This is great. He is small, but he is mighty.
Probably over 10s.
Okay. Everybody happy with this team? We can move forward?
Yes.
Great.
Move along.
Thank you, pilots. Nice job. Perfect. Heading back down towards our 1200
E eu não sei se
meter
todo mundo
mark.
que está ao vivo aí sabe o que é um quitenóforo, mas o quitenóforo é um organismo que, apesar dessa aparência gelatinosa, né, ele está mais relacionado com os outros metazoários do que com as águas vivas. Então, durante muito tempo você achou que eles pertenciam ao mesmo grupo das águas vivas, mas na verdade eles têm 3 tecidos embrionários e a organização do neurológico deles também faz com que as hipóteses que a gente tem é que eles são mais próximos da gente do que das águas vivas. Isso é uma coisa bastante curiosa, né? É um grupo bem antigo, né, que por causa das características dele não tem registro fóssil. E a evidência que a gente tem é baseada em análise de morfologia, as mais antigas, que também é corroborada por análise molecular. Então, um dos nossos objetivos também quando a gente estuda esses bichos é entender essas relações que eles têm entre eles e entre a gente.
It's there, it's just not.
Moving on, my friends. Yep. Middle. Long. Fish-like. It's coming. It's coming. Might come back. Team. Yeah, he's right there by the sampler. Yeah, he's stuck in our wake and just kind of took a little ride behind us. We're good. There was something that looked refracting, Russ, you're right. What do you see? How do you see it? And I don't. Oh, because it's above my view.
The ocean is just beautiful.
The ocean is beautiful.
Like a blurred house.
We're not stopping for those, yeah? No? Okay. Oikopleurid house.
Stop. I'm just calling it out.
No, that's exactly what we want. Thank you. Just want to make sure we didn't need to.
To the left, mid,
Left middle?
fish.
Oh, it might have been a fish.
It's fine, I was just trying to figure out what it was.
I think we're okay. Yeah, this guy. It is a fish. Is that a
Yeah, it's fish.
Yeah, it's a fish. It's
a cyclothony?
Cyclothony, yeah.
Yeah. Do you know what species, Marcelo?
Only forget it.
So this
them, we'll see.
species of
Yeah.
fish right here is called a cyclothony.
Catch the fish and we're gonna
If
see.
we could, he probably just jetted away. Oh, but if we keep him for a second, this is the most abundant fish in our world's oceans. There's many species of cyclothony in different oceans around the world. Very common fish to find and extremely abundant in the deep water.
Traduzindo.
Anything else, Marcelo, I should share?
No, I was going to translate.
Oh, feel
Yeah,
free.
yeah.
Yes, thank you. Marcelo's going to translate for us. Thank you.
Então, esse é um ciclotone, né? Esse é o vertebrado mais abundante do planeta. É esse bicho, essa espécie, né? Na verdade, são várias espécies desse organismo. Aqui no Atlântico estão em 5 ou 6 espécies registradas. É um grupo bem difícil de identificar, mesmo com um exemplar na mão. Então, eles são muito frágeis. O crânio dele parece um papel. Então, quando eles sobem para a rede, às vezes ele vem bem destruído. Mas é muito legal ver esse organismo nadando assim. É a primeira vez que eu tô vendo, eu tô super empolgado, super emocionado. Geralmente a gente vê só em bicho já coletado. Que legal, cara. Ciclotone.
Que massa, cara.
Eles são bem abundantes, que eles se alimentam de plâncton. Eles são o equivalente ao que o crio é para os crustáceos, eles são para os peixes. Então eles são.
Marcelo, you're happy? You're good?
Yeah, I'm good.
Okay, great.
There we go.
He's like,
Oh, you want me to just stand here?
Great. Thank you guys. We can keep going down.
Sometimes helps with you to take the multiple.
That's what I did at the end. I just didn't want to come back after that.
Oh, I'm thinking about it back there.
There's a house. Wait, stop. Oh, sorry, you guys. That was right there. Team, isn't that what we just saw? Russ?
I was trying to annotate it.
Yep. We did just. Where'd it go? He was there.
Underneath.
Oh, he might have gone underneath us. My stopping skills were a little late. Okay, we can keep moving. Sorry about that, guys. I'm working on my stops. Yeah, this guy. I think it's a fish. I think we're okay. This guy up here. Yep, go for that guy. He's definitely not moving like the others. Yeah, that's what it looks like.
Now, does that annotate?
We have a crustacean. Yep.
Probably a suggested.
Suggested shrimp is what I'm guessing here.
Antennae that long?
Yeah, with those long antennae right there. If we could zoom on it just for a second, if possible.
Okay, so that takes it.
So the suggested shrimp have extremely long antenna, and he said goodbye. Thank you, guys. We can keep going. Yeah, he's like, yeah, we're good. Thank you. There you go. He came in close for us. There you go. Thanks
We
again.
have collected that shrimp already.
Yeah.
In the nets.
In the
So
nets.
we actually have a specimen that goes with it.
Great. Yeah, we're good. I think it was a house. That was a larvation house. Nope. Nope. It's fish. Fish are getting me every time. Stop that. As we were just talking about. I think you got it. Yep. Middle. It's a Medusa of some kind. It's a tiny Medusa.
How far out on?
No, got it. Yeah, I see him. If we could zoom on him by chance, that would be lovely. Or that is zoom? Oh no, take your time. Yeah, yeah. Got it. He is tiny.
It's like a small halocread.
Okay, we're saying possibly a small halocread with a question mark. Dougal Lindsay, our jellyfish man, is currently taking a well-deserved break, but we're gonna get some good video here and some good images. He's right there. We got him still. We're gonna get some good video and images, so we can he can go through and identify it at a later time. We're getting screen grabs of all of this, and we get video clips, so we will definitely be able to use this material post-cruise. He is small. He's still there. He's coming back. Eight.
Yeah.
Okay.
Dougal will probably tell me I'm wrong, but it's
Yep.
definitely halocreated.
Okay. We're gonna go with that for now. We can
Go
make
with that, it
corrections
might be
later.
Botrynema.
Okay. You could put that in there if you wanted to. Okay. Thanks, you guys. We can move on. Yep. This? Yeah, that's that 4am. We want this. We want this in the D sampler. Yep, this is that 4am they were talking about before. So it's a rhizarian. Manu was very excited about this. If this is gorgeous view, thank you. If we can get a couple of images of this with video and stills, then let's try the other decoupler for now.
Okay, I heard it.
You see it. Look, Manu.
Finally.
We're gonna try to
finally.
put him in one of the
Let's put it in
desamplers.
This
Yeah.
is the biggest one.
Yeah, he's huge.
Oh,
It's
my
great.
Can you see the
This is a single-celled organism.
This
Manu,
is
what you
a
at?
Foraminifera.
Yep, that's what we had. 4am. Gorgeous. He's huge.
Oh my goodness. What is really strange is that it's a single cell, and the cell is really all just inside. All of these are microtubule dendrites. Oh my goodness.
Yeah, we can if you want to.
Yeah.
So they
What do
are
you want?
super
DPIV
delicate
or do you want
though.
iris?
My
DPIV.
intuition
We'll do
is that
DPIV first
the
before we
laser
go
might
for
explode. We should test it.
Please.
Oh, hey, the section sampler's out. I'm zapping this out.
Oh, oh, did it? Yeah,
And a
you can
cyclosome.
tell.
Yeah, a cyclosome bonus.
So before you do the second
went off.
scan, can you just focus on it for a second? Just
Sure.
to see if the dendricles, the exopoda survived. They might have survived.
Oh, here he comes. This is good. Nice.
Nice.
All right, so can we adjust the lighting so we don't have so much ROV lighting and
Can
we
we
can get the
drop
scan?
lighting? You? Coming back in, throw bonus.
Yeah, this is the Rizaria we're looking for, yeah. I think the last cruise, we also collected some. Jericho was telling me. Silicate. That's the question. Yes, they make the silicate body, but that's all in the center. But I cannot imagine a centimeter scale object made out of microtubules. So that's the puzzle, is what is this actual dendricles made out of? And their relatives make a ton of things out of strontium, calcite, all kinds of silica.
Coming in.
So they are capable, but not for forams. And then this is how they feed.
Cool. Yeah. We do that again and even slower.
And then, huh? Yeah, there's a cell membrane across all of the dendricles.
Starvation house.
It's too long to be whole.
It's right. Yeah,
No,
it's right
it's
there.
single cell. Yeah. All forams are single cells.
Is that okay, you?
Sorry, I missed it. Didn't see it.
It was
Actually, I think it was
perfect.
outside of the frame. Was it not?
No, I think it got it.
It's
It was
got
on the
it
top.
definitely got a whole thing.
This is
Are you
beautiful.
looking? Yeah. Check it on. Did you see it on the DP?
I did not.
No, I think it was outside of my frame. Too far
Okay,
to
so
the
it
right.
has to be closer to the source?
It's closer to the laser source a little
Yeah,
bit.
okay, so this should be better.
Straight in front of the camera.
Okay, we just
Joel, you've
found,
got feet in front of you,
Yeah, these actually show up reasonably well in like the DPI kind of imagery. So
Yeah,
the
I see
only
it
way you
now
can get densities
there.
and distribution on them.
There it
Up a
is.
little bit.
I haven't seen this in UVP
Alright.
data. Have
Now
you?
push it through the
I can
sheet
show
fan.
it to you in
It was in the screen
shadow
that's
graph.
In the
I've got all
shadowgraph.
I've got all
Yes.
sorts of.
I think I looked at it 2 days ago. It
No,
was
not on
fine.
this cruise.
Animals
I've
just
got
a
other
tad
cruises
Yeah.
where I get lots of them, a whole variety
I was
of
looking
different
at the
products.
UVP data sets, and I haven't seen them mapped in the UVP data.
Most people don't pay attention to them in the UVP data. So
except for like the group.
It's a little bit
Yeah.
low.
No, this is
scripts.
the French group that
There it is.
particularly only tags
Circles.
rhizarians.
Yeah.
It must be there, but it just, yeah. Yeah, I mean, this is the
Those are also a lot of vivid.
Not if we can do another one more
shadowgraph you're
attempt.
saying
but-
is much better.
Yeah.
I'm just saying you get a lot of data.
I know
Yeah,
this is
yeah,
not
yeah.
easy, but we're trying to get one continuous pass through. If we like sort of pause halfway,
Give me
it's
the
very
plug a
difficult
disc
to
in, I'll show you
reconstruct
some of the images you
Yeah.
get.
But if it helps, you could turn on the lights for a bit and just get oriented, and then just dim them when we're close.
Out to your left, down and left. Down.
It's gone.
Very associated.
I don't have it. You guys have it? There we go. Wait.
Oh, you see it?
There, up ahead.
Where?
I don't. Yep. It's not my one. Oh, but up here.
Too many cameras.
No.
Oh, there.
Small like the Plura
Wait,
house.
this guy maybe?
No. That's it, right? No?
This is something.
I think we lost it.
I think we may have.
Well, you got some good scans of it, I think.
Sure.
Yeah, we want
If we're not,
collect it.
we don't
There's-
have like a good continuous pass at like fairly constant speed. It's extremely difficult to get any type of reconstruction that looks like a animal did.
So.
Oh, if the speed.
The speed, if there's too much acceleration, or if it's like halfway in then goes back. Later on the section sampler. We can
Hey, we can reconstruct the
web scan
suction
the section-
sampler. We could move the arm of the suction sampler in and out. You can reconstruct it.
The sampler? Yeah.
Yeah.
Yeah. In fact,
We
if
don't need
both
to,
at the
it's
same
actually-
time, we can
There's
put
probably already a CAD drawing for it.
There's a
Squid.
squid. Stop
Squid.
all. Stop everything.
Oh, we don't need to pay
Can we
attention to
need to
this.
zoom on that? Can we close down Iris by chance?
Or DKV, yes.
Oh,
Or DPIV, whatever that was. Oh.
Too many
he's
letters.
Gone.
oh. No, it's down.
It's down.
It's
Do you see it?
no. I
You went
it
below it.
went down,
Oh.
so it must be down.
There he is.
There you go.
See,
Wait,
not
there he
bad,
is.
right.
not bad.
He just inked. That's
Always.
squid ink in the middle
That's
of
an
here.
ink. That is beautiful.
So give me the squid back, my friends.
We scared
I'll buy you
the
dinner
heck
upstairs.
That's a Keatingnath.
That's a Keatingnath.
Heterochronia.
There was a squid, but that is a Keatingnath. I don't.
She thought of
Oh,
it.
See in front of DPIV?
oh, we got the forearm back.
Where?
Hold, stand by.
Right
I
in
don't
the
see
center.
him.
Right
but
in the center.
oh, he's by the squid actually.
Hold
Right
on.
in front of
There
DPIV,
was a
there's
squid. There is an ink, but
Yeah,
that
go
was
for
not
the
squid when we
forearm.
saw
Let's
it.
go for the forearm.
You sure it
Okay,
wasn't
let's
from the
sample it.
Keatingnath?
Yeah. What was
Oh.
that, Russ?
Never mind.
What was that?
This
then?
is
That's
it,
the
right?
forearm.
It's another
But
one.
how come? No, that's another one because there was another
think
one
this
in the
is the
center.
forearm.
Isn't that?
So let's get him in a D sampler,
No,
please, if
far
possible.
in the sky.
Yeah, that's the forearm.
Okay.
No, let's just go do it. Let's sample him while he's here because there's a
Yeah,
squid in
let's
the area.
sample it. Oh.
All you had to do was find it.
Oh.
We're still gonna go in the. We need the second one. We need D2.
Sampling is even harder than the scanning.
It is
You see
perhaps
him?
more nerve-wracking.
You got him up there.
Just
I don't know which.
number of
Okay, I'm
shots
gonna get out of
they
your way.
get. Like you bring so close, sampler should be much farther away.
I can push it out.
That's
Oh, even the
That's
big
so
samplers? That is big.
Yeah,
It's
he
even
is
bigger.
a big.
pretty close.
Oh, come on. We can
Not
do
that
this.
one, my friend.
Oh my
The other one.
God. So what's super cool is you can
Ooh,
see
ooh.
the ectomembrane. That's
Yeah,
the one outside. Oh, we almost have it.
Yeah, we do.
We
Close
do.
it,
Close
the
it.
door! Close the door!
Yay!
Here we are.
Nice job. It
Oh
looked like
my
it looked like it went in.
Closing.
Let's just confirm.
Yes,
first.
I like
Yep,
the
it's in.
confirmation.
There, it's there, it's there, it's there.
All right,
Two for
now
two.
celebrate.
The science team, the pilots, everyone rejoices.
Awesome job, pilots.
Nice job, team. All right, 4 AM collected, brief squid sighting. I have a feeling we will not find this squid again, so we can move on.
It's like really small.
I can tell you it was an
The rest
ogopsid.
of the skin work, I thought it would be too much weird scatter through this long glass-like.
All right, so that was in D2.
Call the bar to see if it's there. Call the bar.
Try again on
Yeah. The bar. Yeah.
I don't think
It's
that's
confirmed. Yeah. Yeah, I did. We did.
Okay. Nice job, team.
Yes, these.
Okay. Not a lot of duplicates.
I mean, there are.
What is the what did the inference pick up on? Did it pick up on the right things?
I mean, shouldn't be picking up on the same as much. So let me put your first drop and everything else and pull up the real bunch, but like, I don't remember us losing it.
All right, for folks watching along at home, dinner has just started on the ship, so you'll probably hear more commotion in mission control as folks come in and out. But we are still on our way down. Haven't met our target depth yet for this dive. I think we're looking at 1,200 meters, and then we'll stair-step our way back up. And currently we're planning to be back on deck at 2 in the morning.
Opa, estamos vendo uma pequena coisa ali.
That was a Medusa, sorry.
These aren't going to be ROS yet.
Right, so
Medusa.
But at 4K, they might not be that tiny. It does overall look a little dark.
Can't decide if that's
That's
fish
what
or
I'm thinking
keeping
too. If we want to start normalizing image brightness a little bit.
Shrimp?
Keating now.
No Keating now.
We're okay. We can keep moving. Yes, if there's any opportunistic suctions, that's fine.
But the images aren't that dense, so we shouldn't be.
We
too
should be
bad
able to catch those in the nets as well.
It's nice to not horribly nice.
Have we caught anything of note in the nets that you can talk about?
So far in the nets, we're using a Tucker trawl system, and we are using net sampling to partner with our ROV and genetics operations, as well as our imaging operations. In the net so far, we've had a good stop. Sorry, everyone out there.
Professora Heather Judkins está explicando
That's what
que,
we're trying to come on.
além das coletas
I
com
don't see
ROV,
anybody in it. Do you, Russ?
estão sendo realizadas coletas com rede também.
A little bit, it might pull in on something. There's.
Yep. Stand by.
Looks like there's a core.
It looks like something's in the middle.
It's just an old house or
I
an
think
old feeding structure.
Isso é uma casa de um animal chamado larváceo, que produz essa casa de muco.
Could be like a copepod in the house or something like
E essa casa de muco, ela é abandonada. Ela é abandonada. E o animal produz uma outra casa de muco com a qual ele filtra partículas que ele usa para se alimentar, né?
No, there's a tail moving.
Really?
Looks like there's something.
Vários animais marinhos produzem muco. E esse muco agrega partículas. Todas essas partículas que vocês estão vendo aí na tela são partículas que podem ser capturadas, né? Algumas delas
Yeah. Okay. I think
são partículas
it might
orgânicas.
be a discarded house. It is lost.
E é uma estratégia
Okay,
de
yeah, too far away.
alimentação, né? Desse material que está em suspensão na coluna de água.
So with the net samples, we are looking at biodiversity. We are looking for new species. We have some really cool techniques in the lab that we can use with collected animals as well. There's a lot of behavior experiments going on as well. This net is designed, this Tucker trawl we're using, it's designed to bring up small animals in really good condition. So there's not a ton of water running through the cod end, which is the collection cup at the end. So animals come up in pretty good condition, and that way we can work through our different experiments and processes that we do. Do you want me to stop you guys? Stop?
Zoom in.
Yeah, zoom in on this guy. It's a lobe 18-4. No? See, my guesses on gelatinous are never good.
Larvation House. Uma outra casa de larváceo secretado. Essa está vazia também. O animal já abandonou essa casa, uma estrutura de
You think it was a larvation house as well?
Professora Heather Judkins está explicando que as coletas com redes são direcionadas à captura de animais pequenos, uma rede que coleta esses animais de uma forma mais cuidadosa.
Right.
Just trying to decide if the animal is in there. I think
Right.
it is. Right.
Nice job on the zoom, you guys.
That's actually funny because it's almost angular on the ends, as opposed to blunt, clubby. But I still can't see the animal.
Yeah, the angle that it's at, right?
Provavelmente essa casa já está vazia, né? Então é um animal que produz essas casas. Quando essas casas ficam repletas de partículas e entupidas, ele abandona a casa e produz uma outra casa de muco. E isso colabora muito para a produção da neve marinha, né? A famosa neve marinha que afunda e transporta matéria orgânica produzida na superfície dos oceanos para o fundo dos oceanos.
Pretty sure she knows. Again, getting these images and focusing on moving animals with moving ROVs and currents and zooming is a technique that takes a long time, and it is still difficult to do. I think it's refracting. It's reflecting light or something. I don't see an animal. Do you see an animal?
I'm not seeing the animal. No, at this point, we should see the tail.
Right, I should see somebody moving. It's zoomed in enough. There's a Keatingnath over to the left. Yeah, I don't see anybody in there.
Então,
Yeah, it's refracting light.
Existem várias câmeras posicionadas em
Great
várias
zoom, you guys. Thank
direções.
you. Are we good?
E vários
Russ,
pesquisadores
you good?
estão procurando por animais interessantes que possam ser filmados ou até coletados.
I don't think the animal's present. No. But it's been abandoned recently because it's still pretty inflated.
Estamos tentando observar em várias posições a casa para ver se ela está vazia, mas aparentemente a casa já está vazia. Não há nenhum animal ali dentro.
Good?
Yep.
All right, we are a good team.
Thank you.
Joel's like,
That was excellent.
that was fabulous, keeping it on that.
Yeah, good.
Nice job.
Good job.
Continuamos então a descida, direção a 1.200 metros de profundidade.
Nicely done.
The Oikopleura always have this impression of kind of like angel wings, nice and smooth and symmetrical.
All right.
And these other ones, and again,
See if we
the problem is
can-
I don't have a lot of
We're at
specimens.
855 meters, still headed towards 1200.
Not Oikopleura.
A professora Heather estava explicando que as coletas com rede têm o objetivo de coletar animais vivos, né? Uma rede que coleta os animais de uma forma cuidadosa. Os animais vêm, alguns não conseguem chegar vivos, mas muitos vêm vivos e nós estamos realizando vários experimentos de comportamento animal, de alimentação, biomecânica, uma caracterização morfológica deles dentro do laboratório para a descrição da biodiversidade dos oceanos profundos, né? Então aqui é uma região muito pouco estudada, muito pouco conhecida, que está sendo agora explorada cientificamente.
You dig now?
Send a reason where the iris lights are.
Is that okay? Is that what you're saying?
I don't know. They're not.
Are you guys okay with where the iris camera is set right now?
Okay, so dinner is 5:30, 6:30, right? So dinner's on.
Yes, you're okay with where it's positioned, the iris camera? They just asked. I just want to make sure you're good. It's just sitting. It
Os
looks fine to me,
larváceos
but I
vivem
don't know.
poucos dias, eles secretam várias casas dessas ao longo de algumas horas, né? E com isso essa produção dessas casas colabora para a formação da neve marinha, a agregação de partículas e o afundamento dessas partículas.
Right. Closed.
Yeah, what we experimented earlier, I think it was maybe before you guys' shift, was actually bringing it up and over above the, if you're out looking outside,
Yeah.
above the DPOV, so you can have it like very central to the view. From here, it was
Tem experimentos para se conhecer como é a visão desses animais que vivem em regiões profundas, né, onde há pouca penetração de luz, experimentos sobre a alimentação desses animais, vários equipamentos óticos de fotografia de caracterização desses animais estão sendo testados também.
But yes, someone should come and switch you out, yeah.
Google will someday go through this and rewatch it all.
Stop. That guy?
top center.
Yep. Top center. Right there. I don't know what it is. I just saw that it was different.
Another mucus
Is it?
field around it, I think.
There's a lot of mucus down here. A lot of mucus. It's a...
It's a something.
It's a snowflake in the middle of the ocean.
Alguma coisa é.
One of the little songbirds or is it
Muito pequeno.
I don't... We're gonna get a little zoom on this in a second. It is not a Tomopterus.
Olha, olha, que lindo.
Nice.
Lindo, lindo. É um sifonóforo,
Yeah, this is beautiful.
é um animal colonial,
Do you guys want to do anything with
quinidário.
this, Connie? We don't have a D-sampler available. Nope.
We only have two.
We only have two.
Oh, there we go. Now we can see the nectosome.
Yeah.
Yeah.
Do we know
What are my options?
Space zoom.
Fisonet. First one. Yep.
Um sifonóforo de Fisonect, um animal que tem nectóforos
Yeah,
que
of
vocês
course.
podem ver
Yep, if
pulsando.
we can get some good zooms while he's opened up like this in a good angle. Uh-huh.
Como se fossem várias medusas pulsando, um animal colonial que vive, né, com vários indivíduos unidos na colônia.
Oh, which one?
Come close to
Yep, yep. We're working on a zoom, team. We're working on a zoom.
Aquele ponto mais acima da colônia é o flutuador da colônia,
This
chamado
looks
de
great.
pneumatóforo.
Any chance for more zoom or no?
Tem gases ali dentro.
I know, I was gonna say, I don't want to push it.
E o pneumatóforo é responsável por manter a
Oh, we're getting
colônia
there.
de pé.
Has it got the one tentacle there on it?
I see from the tail end.
No, this is the apolemia that we had the other day, but the tentacles is not as distinct.
There was
It's
one
a
out
smaller-
early.
It's a smaller animal with fewer
Yes.
nectosomes or nectophores. Nectosomes
Right.
region.
A parte mais esbranquiçada da colônia é a região que realiza captura de alimento. Então há tentáculos com
The
a
general
capacidade de
consensus from
aderir
science that it could be an
outras partículas,
apolemia.
né?
just when,
You know, I've only seen them, like, big ones. I've never seen.
Yeah, juveniles or
Yeah.
developing, right?
That's
Anything else we
cool.
need from this? We good?
Or it could be a new species.
it could.
Well, then we're gonna have to.
And
the
we
option
could
is the
suck it
suction
up.
We can suction sample if you want to suction.
Suck it up. It's a big enough target. They would probably, Dougal wants multiple pieces.
If we can suction sample this baby, that would be great.
Vamos tentar fazer a
It's
coleta
a gentle, right? A gentle suction.
de uma maneira cuidadosa. Esses animais são muito frágeis. Geralmente eles se fragmentam quando a bomba que puxa o animal, quando ele é colocado dentro do ROV, ele se fragmenta. Mas mesmo assim é possível fazer uma análise do material genético desse organismo e assim tentar descobrir se trata-se de uma espécie ainda não conhecida pela ciência. Aproximando-se, a navegação é bastante sensível, bastante delicada, né? Tem correntes, tem a própria turbulência do ROV que atrapalham. Mas a equipe aqui da pilotagem, muito experiente. Vamos
This is ver. what we do. We collect a whole bunch of things that Dougal has to work on, so he won't get any sneaking peek tomorrow.
Very small.
Well, we definitely hit him on the lip.
Quase.
He's close.
Quase, quase.
Yep. Nope. Behind.
Ainda tentando manobrar o ROV para tentar coletar esse sifonóforo.
Oh. Oh, that's it.
Oh,
We got
yeah.
him in. Look for canister.
Conseguimos, vamos olhar na garrafa se ele vai chegar.
Canister number 7.
Na garrafa número 7.
Yes. Are we sure it's canister 7?
Yeah.
Okay.
It should be big enough we can see it
I
when it
don't know.
does hit the
It says
canister.
7 first. Did it not go in?
Yeah,
So
we
it
just
looks like
gotta
it was an attempt.
scoot forward basically and just
Yeah,
let
neither
it
do
get
I.
pushed through.
Neither
It took a
raise that.
while for the last one.
Well, I'm going forward, let's see.
Yeah.
Should I increase the suction?
No.
No.
Oh,
There, nice.
Centro da garrafa e esse não se fragmentou.
Make sure it settles for a second before
Yeah.
you close it.
Esse
Don't do
está
anything
inteiro. Oh,
Should I stop the
I
suction?
think you turn suction
Yeah,
off
turn it
now,
off.
um animal de poucos centímetros.
And then move forward a little.
Yep. And move
Okay,
forward
now
a
we're
bit.
Okay,
And then
It's down.
rotate it.
Alright, now we can
it's done.
Perfecto.
Good.
Nice job, team.
Other way.
Is that the
Nice
right direction?
Good job.
One. There we go.
Beautiful.
Nice flying, Joel.
Fabulous job, team.
Mais um animal que vai ser estudado aqui por nós quando o ROV retornar.
We're gonna get the winning prize for the smallest things collected in the middle of the ocean. Nicely done.
I've been getting lots of
You guys are switching. All right. Pilots are gonna switch out, and then we will continue our dive down to 1,200 meters. We're at 878 meters currently.
Estamos agora 800 metros de profundidade, continuando a descida até 1.200 metros de profundidade.
Thanks, Joel. We're going down. We're slowly going down to 1,200 is our target. There's a jelly right in front of us. Stand by.
All sorts of things.
Stand by. We need to zoom in on this beautiful Medusa coming right at us.
You should come up with a design and to get handed to the pilots and ask them what
Yeah,
they think.
he is very small.
Is it a little clothing or something?
The tricky part is like when you come up, what happens then, right? So like if you're
All right, he is going on.
Yeah.
We're continuing down to 1,200 meters.
We're giving up on that?
Yep.
You just
But you could
want
even
more
do things
squid.
that need to be picked up by the manipulator,
I just think
right? We have
by
a little bit of
halfway
range
through
of
the
motion right
dive,
now.
we should get to
We were
1,200
joking about a
meters.
butterfly net, like yesterday
Just
when we
kind of
were
working our way towards it.
Oh,
Slowly, of course.
Oh.
Was it yesterday supposed to be the leisurely stroll?
It was, because that was a shallower depth.
Just open. Or what's the cup? What's cut? Turn it, the top part.
Agora são quase 6 horas, horário do Brasil, um momento que já na superfície dos oceanos começa a ficar escuro e é um momento em que animais das profundezas começam a subir para a superfície, em direção à superfície. Lembrando, né, que nas profundezas esses animais ficam camuflados pela escuridão. E é nesse momento que acontece um movimento de migração vertical diária, né? Todos os dias, assim que começa a escurecer, os animais ascendem à superfície e aí se alimentam, né? Onde há mais oferta de alimento, né? Lembrando que nos oceanos o alimento é produzido na superfície dos oceanos em função da atividade fotossintética do fitoplâncton, né? Então grande parte do alimento disponível está na superfície.
What's that?
Opa.
Over here to the left. This little guy. I don't know what it is. Just saw that it was different. Coming in the middle. If we could zoom on it, that would be great. You get a handle on it. It's going down.
O que é?
Armadilha pra camarão, vou fazer uma amarrar no rola.
It is off
Pra
my
ver se
screen.
Joga os biscoitinhos lá dentro.
May have lost it? Maybe not? No. Okay, they can keep moving. Yeah, we're good. All good. That guy might be it. Middle.
Aqui nós temos uma equipe de especialistas que está anotando todos os animais que podem ser reconhecidos, né? Então esses animais acabam sendo registrados, né, nessas localidades
I think so. So I'm
e
trying to
através de
look
várias
at it.
câmeras posicionadas em várias direções, esses animais são avistados e
Yeah.
é feita uma marcação
Just the
da posição
shading.
deles.
You see the nectaphores?
Trying to get a little bit better view of it, but
Right. It's small.
Quer ir jantar? Logo? Quer jantar logo? Eu fico aqui ou você
looks like the last one, but
Horas, já é 6 horas.
But without the.
it may just be
Que horas
pointing away from
que
us.
é?
Right.
Acaba que horas? Não acabou já? 6h30?
Yeah, I think they're there. They're in the back.
6h30.
Vai lá rapidinho então. Ou vamos... Oi?
Another cyphozoan. Doesn't look right. It's definitely curled up. See what I'm saying, Russ?
Nice shots.
Yeah, great,
Whoa.
great zoom on this, you guys.
É um momento em que o sol se põe, né? E aí a escuridão atrai os organismos para a superfície.
Agreed. I just don't see it moving.
Aqui na latitude do nordeste do Brasil, mais ou menos 5h30 da tarde, já está bem escuro. E é o momento em que vários desses organismos do fundo do oceano estão em direção à superfície, né?
I would
Okay,
love
that's
to.
probably.
I think we're
probably
good.
good enough.
Yeah, we're good. All right, I'm stepping away for a little dinner. Karen Osborne, extraordinaire, taking my spot. I'm back in it. Anybody else need anything? Russ, do you want me to find somebody to replace you?
Yeah. you
Okay.
find somebody to
I need a
replace me.
squiddle person. Everybody else good?
Yep, I've got my replacement in a few minutes.
Okay.
Fizemos uma troca agora. São descidas de 12 horas, então há um revezamento, tanto da tripulação que faz a pilotagem do ROV, quanto dos pesquisadores que estão avistando os animais e guiando, né, as amostragens e o registro deles.
Stopping and everything. I'll do the same.
Kidding now?
Ou Ketognato.
Something else, we're seeing it from the side camera, if you swing right, see it in the, see it level with just
Verme marinho, um predador, do zooplâncton. Parece que encontramos uma água-viva em outra câmera aqui.
Just bringing it over. Here it comes.
Uma medusa bem bonita.
Does look like Halotrephes.
target.
It's a
Especially
nice
if it's
one.
still hanging out in this shape. Those are the words you've been waiting for, right?
Yeah.
Pouco ainda fora de foco.
So after
Does
we get a
indeed
nice
look
little
like
view on
Halotrephes. Can we zoom when you feel like it?
Animal que parece ser do grupo de traquimedusa, é um animal que não tem a fase de pólipo. Medusa holoplanctônica, né, que nós chamamos esses animais que vivem o tempo inteiro no plâncton, no ambiente planctônico. Um animal grande. Opa, que bonito animal.
Halotrephes, that's for sure. Oh no, maybe it is. It's got a lot of.
I'm gonna give it a shot in a few seconds here.
Oh, that's nice.
Yeah, I think it's.
With
the-
that
the
many
radials are
canals, it
thicker,
looks like
So
trapeze.
it's a halotriatic thing.
Muitos canais, né? Tem vários tentáculos ali estendidos. Um animal que parece até fogos de artifício, né?
Okay to turn on laser? Yeah, go ahead.
E vamos fazer o escaneamento agora através desse equipamento que é o IRIS. Esse equipamento tem uma câmera com várias lentes, é um tipo de fotografia plenotípica que fotografa em várias profundidades de campo, né, e faz uma montagem tridimensional do corpo desse organismo.
Even more ghostly like that.
equipamento desenvolvido pelo Monterey Bay Aquarium Research Institute, na Califórnia. O pessoal está discutindo se é uma alicreia, se é uma crossota, é um animal do grupo Traquimedusae. Mas é complicado essa identificação. Já estou vendo que tem gente aí no chat dando sugestões de identificação, legal.
Question in the chat about how we know it's going dark. The temperature does not change necessarily the weather, but the cameras on the outside show us that it's dark out there. On the outside of the ship. And the time. And at some point we will see everything swimming upward. Not everything. Many things swimming
Wow.
upwards to
Que
go
bonito.
feed in the rich surface waters. Under cover of darkness where they're less likely to get preyed on in the dark, and if they were up there during the day, and they'd be easy
Oh,
to pick up.
another jellyfish.
You don't need it. Yeah. Oh, because the professor pointed out like that.
What do you estimate the sizes of?
It's pretty huge.
About this, because
I could
the
put the
section
lasers
where
on
we're
if
looking
you
at
want,
the camera is
or
on
leads
this
on.
side.
How big do you think it is? 2 inches? 3? No? Below?
Um animal que deve ter em torno de 10 a 15 centímetros, né? Pela referência aqui dos equipamentos do ROV. Que para os padrões dos organismos que nós estamos observando, né? 10 a 15 centímetros é um tamanho grande.
Whales don't look quite as numerous and a bit too broad from the Falkor I'm used to, but I'm also not used to looking in waters off of Brazil.
Yeah.
It's a little low for DPOV.
Agora sim.
It's below and coming in.
Oh. Pessoal da pilotagem do ROV bastante concentrado para tentar enquadrar dentro do feixe do laser. Para tentar enquadrar o corpo inteiro do animal dentro desse feixe aí. Vamos ver se agora vai. Quase, quase. Opa. Opa. É um animal bem grande, né? Como vocês podem ver. Difícil enquadrar todo o corpo dele dentro desse feixe. Tá bem ajustado, tem que estar bem ajustado.
so I can just talk to you. I can send a message and see if somebody can come.
The screen is extra dark compared to mine, 'cause I can see it on there.
Show me how to use this.
Elio, do you want to do this? Katie can eat, or is somebody else coming for that?
I mean, I can help, but I don't know how to use Quill.
Oh, okay.
I mean, if you can show me, there it comes.
Opa,
That's pretty good. Yeah.
parece que agora sim. O pessoal está fazendo revezamento aqui, né? Tem gente que está fazendo a marcação das espécies que são avistadas e agora é a hora da janta.
Thank you, guys.
Parece ser alitrefes, sim, mas é difícil.
Hi there. Yeah, no problem. The important thing is, yeah, yep, if we image or collect something, the time code goes in the column for what you did to it, DPIV iris section. This one's easier. Yeah, for sure. Was that during dinner?
Agora chegou a minha vez, chegou o professor Marcelo Melo da USP, eu, Renato Nagata, tô deixando vocês aqui, um abraço a todos. Daqui a pouco, vou jantar, daqui a pouco nós estamos de volta aqui. Deixar o professor Marcelo com vocês. Em português e mais nossos colegas também vão fazer uma parte em espanhol também.
No. Oh, yeah, no, she should have because it was DPIV.
Do you write it like that or do you write
H-A-L-I-P.
Oi pessoal, depois desse show dessa água-viva, vamos ver se aparece uma coisa mais legal no meu turno, né?
Slightly oversized.
Yep.
Mark, are you able to see where it's in the DPIV?
2114, yeah, it's fine. 2114. I'm looking at the bottom of the screen. Probably. We have plenty of samplers left. It's probably too big, actually. How big did you think this was, Les? No, he's gone. How big do you guys think this is? 20 by 30 centimeters. It's huge. It's way too big to be suction.
Yeah, it wouldn't fit in that box.
Yeah, it would have to be in a D, and unless it's a new species or something, we probably
That I
don't.
cannot have.
What? Seriously?
Once you have the sequence.
Yeah,
It's
then you know.
everything.
So you can help.
Yeah.
Okay, so then over here, you can do as many biological observations as you want. Ultimately, if we were to sample it, it would be a biology sample, so there's blue buttons at the top. Yeah, so if you click on that, it will take frame grab, and then you pick one of those, which group it's in, and you can add more specific information. Since you picked a sample, then you have to put a sample ID and what sample you put it in, and that's it. So, sample ID is what you get off of here. So this number has a number. Good to go. And when you start typing that, luckily it pops in the whole thing, and then you just hit backspace on the mouse button. Put the right number in, so you can just cancel that.
What about
DPIB, do not like that. So research stream. Well, everybody's using Heather's login and everything. But it's not, well, it's kind no, it's annoying to switch it every time. Heather, looking good today. So you could DPIB, put a DPIB thing. Put the button at the top, DPIB. There, deep. Yeah, look, it was like we didn't actually enter it, did we? Oh, you did, but you didn't put anything in about it. Okay. So just it takes 2 frame grabs, one of the sit cam and one of the other camera. So if you do deep PID, I think we can put the information in there about it. Oh no, you can't.
Did you really?
Okay, that's it. Yep.
So only the tail end of the
Submit.
center.
Yep. Submit, and then we'll go back. Maybe can you edit? I think probably not edit. I would do the biology observation. And type in Nigeria. It's okay. And then type in Nigeria. Medusa. It's a fake Medusa. And then additional text you could put in Howard Duffy's. Yeah. And then that's it. Yeah, I see in here when I came in here a few minutes ago. I don't know. Yeah, I don't know. It looks a little different than the halotrephes we get. The canals don't split the way ours do.
Could we, just to.
So it's some kind of halotrephe. What it is. Do you think they'll come off if you suck them?
Probably.
Because it's way too big to go in the suction tube. But we can see if we can get a tentacle or two to go through. Too bad we don't have the biopsy tool. I know. This is the perfect target.
I think that big is
The big tube is like, yeah, big couple of
wasn't as big as this.
Well, they're, skinny little, tiny little bit. So we want to see if we can suck off a tentacle or two with this side. It will plug up the nozzle mostly. Well, it'll clog up this shadow cam. We really don't want it to go in the suction.
Okay.
But we would.
You have a helicopter.
Right.
It's called halotrephes. H-A-L. Okay.
They're abandoned. I think you can have. Can I have actions?
Do you want one?
Yeah. They're better with a layer of just so they're actually, but there's no effect.
Yeah, people want to talk to the data for us.
Which one? Just go with the green.
It's gonna be.
So how deep I think I have a hard time.
Or go like just on the side and not super high suction. And just, I think if you don't line it up exactly, I'm not sure that it will drop tentacles.
Or if you just click how and try to click it. So we just click this, the higher group. There you go.
I could put it in a D, but we're likely to trade it out for something else later.
So there's a question. Yeah, you got both these are full.
Yeah, both these.
Oh, they're full. What did they, what went in the second one?
Capture.
What went in the second D?
Did I give you the sheet?
I think it's such two sizes.
Oh, yeah.
Yeah.
Oh, the two sizing lasers and the one
Pointed
that's
straight.
the one
D1,
that's
D2. We have
Tina 4.
It just has.
Oh, no.
You think that's what that is?
I think
Tina4?
that's, I think that's
Tina
what
4,
it's meant
yeah.
to be. And a 4M. Yep.
Oh, really?
What kind of Tina 4? Foraminifera.
Oh. The revised area, I see.
Oh, yeah.
Oh.
No, it's definitely a 4M.
Yeah. That's fantastic.
Dino4.
Was
Tina 4. I think that's what
I
it's
think you're
supposed
right.
to
I think it's Tina 4.
be. Must have been a pretty special Tina 4.
But they're good for genomes because they have small genomes.
Yeah, okay. That's very sizist of you. But yes,
We get
good to have realistic targets.
bigger
things you
genome
can do.
sampler, bigger genome sequencers on
Very
board.
hard
That
to
makes
do.
me-
Oh, he's sticky. And he's not dropping
No.
tentacles. Mm. Oh, he's very I don't think it's suction that's holding him on now. I think he's stuck. He thinks he has hit the mother load. Whoop. Whoop. No, that's not good. Can we reverse?
That's reverse. So it's too big.
It's way too big. Okay.
Oh, yeah, tentacle.
Might just have to.
Might scrape him off the end of the funnel. Can you push him out? Does it have reverse? No
No, we
reverse?
don't.
No
Don't have
reverse
reverse.
section. Oh,
See if I can
that's a problem.
Add more.
all the way there, son.
Not gonna make it around the corners though.
More suction. You need to reduce more suction. Try to see.
Yeah.
Well, if you can't go out, then yeah, just suck as hard as you can to get him. Gonna be a stingy trip through that little maze on the way
Just
We'll see.
in.
fly ahead.
Coming. There you go. Okay, great. Oh, he actually doesn't look bad.
Yep.
It's organized.
That is a pretty little Keating now.
So this is
Completely
going.
red.
Yeah, we've seen a few of those today.
Where are we putting this?
Yeah.
That is in.
That is in number 8, I think.
H A E T O G.
At least we can see in the shadow cam to see if he makes it through. Oh, we need the lights back there though. Hand you over to Heather.
Yeah.
Yeah.
It is a midwater expedition. We are heading down to 1,200 meters, and then we will work our way back
We're
up.
working on it.
I just want to say
What you got?
where our sample is actually good.
All right. Thanks. Thanks for covering.
Is it,
All right, we are good to go down. Continue forward. Forward and down. Perfect.
Another fine mess we've gotten ourselves into. Did we get it unclogged?
What's the largest?
Did we get it unclogged?
We did. It made it.
At least in the tube. Did it make it into the bucket all the way?
It did. It did make it.
Did it make it all the way to the canister?
It did. It's in the
Oh,
canister for
good. I
sure.
was afraid it was gonna get jammed again once it got
Nope.
bigger and smaller going through Doodle's.
No, look at
That was a shrimp.
I've never seen one that big.
I don't think it
about biopsies.
Yeah. And you're going down?
But it's, well, yeah. I'm
That's
not
what we
sure
need to
which
be.
one it is.
When you are ready. Does the camera go up or out more? Feels like it's low. That's it. Thanks, you guys. Another Keatingnath. We're passing by. We're heading on down.
Heather, can you give me a couple of tips? Like, when
Yeah.
do I do something here?
So anytime we
with
say,
that
like I just said,
Keatingnath, could I have
Keatingnath,
done
yep,
Keatingnath?
you should go biology
Okay.
observation.
Okay. And when I click that, it'll just
Click it in. It'll give it a frame grab, and it
immediately
will.
as soon as I click the button.
And then you click through it. If we can get more.
And these things are like.
You only have to worry about
them.
that one. If we do a biology sample,
we just did
don't worry
one,
about
right?
those. Don't worry about anything else.
Definitely enough.
You did this one.
Yes, right.
I don't think I.
Did anything there? Then I would.
It's too late.
It's never too late. Put in the notes. Just put. You could put here. You could get out of that altogether and just go to a new event and just say sampler number 8. This right here, this big line. You can type in anything and it shows up down here. So just type this animal, the number. That way it's noted in here.
With like a commas. and
Yep,
stuff?
that's fine. Yeah,
Okay.
it's like a free text box. Yep, and then container 8. Exactly. All right, Heather Judkins is back. Karen was awesome and relieved me for dinner. Team has been relieving as we needed to. Dinner is 5:30 to 6:30 on the ship, and it's a very important meal for all of us. Local time. Always delicious. Larvation house. There you go.
Oh, the
See, yep.
observation.
When, yep. Wait.
What?
You gotta hit submit, or that won't go in. Yep. Click it. Yep.
Where'd
Now
it
you
go?
can go. Go biology observation. Yep. You're good.
It's not there anymore. Okay.
It will be. It should be.
With larvation, but okay.
Yep. Larvation house.
Yeah.
And then hit taxa. I think there's even a house. I think there's a house category.
Oops. Okay. Larvation. Other house. Oh, there is.
Yep.
Okay. I'm
Sweet.
just gonna submit that.
Yeah. Perfect.
But I don't think I got the frame grab.
You don't have to. It does it. Every 10 seconds there's a frame grab, and anytime you enter
So
something,
it'll
there's a
know
frame
that
grab.
like
It
somewhere near
matches.
here there's a larvation
Exactly.
house.
Yep.
Okay.
You should. I think.
It's hot in this room.
Yeah. It's warm in here. All these monitors, computer things.
I want to play Star Wars music. See if I can look up some. He's in data logging. John Burns is in data logging. One of the two hot seats there. Elio Chalita. Is that right?
Coming by about 3 o'clock.
Okay. Elio
Yep.
Chalita's in the
Medusa.
squiggle.
Yeah, sometime.
Closing in on Medusa in the distance. When we can get Zoom when you guys are comfortable, that'd be great.
So cute.
Is it another
Hi,
house?
Karen.
If we could Zoom,
That's a different Karen.
or not yet. No, you're good.
Come on.
Come on.
Thanks for sticking with it. Any IDs from the crowd?
Nope.
I think it's the same thing as what we just collected.
Medusa.
Hello, trapeze. It looks.
It does
very
look similar.
different,
You think?
huh? I don't know.
We're gonna stick with Medusa.
Yeah, it was a little bit more. Yeah.
That's a Trekkie Medusa
This is
for sure.
great
though.
video. Do we know what type? Do you think Trachea Medusa? Or do not we want to go that
Go
far?
either. I know either.
Well, I think it's I think it's a track.
Okay.
So,
Can we get can I do a scan of this?
Sure.
By eye?
This is big.
Do you want iris or DPIV?
DPIV, Marco, if you're in a good spot.
Yeah, we can.
DPIV it would be great.
I'm recording and the laser power is on.
Yep, we see it.
okay, since we're doing this, it gets in the sheet.
Yep. Correct. And you don't have to do anything up here.
I got that.
Yeah. There you go. Nicely done.
Learning.
Nicely done.
Oh, this is so pretty.
Agreed.
It's exciting.
It is excitement.
I want to shout, but I don't know if I should.
Going off screen. Yep. There he is on yours. There he is on mine. He's like, I'm out of here. Yeah, he's swimming around.
Yeah.
Okay. And then nice and slow on the way back. Oh, just went a little low. Almost, it was just out of frame on hers.
So fun.
Karen, too fast? I don't know if we can do anything about that. That's the Medusa.
Yeah, I know it.
Okay, just making sure.
Unfortunately, they don't swimming too, so this is.
Yeah.
No,
There.
because the time it takes us to scan.
Right.
It might not be.
Unfortunately.
Yeah, he's on our side now. Yeah, he's gorgeous. Come on, baby. Maybe? Oh. He's dodging it. He's dodging it. Oh, there was a 4am that just went by.
Para também ser escaneada. Esse escaneamento possibilita que a gente tenha uma reconstrução 3D do exemplar antes dele ser
I don't
coletado.
think we went much past that. Oh,
E
yeah,
assim a
we
gente
did.
consegue estudar a morfologia dele e eventualmente até descrever a espécie como uma espécie nova. Então, com esse escaneamento e com o material genético que é coletado do indivíduo, a gente consegue fazer, se for uma espécie nova, dá para fazer a descrição da espécie, sem perder nenhum detalhe.
Thank you, John.
A misteriosa Abissal falou que foi visitar o navio Falkor no último domingo. Foi aberta a visitação lá no porto
So you're
de
good? Okay.
Salvador.
Connie says
E-
we are good.
E aí bastante gente foi visitar o navio. O pessoal de Salvador, alguns alunos de lá foram visitar o navio.
Don't we all? Perfect. Then we can scooch back to heading down. We're only at 860, so I'd like to get down, but we don't need to speed, but it needs to be really cool to slow down. Is that okay? Because again, we're coming all the way back up as well. Russ, you're okay with that?
Yeah. Well, not really, but
That was a
I'll
key
live
nap.
with it.
I don't mean zoom down. I mean stop if we want. Yeah, this is good. Even if we can break 900, that would be fine.
O tema do Blade Runner.
Que loucura. Marca o lado.
Acho que eu te marquei você lá.
Yeah, we're good.
É o misterioso Abissal. Tudo bom? Eu até imagino quem você seja.
Might be a siphonophore. We can zoom on it, that'd be great. See if we want to stay.
Yeah, whenever you're ready there, son.
Yep.
Nice.
Yeah, he's positioned himself quite nicely.
Oh, what type of siphonophore?
Ask Connie again. She just said it. Yeah, Fisonect.
It's a, yes, siphonophore.
If we can zoom just one more
It's a
time.
Fisonect siphonophore. It's that part of
O Renato está aqui do lado, ele acabou de voltar. E ele quer falar um pouco sobre o sifonóforo que acabou de passar por aqui. Está agora na tela de novo.
There you go.
Boa noite, pessoal. Agora estamos tentando, aqui é Renato Nagata, da Universidade Federal do Rio Grande, FURG. Agora estamos tentando se aproximar daquele animal ali, o sifonóforo Fisonect. A gente consegue ver ali um ponto bem branco ali no topo da colônia, que é o flutuador da colônia, é um pneumatóforo. E vários indivíduos, né, porque é um animal colonial, vários indivíduos que são medusoides, né? Pulsam de uma forma coordenada.
Feeding tentacles is the tail section in the back.
These are stomachs and gonads and armor. On the back half. Should I take another picture?
Oh,
Yeah. Well, you
que
can-
bonito.
yeah.
Oh, that's great,
Olha
though.
lá, como se fossem várias águas vivas pulsando, né, de uma forma sincronizada.
Everybody happy?
Oh,
Okay. we're
everybody
moving on.
happy.
I didn't hear this. I didn't hear no. So we're good.
Gotta go.
Tem uma pergunta.
All right. We can keep moving down and forward, please.
Pergunta para você no microfone. Põe pergunta. Uma pergunta, Renato. Essas águas-vivas pulsando, elas são sincronizadas?
Então, esses são os nectóforos, né? Então, a colônia, ela consegue... tem estudos que mostraram que a colônia consegue sincronizar a pulsação desses nectóforos. Alguns, quando ele vai fazer uma curva, eles direcionam o jato para uma direção e conseguem dar um direcionamento para a natação da colônia. Agora, para onde ela vai, como que ela responde a estímulos do ambiente, aí já são perguntas que ninguém sabe, né? Então eles conseguem ter uma certa... Eles conseguem até nadar de trás para frente, né? Voltando. Mas é difícil entender como que ele se orienta, né? Um animal que não tem cérebro, não tem olhos.
Well, I mean, that animal had that house had the animal in, would have been nice to have had a little bit of the look, but usually you can't tell until you
Stop on it.
Till you stop on it.
Okay.
Make sure you don't dust it in the process.
Yep.
I get a very different look view from this side camera, and a lot of the stuff I see in the side camera we're never going to get.
Right.
Or I see it before you actually see it in the other one.
Exactly. That's what's happening. Yeah.
Continuando a descida, então, em direção a 1.200 metros de profundidade.
Ignath.
Quer falar alguma coisa? Vou passar aqui, ó.
Oi, Luiz Felipe, agora vamos torcer para aparecer um peixe, né?
Chamando. Eu já vi que tem gente da Argentina aqui, né? Vários seguidores da Argentina. Vou chamar a professora Silvina Bota, do Instituto de Oceanografia da Universidade Federal do Rio Grande, da FURG, para ajudar aqui na narração da descida aqui. Professora Silvina, junte-se, junte-se.
Stop.
Yeah.
Medusa left. We almost made it to 900 meters. Almost. If we could zoom in on this Medusa, please. It is a little guy.
Sim, tem gente que está perguntando sobre um membro aqui da tripulação, da equipe científica do navio Falkor 2, que é brasileiro. Sim, tem o Léo Alcântara, que é carioca e também faz a pilotagem. Só que agora não está no tempo, no turno dele.
Okay. If possible, Sunny, if you get a little closer, that would be awesome. recognizing this is probably the size of a quarter or smaller. It's like an egg.
Acho que esse aqui é outro.
Get towards the top of that bell to
outra
focus. That
plataforma.
gives us some of the characters we need to look at. Yeah, we're getting there.
Olha, agora uma pequena medusa. Bastante rápida.
Yep. Yeah, he is small. Yeah, yeah.
Provavelmente da família Rhopalonematidae.
Okay.
Esses animais de profundidade, eles não têm a fase de pólipo, a maioria dessas espécies, né? São apenas as medusas no ciclo de vida.
We're okay to move?
Yeah, Dougal could probably do something.
Yeah, I think we get it from the frame grabs. Okay, we can move on, please. Down and forward.
A maioria dos...
You're nearing the bottom of the mesopelagic zone. There's different zones in the ocean that go from the surface all the way down to the seafloor. And that varies in depth all around the world. The epipelagic is the top 200 meters. The mesopelagic is from 200 meters to 1,000, and then 1,000 meters to about 4,000 meters is the bathypelagic. So in our trip today, we will get to just the top of the bathypelagic zone in our quest for biodiversity of the midwater.
Vou passar então a palavra para a professora Silvina para ela explicar rapidamente para nós o que eles estão fazendo aqui a bordo do Falkor 2, na missão Design the Future Tree. Fala, Silvina, para o pessoal.
Hola,
Middle
buenas
red.
noches.
Yeah.
Acá les habla Silvina Bota. Soy bióloga y me recibí en la Universidad Nacional de La Plata. Y así como Julieta, estamos acá colaborando con este proyecto junto con otros colegas de Brasil, ya que las dos trabajamos acá en Brasil.
No. Okay. We're questioning as we submit. Okay, agreed. Shrimp-like creature, very long antennas, but he's not a suggestive.
Están tratando de describir qué animal es ese que estamos viendo en pantalla ahora.
Where are all our shrimp people? On the internet.
Who is this guy? I haven't seen this guy.
Let's go to the deep sea guide.
Están preguntando acá los expertos a ver qué,
Yeah, I was gonna say, if
qué
we can get
clase
some
de
zoom on
animal
this guy
es
for a
ese.
minute, that'd be great, if possible.
Estamos en lo que se llama el nordeste de Brasil, en las aguas oceánicas enfrente al estado de Alagoas en este momento.
Okay. There.
Y acá hay una pregunta de si el ROV va a bajar hasta el fondo y no, en esta expedición el ROV no va a bajar hasta el fondo. Va a quedarse a profundidades como máximo de 1.200 metros. Sí, Marcelo Melo está acá atrás.
Okay, can we, yeah. Sunny, you've got this. Oh, maybe. I've heard of cystolapsis. We have those in the Gulf of Mexico, I believe. Okay.
No, it's a shrimp.
Oplophorids. Aplophorids. That's how you say it. Aplophorids.
Is that what I'm saying?
Yeah,
Okay.
Aplophorids
Great.
is one of the groups, one of the
Then there's
families. Yeah.
Acanthophyra.
Es un camarón.
I don't think that's Acanthophyra. I know what those guys look like.
We don't really have enough detail to know
Yeah,
whether they're
okay.
right shrimp.
We can keep going. Thank you. Alright. Down and forward.
Fala pra eles que vai ter começando mais avermelhados, que são mais comuns, nessa parte, nessa profundidade. E todos estão subindo em direção vertical.
Bueno, ahora estaba consultando acá con el especialista en estos animales, el profesor Renato Rangata, de la Universidad Federal de Río Grande. Y él me estaba explicando que en estas profundidades es más común, vamos a empezar a ver animales de coloración más roja, que es más común en estas profundidades, y están todos en este momento haciendo migración vertical, que es bastante común en este momento del día, que hagan migraciones verticales, o sea, subiendo. El color rojo se debe a que en estas profundidades es un color invisible para otros organismos.
Oh, it is a fish. Wait. Is Marcelo still here?
Olá, Marcelo.
Marcelo, what's this fish?
Come in, Marcelo. Come in, Marcelo.
Acá le estamos
Marcelo,
preguntando a
ven,
Marcelo, que es
ven.
especialista en peces de profundidad, ¿qué pez es ese? Ahora él nos va a hablar
It's a hatchet fish.
y va
These guys are great.
a
Can you get a
contar.
side view? He has these awesome photophores along the bottom. There, you can see him there. This is one of the hatchet fish, right?
Yes, that's a hedgefish. Oh,
And he's got a parasite on him.
Yeah.
Yeah, we're gonna need to stay with him if
Get those
if
cocoa
possible.
Oh.
He has a parasite on the side of his body.
No, I don't do parasites as a
Oh,
professional
can
person.
Yeah.
The tail,
I appreciate that.
he's like, I'm out. There he is. You can see here in the image that we're getting with the video that they are laterally compressed, which means they're flattened side to side, which adds to their advantage in kind of evading predators, right? If you're looking at it head on, it looks much smaller than when you look at it from the side. If we could, you know what I need, focus, that would be great.
Exoskeleton bound.
There he is.
That's
You
probably
can
not
see here, he's got
Pelecus.
that shiny silver that's lighting against the lights from the ROV. Oh, that's great, you guys.
Oh.
Yeah, that parasite's great on the side of his body.
Those would be parasitic copepods.
Parasitic copepods?
Parasita, yeah.
Thank you, Russ.
It's a crustacean.
Yeah, if we can get another
Esse
little bit of
parasita que tá nele é um
footage, and
crustáceo.
then I think we're gonna be good to move forward.
Ele entra na carne do peixe e fica uma parte para fora. Essa parte que a gente está vendo, a parte das gônadas, fica exposta. E é curioso encontrar esse peixe nessa profundidade agora, porque ele devia ter migrado para a superfície. Esse é um dos grupos que faz migração vertical diária.
We may have lost. Okay. We are gonna continue on. That was awesome.
Hatchet fish is a really good representative. Oh, there he is again. He's come back to say hello. Really good representative of one of the midwater fish in the mesopelagic zone. There's lots of species of hatchet fishes, and they all have this similar shape for some of the species. All right, Marcelo, you good? We can move on? Okay, we're moving on, my friends.
Marcelo estaba contando y explicando acá en portugués que ese pez tiene un crustáceo que
Thank
es
you,
un
guys, that's great.
parásito. Entonces lo pueden ver como que le está saliendo del
Nice.
cuerpo y que además ese pez debería estar haciendo migración vertical. Entonces que es bastante raro haberlo encontrado en este momento en esa profundidad.
Hey, you broke 900 after all.
Hey, I wasn't gonna say anything. Didn't want to jinx it. But yes.
Yeah, another hatchet fish.
Bye, buddy. Siphonophore. left. Gorgeous.
Look at that.
I know. If we can get this guy on the left going upwards. He's going almost off my screen, right at the top of the middle. Ooh. All right, Russ, what do you got? What is he?
Siphonophore.
Well, thank you. A Physonex siphonophore. Dougal. I know. So it's a Physonex siphonophore. All of those stringy tentacles are hanging down to capture prey. This is great. Gorgeous. Thank you, pilot. This is great. There is this green yellow to those nectophores.
Definitely. Yeah.
Would we like to? DPIV or?
DPIV, please.
Oh, there's Dougal. Dougal, what is that?
Exactly, that's the right response.
Google wants it.
Holy mackerel.
We didn't tell Dougal all the things we've already gotten him today.
Yeah, no idea.
It is like Christmas for Dougal today.
Ah,
Yeah, that's
misterioso
exactly what we
abissal
want.
só
We want
pode
these
ser o Luiz
nectophores.
Felipe.
If you can zoom in on those, the feeding. Apolemia.
APOLE. Wow, I've never seen one with a yellowish green.
That yellowish green is different. Yeah.
É da ponta. Deixa eu só falar.
What is this?
Yeah, exactly. Whichever way.
I don't know what's giving us the green.
It's not. Are they fluorescent?
Those, the grape side, those are the smigdals, the nectar.
Agora então avistamos um sifonóforo do grupo de Fisonect, né? Próximo de uma caravela portuguesa, né? Um quinidário. Na ponta ali, vocês podem ver aquela parte mais... Tem a parte mais esverdeada ali, né? São os nectóforos. São responsáveis pela propulsão da colônia. E toda a parte mais avermelhada, com vários tentáculos ali, é a parte da colônia responsável pela alimentação, pela reprodução. E ali na ponta... Ah, agora dá pra ver os nectóforos pulsando. Olha lá. Essa cor aí é bem curiosa, né? Tô conversando aqui com os colegas. Pode ser do gênero Apolemia, mas essa coloração tá chamando bastante atenção, as colorações esverdeadas. E agora nós vamos fazer o escaneamento do corpo desse animal com um equipamento aqui de imageamento. Tentar fazer com que a colônia toda, que é uma colônia grande, passe pelo feixe de laser.
That color, right? I have never seen that color on a siphonophore.
Show de imagens agora aqui com equipamento DPIV, realizando o imageamento, né, tridimensional do corpo desse animal. Essas colônias elas podem ser longas, tem registro de colônias de até 40 metros de comprimento.
Right.
Yeah, Echolemias are really sticky. There's that off on the review. Normally they drop a cloud of zooids as well. But this guy's contracting.
Now he is, yeah. This is the first time. He was very expanded when we saw him.
Yeah.
São vários tentáculos, né? Então o animal estende esses tentáculos e outros organismos como
Very cool.
My prediction is Dougal wants it.
Looks like some fire.
Would it fit? I don't think it's gonna fit, right?
É bem curiosa a orientação espiral, né, dessa colônia. Como que ele consegue entender o ambiente para nadar e gerar essa disposição em espiral? Essa é uma pergunta que nós não sabemos.
You get it from the head end because otherwise it may be clogged by the time If you grab it mid-piece or at the other
Yep.
end, it'll be clogged before you can connect it, so.
Do we need the whole thing or just a piece?
We have 4 canisters left, and we're not to
We can go
1,200
for
yet.
the back
Just
of
don't-
the
I don't want to
surface
jam
zone,
the
maybe.
hoses.
Couldn't quite.
Really nice to have that zone, one zone, but if it's not going to drop them, that's what you do about
It's a nice position to be in, having them all full. Get a
Exactly.
jellyfish somewhere out there.
Karen
Agreed.
would want no less.
Just went through the,
Dougal, you let me know. Do you want to collect this?
Lembrando que são mergulhos
Oh, not
diários
yet.
que o
I'm just
drone,
meaning
que o ROV
after
está
you're done,
realizando.
Well, it's more, you know, as we
São mergulhos que
continue to
levam até
scan it,
2
we might
horas.
actually break off a piece
12 horas, aliás, de
for
realização.
the cycle zone.
It'd
Yep.
be really nice to have, like, I'd need a piece of the tail. And it often drops the bits of the tail anyway throughout
Right.
life, right?
So we might pick some up.
Can recover, it's
Like
okay.
this.
Ooh.
This is
There
why.
might be a tail
Agora
This is why.
colidiu com o equipamento.
Right. It might break pieces off.
It would be nice to have a couple of nectophores as well, but I don't really want to collect the whole animal, given
Yeah.
that it might be
Vamos tentar fazer a coleta desse animal agora.
maybe as well.
Right.
Oh, saying that as
So for those at home that are paying attention, Dougal,
Oh,
our
ele se
gelatinous
enrolou
expert,
agora.
thinks that this animal
not sure how
might be
undamaged
pretty
it really is.
old, like over 100 years
fine,
old, due
that's
to the
fine.
shape of the nectophores, which are those parts in the bells at the top. Uh-oh.
Okay, so Marco,
Professor Dougal Lindsay, do
Yeah.
James
There we
Tech
go.
Japão, acaba de comentar que é um animal que pode ter mais de 100 anos de idade em função do número de nectóforos que ele possui, né? Conhecendo a velocidade que ele demora para se desenvolver,
Okay, so Kakani is good.
é possível fazer uma previsão da idade dele.
Can
Sim.
really sting you, like deep sea ones. So
Yeah.
watch it when we are touching that later.
So Google, we should image this.
I don't know yet.
Yeah.
I was standing by.
You read my mind.
Mm-hmm. I've been thinking that for 5 minutes.
Wow, even after that it didn't drop.
It didn't. It didn't drop anything.
Wow.
So are we collecting this or no?
If we were to get, we would like to collect the part of the cycle. The tail.
Almost.
Sim, tem,
I mean, probably in
tem
How- you're
lasers
gonna
que
get-
podem fazer uma estimativa do
The way the
tamanho
other
desse
stuff
organismo.
is gone today.
Because it's not gonna.
It's not gonna break off. If it didn't break off with that.
Unless we like.
Ele não é exatamente uma caravela, mas é do mesmo grupo, né? A caravela também é um sifonófero.
D-sample it.
What do we have in the
Number 3.
There's a T4 and there's a 4M.
Let's get rid of the
In D samplers?
Yeah, the D-samplers are full.
Can you get rid of T-samplers?
We did get you a T4. He's adorable. We have lots of things for you in these samplers.
How many hours since we started the dive? The shadow cam still
I was like,
Oh,
why did we
Alright, what do we,
I
what do we
lost
do?
feed, though.
Do you wanna collect the whole thing?
How many hours since we started the dive?
We started the dive at 2 o'clock this afternoon. Well, 1:30. It is now 7:04. We still have not hit our 1200. We still haven't hit our 1200 target,
Alright, so the
which
shadow
I'm
cam should
determined to
still be
hit.
going.
What was that?
The shadow cam should still be going.
Yes, it should be. Yeah.
On sample number 3, we have a very fine mesh on the rotating sample.
Yeah.
on this one though.
So. Sample number 3.
Like the valve speed.
I'm listening.
has
We lost
very,
speed on
very
this one.
character
That would be
number 3.
Alex. Alex.
It might just be too dark because the.
No, it's
Oh, it's lost?
yeah, it's over there. You can see it, but on this
Okay.
monitor.
I think it's over 10 is gonna be having the one decent.
Oh.
I don't think we should do it.
Maybe a small. I mean, that could just be Weaver, it's far.
But let's see what the experts say.
Probably pull it up for it.
Well, actually, that one's hardwired. So, yeah, I can see the dangling HDMI.
We can't use the left hand manipulator to.
There's HDMI that got
Prop it up either,
disconnected,
can we?
and there's one.
I don't think so. Iris is attached, right?
What happened?
We already spun it around the laser pretty good. And it didn't
Right. It didn't break on that laser.
Who's got?
Snip it first.
Can we get this one monitor
Yeah,
back
there's one
up?
over on this side as well. The HDMI just got disconnected underneath the desks, that's all.
Where is it?
I think
As long as you promise.
this one, maybe this guy.
I guess we
We
gotta
will do
do a nice slow suck.
Oh.
And
Is that gonna get us anything back? There we go.
Once we have a little bit in, maybe try and go up and try and break it on the edge of the funnel.
So we're gonna try to get it from the tail end, right? Yep. Tail end. A piece would be preferred if there's a way to break some off as it's in the funnel.
Agora vamos tentar fazer a amostragem desse animal, coletar pedaço da
Is an
colônia.
option. And there's still tentacles
O ROV
from
está se
another
aproximando para tentar pegar pedaço da porção distal da colônia, né?
Oh, man.
We'll switch it.
We almost jammed the suction sample with the helmet.
Yeah, that's why I'm concerned. I'm a little worried about that, but
No, I mean, like Marco obviously has done this before. You could hopefully get one end and then.
Do you care which end you get first?
Well, that
Assuming
yellow,
you can't break it?
that
or what?
yellow is so weird though, right?
Yeah, we should take the
Let's
next.
take the, let's
Take the head end
Take the
first.
some and some of the tail.
so the head and the tail.
The head and some of the tail. And these things, these things can float for like
Okay.
months, reproducing while they're left behind.
Either end, if you can get into the
You
sampler
can't
and perhaps
regenerate the
break the
front end
piece
though, right?
if
Once you break
possible.
off the
That's
end.
what they're asking.
Oh gosh, that's huge.
Oh Jesus. We cannot take this in the funnel.
Thank you.
Is it still caught?
Yes.
Give it a quick pulse.
Yeah.
And that should chop off the rest of it.
Yeah, but the
We can't go
nectosome
reverse.
is hanging outside.
No, it's inside.
No, the nectosome's inside.
It's inside now.
Ali é
Nope.
a parte anterior da colônia, que tá ali no equipamento que tá
Pilots
sugando.
are good.
Ali é o flutuador, né? Seria equivalente ao pneumatóforo da caravela, né? Indivíduo da colônia que tem gás dentro do corpo.
I don't think we're breaking them off.
I don't think he's gonna break.
Alright.
I don't want to collect him, but I don't think he's gonna break. And I think I don't want to clog up the
We
hose.
should collect him though, we've got,
Do you want me to ditch the?
Yeah, we
I think it's gonna wash off.
Yeah.
All of this is gonna wash off.
Zoids.
Not a touch in the ROV.
I've never seen a color like that though.
Uh-uh.
Have you ever seen a color like that?
Never. Not on any of the other dives I've done have I seen the yellow. Oh, that's better. All right.
Oh yeah,
The green caterpillar.
This was a yellowish green.
All right, welcome back. In your absence, we opened up the detritus sampler and got rid of the small ctenophore that was collected earlier in the dive, and then that large siphonophore that you saw earlier, we got most of that down into that detritus sampler. And now we are continuing on our way down to 1200.
Nope. Nope. Nothing. I'm not stopping until we hit 1200. Unless we see a giant squid. That's a 4am. It's lovely, but bye-bye. We can go faster.
What's the red dot?
What red dot? I
Agora acho
don't
que
know what that
voltou o
was.
streaming.
Actually, Manu, I did see it. I did see it, yes. You were not.
Nós estamos indo direto para 1.200 metros de profundidade, ignorando qualquer coisa que aparecer. Foi isso combinado entre os pesquisadores. E de lá nós vamos voltar devagarzinho, olhando e tentando observar e tentando escanear os animais mais interessantes que aparecerem.
No, it's a darker green.
You can see some of the goop there from the siphonophore. That's what's at the bottom of your screen. So while we're headed down, we'll probably see that waving around. And then once we start heading back up, hopefully we'll see it a little less, not obstruct the view.
Most of the stuff is
It's on mine. I want it off. Take it off. And here you adjust everything.
Oh, nice. Back in, without actually catching an animal, when I saw him back in 1998. It was the only time I ever seen him. It was, man, some years old dogs. There's almost no video. What there is NPSC.
There's a question in the chat that asks about what happens to the animals once they get samples. So we have lots of experts in the room that have lots of goals. So we want to identify them. One of the big missions is to look for new species in this region because the deep sea here has not been studied extensively. So we are going to most likely take tissue samples so we can see what the genetic barcode is, right? To get a DNA sample. Compare the morphology with other organisms in the group. They will be preserved at the end. We understand that if we're taking an animal out of the ocean, we want to use as much as possible for science. So that's what we do. We keep everything that gets collected and we will preserve it in the right way. I'm not sure the best way for siphonophores to be preserved. Do you know? Somebody's asking what kind of preservation.
3% formalin.
3% formalin works well with siphonophores. So that's what we will do when we're done imaging, investigating, sampling the animal. Thanks for the question.
Professora Heather Judkins respondendo aqui uma pergunta do chat, né? O que é feito com os animais que são coletados? Então eles são usados em estudos, experimentos, né? Eles podem ser trazidos para a superfície e ao final eles serão preservados para estudos de biologia molecular, né? Então eles vão ser preservados, eles podem ser preservados tanto em formol para serem armazenados em coleções biológicas, como também em etanol para serem usados em estudos de genética, né? Para saber se é um organismo conhecido ou não, né?
Maybe.
Other pigments.
Right.
End up looking like that.
What do they eat?
Apolemia?
Yeah.
Pretty much anything.
Yeah.
They're pretty strong, very strong venom.
Okay.
A lot of nematocysts.
Right. Okay.
Some will eat fish, they eat other jellies.
Right, so it might not come from diet.
Crustaceans,
No,
no.
right.
Definitely not. Yeah. If it was only in the radial canals of the nectaphores, it's
Maybe.
a possibility.
Right, but that whole thing
But
was.
this- yeah.
Right. Yep. Symbiont? Could it be a symbiont?
I wouldn't say so.
Okay. I'm just looking at my other options.
Yeah.
This is why I don't study siphonophores.
Hey Google,
know the exact
don't you think
citation.
it's why?
No.
The normal one?
No.
Why is not?
Apolemia uvaria has a combination of palpons, long white ones and short brown ones. It doesn't have the fluorescent.
The fluorescence is
Yeah,
quite,
the
quite
nectar
different.
cores. I didn't see any nectosomal zooids, like the nectosomal tentacles between the bells.
Didn't have any normal, just collected.
I didn't see any at all, but Apolemia does. So if it didn't, then it's definitely Apolemia from all the
Yeah.
rest of it.
Yeah.
But it might be the first one ever without nectosomal tentacles, which is definitive for the family right now. And right now, all the Apolemias are a single genus,
It's
but this
single
looks
species.
different enough that it could be something else. Like it,
Yeah. Cool.
How about pseudoapolemia?
Yeah.
Or para-apolemia?
Pseudapolemia.
Welcome to the bathypelagic zone. We are finally at 1,000 meters of our 1,200 meter dive today. We are going to continue on down to 1,200 meters, and then we're going to work it slowly for the rest of the cruise
Back up
Man,
towards the surface.
You got to think so many things while I was asleep.
Yeah, this is a Christmas day for Dougal for sure.
Bueno, acá hay una pregunta sobre microplásticos. Nosotros no estamos muestreando microplásticos, pero claro que si encontramos algún plástico, principalmente
Somebody
en la
else asked
red
a question on the
de Tucker,
chat.
que pasamos después de que el ROV sube, las vamos a colectar. Hasta ahora, en los que hemos hecho, no hemos encontrado plásticos.
So we are looking for other things if by chance we've seen them. So far on our dives, we have not, which is encouraging in itself, so that's great. But we do record it if we see it. So thanks for the question.
That's the nice thing about deep
Estamos yendo directo a 1200 metros para después empezar a subir de nuevo, ir muestrando lo que encontramos en el camino.
Manu, I was protecting.
I am sure it was considered.
No, not by me, Manu. I knew the 4am was to be protected at all costs. Nope. I had you covered, buddy.
I did not suggest it once, you will remember.
But I saw your face.
Yeah, cover the whole forearm.
If I had any of the ROV controls over here, I might have slipped.
Oops.
So glad.
Ooh, what's that?
Oh,
Stop.
FS
We do have to
going.
stop. Darn it. My fault.
It's there.
I know. We're getting there. Sorry. And our quest for we were never gonna stop.
No.
We did make it almost 1,000, or yeah, maybe. We're trying, team. It's top left still.
3 meters.
I don't see it on my screen yet.
I'm not sure.
It was a round jelly-like organism.
Yeah, it's another forearm.
Anything?
Log it.
No, you guys, we're good.
It's probably still there. Just slightly forward and keep going up.
Slightly forward, keep going up.
And
He said most likely
we
there.
need to rotate a bit left, Google, or you think we're good?
Oh, he's right here. He's in the middle. All right, this was worth a stop. I'll give you that. We're not ditching anything for this.
Entonces
But if you guys can film
van
and
a
get some
parar
great video, that'd be
para
awesome.
mostrarla porque realmente es
Between the statuses,
muy diferente.
are they groups of 3 tentacles or 5 tentacles?
Okay, so we're gonna need to zoom in pretty close.
And is
And
the
this
top of
would
the
be a
bill
good-
round?
this would be
Yeah.
a
You
good
could keep it
target
right
for
there.
Okay.
Yeah.
What is
Dougal, is this close enough? We're zooming in more.
Yeah, this is good. It looks like it might be 5.
It,
1, 2, 3, 5.
Yeah.
5.
So I'll put it as Paraphilopsis right now for our ID. We can do a 3D scan with the light field camera maybe?
It'll be too big, won't it?
Le van a hacer un escaneo 3D. Ahora Renato nos va a contar un poco sobre esta medusa diferente.
Mission here is looking to see what each of the technologies is capable of and which is best for what.
So Kokani,
even if it's
DPIV or
Iris?
Iris.
Oh, yeah,
So we're gonna go Iris on this in a bit, but let's get some video. You want video first, or doesn't it matter?
Agora uma medusa
Video
cifozoa
first, let's get some more images or
do
print
grupo
grabs if
Coronata.
they need 'em.
É um animal que tem um sulco
I would
ali
love
na
for him
umbrella.
to
A gente
turn
pode ver
around a
que
little bit.
tem uma parte da umbrella que é mais pontuda, que é o
You can
topo da
just show us
umbrella,
the
e
top
ali
of his
abaixo
bell. That
é a
would be
margem da
helpful.
umbrella que a gente consegue, é possível ver a inserção, né, de alguns tentáculos.
Yeah. This is a Paraphilopsis jellyfish.
Provavelmente
Paraphilopsis jellyfish.
uma água-viva
Thank
do
you,
gênero Periphyllopsis,
Dougal, does
um
that help
animal,
with the bell?
um animal vermelhado, né, como é comum animais dessa profundidade terem essa coloração, uma coloração que deixa eles camuflados na escuridão das profundezas e estamos tentando.
After we finish filming this Paraphilopsis, if we could also try zooming in on the apolemia that's sticking out of the sampler, getting some really tight in-focus zooms of the zooids.
Okay.
While it's all still in one piece.
Yep.
Well, too big to suction sample too.
Oh, that's looking good. That's looking good.
Yeah, this is good.
Separate tape aside. Sorry, say again. Keep it separate. I'll take the side. Sorry, say again. Keep it in view with the set and out because I need the side camera.
Oh, okay.
Check unless download saving.
Oh,
I mean, I guess you do.
Okay, go ahead.
It's okay.
Yep.
Oi? Ela tem uma consistência mais resistente que as águas liso. Oi? Sim, sim.
Adam? You got this, pilot.
Oh, because if we take get rid of this, it'll default.
Yeah, essentially like
Is there any other setting I need to do?
pretty short exposure. It was something bright.
It was one.
Yeah.
Above? Still up?
Just pick those lights back on, so I'm fine.
You can leave whichever lights you like on.
Yeah.
Lights don't matter?
Nope.
Doesn't matter. We just need lots of it. But also cut that much, but
Light up.
A medusa? É. Periflocos.
Zoom in, zoom out.
Mas falei? Falei.
We may have lost it. We are looking. It's trying to evade us in the dark and succeeding currently.
Aparentemente nós perdemos essa água-viva. Acontece.
We'll give it another minute and then we'll hover here for just a minute or so to see if it comes into view. And then if not, we'll continue on down. We did get some great images and frame grabs, so that's a plus. I think we did too. I don't remember. It had to have been in the. Maybe he finally moved. Like he did it. It did it. Okay. I think we've lost him. If we see him, we'll stop. And we're going to start descending back down to 1,200 meters. We're getting there. Getting close. Thank you, pilot. We can go at the same speed we were going before, so pretty quick to see if we can get down to 1,200.
Agora sim, nós vamos voltar para a descida até 1.200 metros. Perdemos a água viva, não conseguimos fazer o escaneamento. E estamos indo um pouco rápido e vamos subir devagarzinho, né, tentando registrar animais interessantes que aparecerem. Nesse momento nós estamos indo na contramão da maioria dos organismos que estão subindo, né? Eles fazem a migração vertical diária.
We lost him?
Yeah.
We're on our way.
We're gonna move him our way back down. Yep.
Our eyes are closed now, are they?
E a maioria dos animais, de um modo geral, estão subindo para a superfície, em direção à
Did we do a
superfície.
zoom in of the apolemia?
We lost it. We lost it.
Hold on.
What's?
He's breaking free.
Jailbreak.
We have a break free. Stand by. If we can get him back in the decoupler, that would be great. We've still got it. Don't lose it. Everybody keep your eyes on it. It's going off to the right. Top corner.
Catch and release.
Nesse momento nós perdemos um animal que havia sido coletado, um animal
He will not be contained.
com corpo duro, né? E quando a garrafa foi fechada, esse animal não permitiu o fechamento completo da garrafa e a garrafa acabou abrindo.
Well, we got some pieces still attached to the D-sampler.
Should we try to get those in the
Stick 'em back
D-sampler
in
or
the
suction
D-sampler and
those?
close the lid again.
What just went by below us?
Those I think are I think those are still attached to the sampler. How did it open?
Oh, you don't that.
Got it. Okay.
Be free.
He would not.
Is the forearm still in there?
Yep, forearm's still closed.
Wrong switch, man.
Alright.
That was what I was planning.
Google paid them to do it.
Wrong one.
Backfired.
Do we want to slow down, get the manipulator to pull that stuff off the D-sampler?
Right, that's what we're starting to talk about.
Yes, yeah.
If we can do that, that would be great. Sunny, is that an option? To try to use the manipulator to get those feeding tentacles back in the
We
coupling?
need the,
But yeah, just pull them out into the water column and then you can suck them up the D-sampler.
or in, yeah.
Or in the
the
Or in
D-sampler
the suction
itself.
sampler at this point?
Yeah, they're probably not going to come out of the suction.
You can attach it in the
Right, they're
D-sampler.
gonna get stuck.
Like, go
He
up
doesn't think
and...
they're gonna come out of the suction sampler.
Well then,
Yeah.
fine. So
I'd do D-sampler.
D-sampler, if there's a way.
even a little bit there.
Yeah.
Marco.
Some of that tissue.
Right.
Por algum motivo a garrafa abriu e perdemos um organismo que tinha sido coletado. Agora o pessoal está discutindo aqui o que vai fazer, porque tem um pedaço dele ali ainda enroscado na garrafa e é possível coletar esse pedaço para pelo menos estudar o DNA desse organismo. Né? Isso que vai ser feito.
Okay, and
got some in there.
close.
Should we just try to close it?
All right.
Good. All right, we got some in there.
Okay.
Should we just leave that like that?
Yes.
You guys are good?
So this is an original thing to catch and release and get
Yep.
some tissue. from
Catch and
the
release
exam
is
lens.
our.
So we're on track.
We're good.
On track.
No harm, no foul.
All right, I want to know who the leading card is. Or not the Tinophore, sorry,
No, both the Tinophore
And the siphonophore. Let all the gelatinous
Dougal's
go.
reassessing that, you see.
All right, we're gonna head back down to 1200 because I am determined before the end of my watch ends that we are gonna get to 1200 meters.
At least, so we've gotten a few stands and then
15
more,
more.
so
Think we can do it?
that isn't complete lost.
I'm not stopping.
Can you reconstruct a neck?
I don't care what comes. Exactly.
To get what
I
it
might not
looked
even
like.
stop for a split of this.
Maybe it's a
We, I
That's
mean,
how
I'm sure
serious.
Jamie
Oh,
could do
wow. I know. You heard me. That's serious. We got to get there.
Yeah.
Yeah, okay. All right, all right.
Can't we just feed all that information into AI and
Well, the models aren't great right
I
now.
know, yeah, I know.
We've tried.
So we can try getting this face, at least like
Well, we would get
as a test
mesh.
on us would do. So yeah. Well, that's good for the flow and the idea and stuff. And then if I get enough zones from the tail,
There seemed to be a bunch in there.
out on tips and all the stuff that won't show up in the image, then
We should have enough there.
the yellow green we can't ID because we didn't end up with a hyperspectral camera and a shadow cam and you guys don't have a hyperspectral camera?
We do. I mean, Paul's got one, but I don't, they were using this right now.
But if you could like, I mean, rad samples might be able to encapsulate so that the illumination is completely controlled, and then image, even if it's not an image, just like a pigment profile kind of thing, but it was in the space. Would need more power and comms, I guess, which we don't really have.
Yeah.
Nothing to see. Nothing to see. Mm.
It's okay, don't worry, it's just my phone. Well, I mean, hopefully we've got this, and we also got material, and we've got section sample.
There'll be pieces. Marcel.
You definitely, and there's some still on the.
There'll be a couple of
There's
tints.
still some on the. There's some on her camera still too. We got options.
Yeah, there definitely is stuff around.
We've got options. Crustacean.
I wonder what the yellow green was. That's like, that's completely new. We had a camera calibration and we had the RGB there. Something helped those.
We did the light balance today.
Yeah, we did.
We have 100 meters to go till we hit our 1200 meter mark. And then we'll slowly head back up.
We also have a lot.
open my eyes soon. I'm gonna go to sleep.
You only have 80 meters to go, my
80
friend.
more meters.
80 meters. Stick it out. Stick it out.
I'm actually relieved.
Now we still can get
In
stuck
case
by the
something
front of
comes
the
up.
vehicle.
Right.
I always find sampling.
We have plenty to show for it. You can do a lot with those feeding tentacles. Yeah, I think you've got plenty. Yeah, you can describe it with that.
And we've done that. Now
And we have video grabs and video. We're good. Maybe we'll see it on the way back up. I was like, I don't want to see it again.
Wishful thinking.
Yeah. I like the optimism though.
We encapsulate
Hey,
small
that's what I'm here for,
spheres
kid.
of fluid
That's what I'm here for.
with the tracker, and then we go pick them up by small buckets.
I mean, Bruce wanted me to think more and more about how we tag.
How we tag some of these. That's what I want to think about. I've been trying to figure it out for the deep sea squids, and I haven't figured it out yet.
Station you can plug in and out of that takes off payloads.
Yeah.
So,
Control.
I mean, that would.
I need a sharp this baby.
Doesn't a bar have something like that for experiments where you can like go in and plug them into?
The midwater restaurant.
Yeah, it's the same kind of idea, right? Only you actually have to have a base, fly it, plug it in and detach it, take blank. Well, to get more desampling. The time you may spend trying to build that, actually do it, may offset simply pulling the vehicle and putting it back in. And if you looked at bang for the buck.
2232.
Mark,
Just want to give
how
them
many
a
D-samplers do we have on the wicket?
12.
And then 20-something sections here?
Yeah, 20 suction bucket.
Oh, you've increased the size of suction sample?
Thank you, sweetie.
To 20.
Yeah, because we've added, it's the mini ROV section sampler and then the original Doc Ricketts one, right?
That's correct.
Yeah. So this other ROV, they let me fly more. It has a smaller, it's basically I think it's the design that we see in SOBastian.
Oh, with a bigger carousel.
Well, it's a smaller carousel, so it's the size of the carousel that we have here, plus the Ricketts one, which is bigger.
Oh, so smaller canisters or the same number of canisters as ours?
There's
So this is the
like 10 or 12 of them, they're smaller in size.
Yeah.
Equivalent to the size of these.
No,
And then we have bigger ones.
I mean, the ROV is still limited to what,
6 meters.
ROV is actually 12.
12 D6.
So close.
Yeah, two's pretty restricted.
Yeah, I thought we had 4.
I think it was just.
Team, we have hit 1,200 meters. We are at our target depth. Woo-hoo. Thank you for getting us there. We can now open our eyes, start annotating again. Not that you stopped anyway. So we're gonna go up, slow, forward, and find cool stuff. Thanks, pilot.
Oi, pessoal, aqui tem uma troca, o Renato saiu.
We will be
entrou eu,
diving
Marcelo.
today until 2 a.m. local time. We're off Salvador, Brazil. And now we are gonna slowly scan back up towards the surface for all the life we've been finding so far.
E a gente chegou na profundidade que a gente queria alcançar, de 1.200 metros. E agora vamos começar a subir lentamente, procurando coisas.
Middle low. Middle low looks like a Medusa, perhaps, or a house. Medusa or a house.
Yeah, it's a house.
It looks like a house.
Can we zoom in?
Yep, we're working on it. I would love lights to help our cause. Thank you. Zoomy zoom, if you can, pilots, that would be great.
I'm not sure. It doesn't, no, it's not very structured, but it might just be that it's very transparent.
It looks very odd to me.
Yeah, it could be.
Yeah. I think it's that transparent.
Could also be a piece of, that's what I'm trying to figure out.
Or is it that far away?
That's some-
It's close.
That's probably a camouflage.
No, it's pretty close. Yeah, it looks like a house. No. Yeah. All right, we're going larvation house.
Oi, Luísa. Pessoal de Buenos Aires.
Yeah, a pteropod feeding net.
Córdoba.
Oh, yeah. Okay. All right, we're gonna keep moving slowly. Yeah, I forgot about that mucus that the pteropods do.
Pilkies also create kind of a mucusy, yeah. They do. I know some of the culinarians do as well.
Yes.
Yes.
We've been
so
trying to scan it, and they drop it.
really quick,
Yeah.
you know.
Yeah.
I mean, they gotta have, I mean,
They
that,
do want to scan it.
yeah. I mean, looking at the mole parts, it's not like it's a predatory.
Even rosarian.
Yeah, I've seen rhizarians and, just on big mucus
Oh,
sheets
yeah.
that kind of scavenge along.
Okay, who's good way? I don't know. First choice.
How about the shrimp?
Like the
There's a
big-
poop
big,
goes down
uh,
this, it's got this big long poop hanging off it.
I see we still jar one open.
Looks like a house.
Unless you got something for that.
Small larvation house. Hi.
Oh, good point.
Okay. Did you correct me?
Yeah. No, I didn't.
Okay.
No, that's fine.
Good call. Alligator.
Yeah.
Okay. Forearm going down.
I'm gonna capture it.
Okay.
I'm just gonna sit there.
I wanted to correct my description of where we were. We're more north of Salvador at this point. So offshore of Brazil, north of Salvador, Alagoas is what Marcelo is telling me.
Yeah.
Thank you, Marcelo. Can you guys stop on this? Vertical, it's a fish.
Apareceu peixinho agora.
The head looks cute.
It feels fishy to me. But let's see. Let's see who wins this one.
Either way, it looks fishy.
If we could zoom, that'd be great. Might be a Ketignath. I think you might be right. No, yeah, that's a Ketignath. Yeah?
É, não é peixe, é Quitognatha.
Yeah. Yep. Yep. Good call. Thanks, Russ. We are good to keep moving. The Ketignath is lovely. We are going to venture forth.
Yes.
E nesse momento a gente está ao largo de Alagoas.
Yeah.
Nós estamos
There's no
fora da
more
zona econômica
decouplers.
brasileira, em águas internacionais, mas ali na mesma latitude de Alagoas.
It's a great question. Dougal, are we collecting Niskins at this depth? Aren't we supposed to collect Niskins? We have not done anything yet with the Niskins this dive.
I would say probably yes.
And what, every 200 meters? How many do we have? Are there six?
Six.
So every 200 meters would get us to the surface.
We get lots of Apollinia
So you
DNA.
want one at 12 inches?
I think that would be great.
Apareceu camarãozinho ali
Sorry, my
agora.
friend.
Yeah, we're at 11:19 seconds.
Yeah,
At least
I
we
think
did.
we're okay here.
We still zoomed in.
That'd be great. If we can get one Niskin now, and then we're gonna go up
So-
every 200 for another Niskin.
Yeah.
Thanks.
So,
Thank you for remembering that.
You look with this eye camera to the left, to the flags.
Yes.
É?
Sonny, if you could zoom out for searching.
Uh,
You need to do the Niskins. I'm sorry.
Então,
Thanks.
nesse momento agora a gente vai usar a
I
garrafa
don't
de
think I've
níquel para
been
coletar
here
amostras de
for a
água.
Niskin attempt.
E a
Good
cada
luck.
200 metros a gente vai coletar uma amostra de água. Essa garrafa é uma garrafa oceanográfica especial para.
So what we're doing right
ela é
now
controlada
is
remotamente.
we. Oh, sorry, Marcelo. Go ahead.
Ela é controlada aqui por dentro do laboratório. E aí a gente consegue fechar a garrafa no momento que a gente quiser. Ela desce aberta e quando a gente quer, a gente dispara ela fecha e traz a amostra de água para cima.
Okay, Heather, go ahead.
What did they say? Once it fires, it's coming.
Yeah.
You have to record this, I think.
Yeah.
What do they do?
So we tripped 1 and 2.
Okay. So then, so 1 and 2. That's Niskin 1 and 2.
Was that a Niskin go fire?
Niskin 1 and 2 just got fired at 11:97. So should we go every 400 then? What do you think? Is this for Sylvania and Julieta? Is this stable isotopes or eDNA? What is this for? All things water?
Yeah.
So what would you like? So we just did a fire at 1200 and we have 4 more bottles to get to the surface.
One in the oh, I guess you can drink. The maximum chlorophyll layer.
Do we know where that is?
50 meters.
50 meters?
More or less.
Okay. So then that's 1. You got 3 more.
In the bottom.
Jump in.
Close to the
You want,
surface.
so you want all at the bottom and all at the surface and none in the middle? Okay.
Really?
So-
Setting seems to be working pretty well.
So you want 3 now? 3 at 1200 and then 3 at 50? Or 75 or something? What do you think? 50? 75? 50?
50's okay.
Okay.
We going down.
So now, so Joel, we need 1 more Niskin at this depth. Yeah, you're making some decisions on the fly for what they need for water. Of course. Thank you, Marco. Thank you, Sonny.
Yep.
So the 3rd Niskin bottle should fire here. Is great.
All right, now it's 2 is
Yeah,
low.
yeah, put it 27. Put it 27.
Buenas noches, gente que está desde Buenos Aires, Argentina y las diferentes provincias. Ahora está hablando Julieta. Sí, tuvo que salir un ratito, pero y me imagino que ya vuelve. Ahora lo que estamos haciendo es colectar muestras de agua a diferentes profundidades. Ay, volvió. ¿Querés tu lugar? Vamos a colectar muestras de agua para filtrar para análisis de isótopos estables a diferentes profundidades. 3 ahora acá, esta profundidad, cerca de los 1200 metros. Otras 3 cerca de los 50 metros, donde se encuentra la máxima concentración de clorofila, y otra cerca de la superficie. Ellos consiguen, los pilotos del ROV consiguen cerrar las botellas a la profundidad pedida, a la profundidad necesaria, todo controlado desde acá, desde el control. La idea de esta inmersión era llegar hasta los 1200 metros de profundidad, y ahora, luego de haber llegado hasta el fin, vamos a volver a subir despacio para ver si encontramos algunas cosas. Oh, ahí.
If you could, that would be absolutely my bad. I am almost done with this pencil. Here you go. Pencil.
There it is.
Ahí
Hello?
parecen que encontraron. Oh.
It looks like a bathychorus to me.
The tentacles are
Just
all
saying.
punching and spread out.
It looks like a bathychorus, maybe, yeah.
We don't have anything, it's like here.
It's a bathychorus, yeah.
All right. Gentlemen are saying it's a bathychorus. This is a rhizarian, yes?
That'll be in your speech, just if you suck him up.
Oh, would you like to suck them up in the suction sampler?
Parece que lo que están viendo ahora acá en pantalla es un bathychorus,
Thank you.
una pequeña agua viva. Ahora un Renato les va a poder contar mejor sobre.
That ring of the stomach and the marginal tentacles.
Essa é uma água-viva de um grupo que se chama Narcomedusa. elas têm os tentáculos apontados para cima, né? Então ela tem uma posição bem típica de tentáculos, as medusas desse grupo. E vocês podem ver ali, são 4 tentáculos, todos apontados para cima, juntos ali no centro. E o professor Dougal Lindsay, do Japão, está nos dizendo que pode ser até uma espécie desconhecida da ciência. Estamos tentando olhar mais de perto o animal, né? Veja ali como ela pulsa como uma água-viva típica mesmo. Autoporping, yeah.
The disc is weird. Dougal, can you explain this animal? Or Russ?
Yep. It's a Bathychorus. It's the only described member of the genus comes from the Arctic Ocean, Bathychorus guillone. And it has primary tentacles that bend like an elbow.
Yeah.
And it's got small secondary tentacles. You can see
I
them
see 'em,
flanked by the statocysts on either side.
On the sides.
On the edges of the bell.
I see it.
Usually,
This is
the
great.
species from the Arctic has 3 stomach pouches between each of those tentacle pairs.
Yep.
This guy appears to have only 1.
Right.
It's
Very
eaten
cool.
something that's slightly orange, or it
Yeah.
could actually be something from the species.
Yep.
And this one's a lot more pointy than the Arctic
Other ones we've
species as
seen.
well.
Okay. Thank you, Dougal.
If
Can
we
we
can
go
suck
to
him
suction?
through, yep,
Yeah, let's go
and
for
get
it.
some
If you
shadow
guys can
cam
do this,
images.
that'd be
Professor
great.
Dougal explicando aqui.
Don't lose them.
É um animal que tem 4 tentáculos principais, né, voltados para cima, mas na base, na margem da água-viva, tem tentáculos menores, que parecem com verrugas, né? É possível ver essas verrugas ali. É um gênero que tem uma distribuição no Ártico e nós aqui nos surpreendemos de ver esse animal aqui, quase na região, quase no Equador, né, muito mais próximo do Equador, porém em águas profundas, onde as águas são geladas também, né?
Here he is. Did you want me to try and extend it? We got it in the wash or something. Oh, yeah, he was up off my screen. He's up.
Estamos tentando fazer a captura, dessa água-viva, dessa medusa, com um aparelho que faz a sucção e que leva ele para uma câmera em que o animal é fotografado. Não temos mais garrafas disponíveis para trazer nenhum organismo, então ele vai ser fotografado na câmera, que tem uma câmera de acrílico com câmeras fotográficas dentro, e depois liberado.
Here he's back in. Yeah, I see what you're saying. Yeah. I got you. Right, it's below. It has to be below. It's below. I just watched it drop down. There you go. I know it's tricky, isn't it? Yeah, this guy's a little. He moves so well with current. Off the screen again for me. I still haven't seen one. Up top here.
new species.
All right, he's right above it.
Yeah. So we can even give it a name if you want.
Let's do it.
Something
Let's do
Brazilian.
it. Let's do it.
Yeah.
Name after me. Alagoana.
Come on, should we name something
Oh.
Carnival?
No, those are the feeding tentacles of something. I haven't seen them come back in the screen.
Yes, up and
No, we might
Because
have
it's got the things on the head, yeah.
Can't be alive. Zico, yes.
I think he might be gone.
O professor Dougal Lindsay sugeriu darmos um nome a essa água-viva, que pode ser uma espécie nova, não descrita, um nome relativo ao Brasil, né? Baticoros caipirinha, Baticoros Pelé, tem vários candidatos aqui a nome para uma água-viva não conhecida ainda pela ciência.
If someone in the eDNA can find a Baticorus sequence, then
There you go. We'll
we
use
can at
that.
least strongly suggest.
Actually, according to the code, you can describe it as
All right, if we could
You can,
angle the
yeah.
camera forward and then zoom it so we go to that search mode again.
Well, you can.
Yeah, you
And
can.
up
You can.
and forward. Fabulous.
I reckon that's.
If you record a coelacanth, of course, I'm going to describe it.
Yeah. I think if it was like the fauna of, and then there was a species description embedded in it, maybe you'd get away with it. But just that's new species of.
But you can.
Yeah.
You can set it the features the holotype.
Yes, yeah, you can.
And designate the features the holotype.
My personal philosophy is I always want a DNA sequence that goes with that. Images and DNA sequence, I'm 100% on board.
About specimen and no DNA sequence.
Specimen and yeah, it's still okay. But some of the groups, the morphology is not enough to tell them apart. And in that case, you know, it's a limited value really. You have to redescribe it with a sequence later, which is turnaround.
Yeah. But I think I'm almost there. I've got 8,000. It's probably enough. Test set. It's a waste of effort. Yeah. Oh, that's
Got it now.
threshold.
Everybody comfortable with how this is?
Now if we can go forward a little bit more so we come up again, up on top of
Oh,
the...
stop.
Oh, stop.
Stop, stop.
Cresota
Stop,
Milzai?
stop, 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 stop. Oh, stop. We would love to zoom in on this organism in the middle when you guys are comfortable.
It's funny, there's something beside it too, and then we have another reducer.
Has he caught something? No.
Oh,
That one, huh?
yeah, that's what we want, this guy.
But there was another
Yep, the
reducer
tiniest
beside it.
little things we find for you.
Any babies inside?
We just had some huge ones earlier, but you guys were not here for it. If we could zoom in some more, that would be great. When you feel you can get there. Yep, yep. This is lovely.
Wow.
Dougal, you happy?
Yeah, focus.
Are we close enough?
We got a focus one.
Yep, if we could
looks good.
focus a little. There we go.
That's good,
Yeah?
that's good. They often have little babies on the inside of the bell near the stomach. Maybe that big white thing is an amphipod. Maybe it's something gonadal. I'm not sure. Yeah.
He went
nice.
up.
Thank you.
It's great. Do we want any more video or any more footage?
I think
Since we're
good.
here. Okay. Good?
Yep.
Okay. We're moving on. Thanks, you guys. What was the
I
species,
think that's a
Dougal?
baby. Cresota milzai.
Cresota milsa.
Cresota milzai,
Crossota.
named
Got
after
it.
Claudia Mills.
É uma
that's
espécie
awesome.
de Crossota mills. Uma
Cresota
espécie-
milsa.
Ah, que lindo esse animal. Notação
Yes,
muito
please.
rápida, né? Pulsação.
Forward, zoom out, up.
It's one for Ricky Kun, my student doing tracking UGC right now. Are you happy with that?
Uma espécie Crossota mills, espécie de traquimedusa.
You almost need a like dedicated class for collecting machine learning data. Like, oh, do you have it from that angle yet? Kind of thing. Oik.
Don't have to stop.
No. I'll scream.
What?
Don't worry.
It will scream. Yeah, that was just his casual. That was his casual annotate. It will be a scream if you really want something. I didn't have to look up.
What?
It's much more interesting that show.
Oh yeah. Yeah, the Keat.
Keating Nash.
She's long forgotten me, yeah.
What is that?
Where?
What did you say?
Ooh, Keating Nash.
Keating Nash. Yep.
That's what it said.
Hairy jaw. Basically.
It's a little Skolix, right? Is that right? His mouth parts are amazing.
Octobedema.
Hmm.
To bash this pouch.
Jelly coming up in the middle on the
Slow?
right. Oh no, it's copepod. Sorry.
Oh.
Oh, it's a nice copepod. though.
We can
Look
look
at
at
that.
it for a
We can
second.
look at the copepod.
We'll give a little copepod love for a hot minute.
Oh, it's a beautiful copepod.
That's what I'm
yeah.
saying. See?
Zoom in
Everybody
on it.
gets
Share
some
this
love.
with the world.
Copepods are extremely important to the world, literally. They are major connectors in the food webs of the oceans, both shallow and deep. There's something like 27 to 30,000 different species of copepods around the world. That's a loose number on my part.
You get a better idea?
And again, they're in the
It's probably a
arthropod
mega
group.
calandid at this depth. To show
The
up like this.
scientists are taking a look at it
Really,
once
really
it gets into some
visual.
focus to see if they can get it to a family or a group.
She brought a picture and just like set it up on your desk.
Again, extremely important organism to our world's oceans.
Yeah.
They
It's a
eat lots of the smallest
Mega
of things,
climate.
a lot of this marine snow and small organisms that we are looking at as dots.
Mega
They're eating. Oh, that's great, you guys.
Radicale.
And then in turn, they get eaten by lots of things. The Keating maths we've been seeing and talking about, for example, they eat
If it
these
was you
guys.
heated, you'd see kind of the lyre shape.
Esse agora é um copépode, um animal que mede poucos
Mega
milímetros de tamanho,
Calandid.
mas
Mega
tem
calandid.
uma função muito importante nos oceanos porque ele serve de alimento para vários peixes e outras espécies.
Yeah. Are we happy, Russ? Can
Talvez um dos animais mais abundantes do planeta.
Are you sure? Yeah. Okay. We can keep moving on. Thank you, team. All right, zooming out.
E por serem animais tão comuns, às vezes a gente não dá muita atenção aqui, né? Ficamos com foco nos animais mais raros.
Yeah, this is perfect. There you go.
We want the snow coming at us, and we want the widest field of view so we can see in the
Things-
search mode
Right.
things that are further back.
Combination of Star Wars driving and Men in Black windshield bug roadkill as an image to have.
It's an image that could stick with you. That's what everybody has been saying this. Yeah.
A little bit more forward.
Yeah, I like the idea of forward more. I want to like we're in a snowstorm, like it's coming towards us. Not too fast, of course. A gentle snow.
Just entering hyperspace.
Yeah. Dougal good,
That's
yeah?
it, that's it.
This is great.
I'll just hold that for 5 hours.
I think we're less than that at this point.
Yeah, your hand
We got 3.5.
cramps on the joystick.
No, we do have 5
after.
hours.
This is recorded, right?
Mhm.
All of this week.
Yeah.
Yeah, this is great.
Mhm.
It's good stuff.
Yeah, no joke.
I can't see the gamarins you normally have swimming around.
Medusa. There's a Medusa. He was little.
Yeah, not for them to be worthwhile.
Agora estamos indo reto, né, nessa profundidade de mais ou menos 1.100 metros, né? Uma profundidade já fica abaixo da camada mesopelágica. Opa, passou alguma coisa. Parece uma casa de larváceo de um oicopleurídeo.
So I guess already been.
I think
It's
it
already
was.
been, sinking.
Yeah, it was a sinking house.
É.
All right, so we're good?
Yep.
Yep, we're good. Continue.
É
Thanks.
possível ver várias partículas, né, que compõem o que se chama de neve marinha. São animais mortos, fezes de animais, pedaços de corpos de animais que vão caindo, né, e muco que eles produzem. E isso é a forma chamada neve marinha que abastece os animais do fundo dos oceanos, né? Ao mesmo tempo que ela faz essa transferência de matéria orgânica da superfície para o fundo, ela acaba sequestrando o carbono e colaborando, né, com o armazenamento desse carbono em áreas profundas dos oceanos, né? Então o oceano tem esse papel tão importante de absorver o carbono atmosférico. É um processo que nós chamamos de bombeamento biológico ou bomba biológica. O professor Marcelo deu uma saída. Agora. Estão dando oi aqui pro Marcelo. Lembrando que são 12 horas de mergulho do ROV, então a gente faz o revezamento, né? E tanto a tripulação que faz a pilotagem do ROV como os pesquisadores se revezam para sempre ter alguém participando aqui do mergulho. Quando? Ah, não, põe vocês aí, põe você também. Ah, tá.
Dougal? Yes.
Por você também, Julia.
It says, someone tell Dougal that Maxie told me to tell him hi, and they still need to fix the
The
There you go, ID. His words, not mine.
Yeah, I told you. I shared that message.
Oh, okay, you've done it already, sorry. Looking
Thanks,
forward to it. No problem. He's been embarrassed in public now, that's good.
Thanks, Lisa.
Who's
Yeah, they
you
got
know
some-
this person?
Added to the to-do list.
This is from Larry.
No, a long-term, like, live stream.
Okay.
Showed up in the.
No, no, no.
She knows a lot
This is
of
good.
the jelly, right? It's just
We're going now? Yeah, we're good.
Inaccurate
Yeah,
information
perfect.
online.
Yep.
that,
I like it. Oh, good.
Very few crosskeys.
Yeah,
What is that?
Another big cobra.
that was a copepod, yeah?
Yeah.
Sorry?
This one. What is this one?
That's Twitch.
Another
Twitch.
cobra.
You can look at the,
Something just happened with the focus.
Exact convenience.
I don't know what's going on. You trying to make it harder on us? That was a house, right?
Too hard to stop on.
It was a house.
Yeah, I think it was a sinker again.
Yeah, that's what I think. Larvation house. I'm sorry.
see it.
We're going to try and only call them when they're coming in from the top near the center.
Could be from any kind of organism or
Larvation.
larvation.
Yeah, larvations make this big mucus house, and they shed the house. I've seen in some of the papers they can shed their house 10 to 12 times a day. So they use it to trap food, etc. And they only live a few days, if I'm correct. Russ?
What?
Larvations, lifespan? Isn't it only a few days or something?
It depends. We don't really know how much the deep water ones live, but in the Arctic, some of them probably only do 2 generations a year. They don't grow nearly as fast as the tropicals.
Okay. So it depends possibly on latitude.
Depends on how big the animal is and temperature at which it's living in the ecosystem it's in.
Okay, thank you.
But it can be as short as days, small species, tropical water.
Yeah, that's what I had seen. It must have been that paper. It was a tropical paper.
Thank you.
Me too. I'm learning a lot.
Okay, but the next one, the next listing.
Not till 50. We're good for a bit.
Come to me, you will. Something big off to the left in the side camera.
To the left.
Oh,
To the right.
Whoops, my other left. Sorry. Don't worry.
Something big to the
Yeah.
right. Do you see it, Dougal?
Yeah,
No,
everything turns to left in the southern hemisphere, right?
Wrong hemisphere, yeah, sorry.
Maybe that?
It was like a siphonophore
Or a
fish,
medusa,
very
or-
long.
Oh, you say long. I see
curved
small and
over on
round.
its- curved over on
This
its
is a
top.
medusa or a house.
Oh,
What's
that's
this?
good. Is that a Sudusa?
This looks like a doliolid. No?
Yeah, it's
Oh.
a Sudusa.
What does that mean? A
It's
sedusa
a
is what?
carnivorous doliolid.
Oh, I was correct on my doliolid. Yay! That makes me happy. If we could focus in on this baby, that'd be great.
Bonitinho, agora nós encontramos
Yeah, yeah.
Doliolídeo.
We had a lot of movement going on.
carnívoro.
Yeah, we had a lot of
Que é
movement going
animal
on.
dentro do grupo dos cordados. Geralmente esses doliolídeos são animais que comem partículas pequenas, né? Essa espécie é uma espécie carnívora.
Alguém falou oi pra mim?
Thank you, man. Falou. Professor Marcelo
Oi pra quem falou
Melo,
oi pra mim?
de volta.
Oh, that's great. Dougal, you good?
Oh, yeah, nice.
Yeah. It's a carnivorous doliolid.
Sudusa boscregrinus, probably. Oh, probably an Mbari, but I don't know if it's...
That's another ocean, another hemisphere away.
That's true. How many suction samplers we got left?
We have 4. You want this, don't you?
I think I do. We
Okay.
have...
Let's scan it.
It,
Let's do
yeah,
it.
maybe let's scan it.
Want to scan first?
Yeah, let's do DPIV, please.
Yep. DPIV, please.
So far, we only know it from Monterey and off Japan. Unless you guys have got some in Hawaii or somewhere.
Does it always have that little kind of
I've seen it in the CCZ.
Idea of where you're at?
It's going below us.
Oh, okay. I'll just eat all of them.
It went off that screen.
Muito tempo. já.
Here he comes.
Jam steak.
Ready for the lasers?
You can't collect it if it's-
Lasers on when you're ready. Yep, yep.
Sabe de tudo.
Do what you gotta do.
Pontinho assim, né?
Closed it.
Lasers on.
Synced up.
And I'm recording.
Fabulous.
Emits.
Currently tied up.
Turn them off uppers.
Is that kind of process?
Oh, nice.
Okay.
Let me know.
Super slow is the goal.
Yeah, that was a good
Let me
one.
know when you want the mids off.
Kakani, do you want the mid lights off as well?
It would help.
Okay,
But
go for it.
right
Oh.
before you get it to the laser.
Oh yeah, right before it gets in laser. It's that last minute thing.
Não, português e espanhol botei.
And as slow as possible.
Não, não vai ter também, porque eles têm que fazer pilotagem com a tripulação aqui, né? É, pode ser. Por aqui.
It's coming.
It's Skinner again tonight.
Yep. Nice. Come on through,
Agora
baby.
estamos fazendo
Now it's
aqui
just toying with us.
o
It's above it,
escaneamento
it's
do
toying.
animal, né, através do iris.
That was such a great start to that scan. It's coming back down. He's right in front. There he is. Keep coming, keep coming.
Opa!
Toying with us. Oh no, you do good.
É difícil fazer, mas ele tem que passar dentro desse feixe de laser, né? Para conseguir fazer essa fotografia tridimensional, né? Que é a técnica de fotografia plenótica, onde através de uma câmera que tem milhares de microlentes que captam a profundidade de campo e a direção dos raios de luz, o animal é reconstruído tridimensionalmente. Ou seja, não fica uma parte do corpo do animal desfocada na imagem, né? Então, é uma técnica utilizada por esse aparelho aí chamado iris. Na verdade, é uma fotografia 4D, né? Porque ela inclui também o tempo, né? Então, ela capta em vários campos de profundidade e consegue focar, né? Através de um algoritmo que utiliza várias regiões da foto que estão em foco, utiliza a borda de cada área dessas regiões que estão em foco e consegue fazer uma sobreposição das áreas em foco e montar uma estrutura tridimensional. Sim, tudo isso coletado com dados oceanográficos. No CTD ali tem garrafas que estão coletando amostras de água em várias profundidades. Então é uma exploração completa, né, dos componentes abióticos também, além dos componentes bióticos, que são os organismos. Incrível como a natureza desenvolveu, né, em vários grupos de organismo uma estratégia de propulsão parecida com a das águas vivas, né, com uma ondulação do corpo parecido com o que a água viva faz a pulsação da umbrella, né? Então, esse doliulo, que é um animal cordado mais próximo dos vertebrados, que são como nós, também ao longo do processo evolutivo desenvolveu uma solução semelhante para a locomoção dele, né? Assim como diversos outros animais que realizam essa compressão, né, de um volume de água para gerar a propulsão.
Maybe. He's gonna be back here. There you go. That's it, right? Oh yeah. That's nice to have.
It's not like it's always gets a little better. It's like this.
Going right below.
It's like rolling the dice on. So why not design that so that you've got the light right down at the side? Because you're using the light scan.
Oi, Léo, aqui a bordo do navio tem eu e o Marcos. Eu sou o Marcelo, professor lá da USP, e o Marcos é doutorando em oceanografia da USP. E além disso tem o Renato Nagata, a Silvina Bota, que são professores da FUG, e a Julieta, que é pós-doc lá na FUG também. Então são duas universidades brasileiras.
You could just get 2 inches this way.
E, Léo, nesse momento a gente está na mesma latitude de Alagoas, no Lago do Nordeste brasileiro, mas no Atlântico Sul em águas internacionais.
There, it's lost them, yeah. It's right out front, keep going, yep, coming down. There he is, right here, there he is. There we go. Lights are on, unfortunately.
Sorry, I was thinking the one with the camera and the other was,
Well, maybe.
was a recording camera.
Turn off. A little too far outside. Lovely. Those two are not usable anymore. And at 50.
Are those 3 and 4?
And at 50.
Are those 3 and 4?
1, 2, 3, and 4, I think, are available. No, those are D1 and 2. Those are 2 other things. So that is that, and then this is going to be when Russ is up, because it's the last bit. There's 3 Niskin bottles that need to be fired at 50 meters. So if you could just pass that along to Russ, because that'll be during his. He's got 11 to 2. You've got to 11.
No momento nós estamos na latitude de Alagoas, porém em águas internacionais.
And he may have missed, but we did catch a good copepod.
No.
Catch video and images, right, Russ? Yeah. We did stop for a copepod.
I love that. Yep. So we thought we'd make you happy.
So you gotta let that thing settle if possible.
So suction's off, and just move forward, and then he'll come to the bottom, and then we'll change canisters. Yeah, that little bit of forward movement. Hopefully it'll.
Which canisters are you?
1.
Settle
If we
down.
still, I haven't seen him settle though.
I haven't seen him settle either.
I haven't either.
He did go in.
He did.
Yeah, I saw it go in.
Maybe do a light little. Maybe also just
Not
maybe
the bad, but the
tiniest
we're
bit,
using
see if that'll
those
like.
and then we allow light like
Tá
even
checando no VAR agora para ver se entra mesmo.
So even with like a regular.
Once we've calmed down after the sampling event, if we can zoom the camera into the bits of siphonophore that are outside the
Nós estamos
desampler,
agora, na verdade,
get some
fora
nice
já da
height.
plataforma, né? Estamos em
It's against.
I mean, I saw it.
Continua.
So.
Well, normally they bounce off as soon as you take the suction off.
Dougal wants to take a look at it, see if there's around the top of the suction sampler. Yeah, like the red.
Suction sampler.
Sorry, no, the
desampler. All the stuff stuck to the top might wash off at the surface, and I can get information on the zoids, all those little pieces, what shape they are.
We're gonna curve it if it's still inside the canvas.
blowing in the wind.
Yeah, but it's always
Yeah, but most of the part.
the jelly-looking parts, especially, you can raise the contrast a bit.
Salve resolução da câmera. Parafusinho dele. Olha o parafusinho, cara. Hey Dougal, how many megapixels is that camera?
4K, so 4096 by 2040 or so.
Lower
So
sample.
you have to do the maths. I'm not sure about megapixels. I can get my calculator, but
Yeah,
It's
don't need that. Don't need that
effectively
now.
4K. That's where the number comes from. No, you're right.
4096.
It's 6K or
Dougal,
something
are
like
you
that.
looking at
True
this?
pixels.
Yep, I'm looking at that. It's great. What else do we got? We got some tentacles, a couple of brown pelpons. Is this full zoom?
Yeah.
Okay. Can we tilt down a little bit to see what's closer? That's it. And focus on the whoa.
On the tip?
Yeah, on the tip there. That's good. That's good with that high contrast. Yeah. All right.
Nossa, olha só, cara, a qualidade.
We should have some good shots there.
Of course, it's not gonna stay in one spot.
Beautiful
Seems
shot.
good. Okay. Thank you. Can we tilt down a little bit? And there's another piece of it that's a little bit further down the desampler.
On the desampler, not on the tip.
No, not on the tip. Go directly down and a little bit left. go. On the left. Take that guy. That's looking good.
Great. I think there's more stuff.
Okay, so
Up-
there's little
up and to the
clear
left.
palpons as well. You want to go up and to the left?
Yeah.
Okay, up and to the left, please. Zoom in.
Or the other one.
So back here again.
Yeah.
That guy in the shadow of the of where it's rotating maybe is not moving enough that we can get a good.
Isso que está preso ali é um pedaço de um sifonóforo que a gente viu há umas 2 horas atrás.
Zoom out a little bit.
E aí foi feita a
And below and to the left,
coleta dele, mas ele
somewhat,
conseguiu escapar e ficou preso só
there's
esse
another
pedacinho dele,
attached
que já
to
é
another
suficiente
piece of
para
equipment
fazer o
down there
DNA.
somewhere.
Ele passou pelo scan
Might have to
também,
zoom
foi
out a
todo
little bit
escaneado,
first.
digitalizado, né? E agora com as imagens e amostras de DNA a gente
Go
consegue, o pessoal
down a
que
bit.
trabalha com isso consegue delimitar a espécie, identificar a
There it
espécie.
is. Zoom in on that white strand down there. One on the one next to the zip tie.
Zip tie on the bottom.
There's not enough contrast to see the transparent stuff.
Uma câmera
And the fuzzy
4K,
stuff
né?
that's floating around there
Qualidade
as
da
well.
imagem.
It's on the other side, right? Okay, that's
E o
wrapped around.
professor Dugal está pedindo para que movam a câmera ao redor do amostrador para ver que tipo de tecido do animal que restou. Porque o amostrador, na verdade, ele abriu, né? Se abriu acidentalmente
Okay,
e o animal
let's go.
escapou, porém
Got some good
ficaram
info
alguns
there.
pedaços. E com alguns desses pedaços é possível realizar a descrição, né, da espécie e identificar se realmente é, de fato, uma espécie desconhecida pela ciência, né?
So they're the
Yes,
pieces we got to
down
pick
there.
off with tweezers when the ROV comes up.
But still, it's amazing.
You say we.
Yeah, I was gonna say, you mean I.
Not sure who the we is there. Alright.
The royal we.
So Dave,
So we're
you
going
get to tell
for-
the pilots what to do next.
Yep, and so we're
Good
going
luck, my
forward.
friend.
Forward.
Uppers coming up. You got this.
Yep, and going up.
Paperboard's all the way on now, eh?
Yeah,
That's
exactly.
it. Start collecting.
Stopping on every cut.
Let's release all these jets.
We're done with this gelatinous stuff.
This is good. Nice flying.
Yeah.
Like that. Alright. So what we got next?
A gente agora está começando a subir. O ROV chegou a profundidade de 1.200 metros
And what has
e
been
agora
the
nós estamos subindo
upward
lentamente.
speed? We've been just slowly.
Slowly, yeah.
Yeah.
Like, going up and forward at the rate that you can stop on stuff, but not so fast that, yeah.
Agora,
that everything
quem está
streaks.
I'm not sure
conduzindo
what all that would mean.
o mergulho é o professor David Fields,
See how it looks like that
que
steady step, the
é um
depth?
especialista em crustáceo,
No,
em copépodes,
where you
né?
looking?
Então
Oh,
ele
up
que
here.
Yep. The depth
indica a
one.
direção e o
It's
que,
like
que o
That's what we're looking for.
ROV deve
Okay,
fazer,
we were
né?
going great, and then we just
And then we
settled
kinda
out with that animal,
plateaued.
right? And now we're gonna go back and try that.
Yep.
That's the goal.
Respondendo aqui algumas perguntas do Twitch,
So how many
Did you hear
cans?
that?
essa pesquisa está sendo feita fora da Zona Econômica Exclusiva
We have four
Brasileira, em águas
canisters
internacionais, no Atlântico Sul. O cruzeiro partiu de Salvador. A gente foi em linha reta para fora da ZE brasileira, fora da zona da plataforma também. E a gente vai fazer o contorno no Nordeste, quando chegar mais ou menos ali no nível do Rio Grande do Norte, a gente volta para a ZE brasileira e para de fazer a expedição e vai direto para o Porto de Salvador.
I've not seen any pairs of them.
2, 3, and 4 are 1 to
Only the ones attached to the front.
Right. That seems weird.
2, 3, and 4
O
are
Schmidt Ocean Institute, ele está realizando na década dos oceanos pesquisas em diversas regiões dos oceanos. Cada ano ele está em uma região e esse ano ele está no Atlântico Sul. Já passou pela Argentina, já passou pelo Uruguai, Rio de Janeiro, subindo agora a costa do Nordeste do Brasil e vai contornar, né, o Nordeste indo em direção à região norte do Brasil entre 2026 e 2027. Então essa é a área agora Atlântico Sul, a área de foco do Schmidt Ocean e do navio Falkor.
Beautiful starry night. Oops, let's slow down. Let's go have a look at that.
That was a shrimp.
Oh, was it?
Or decapod.
Suggested it is a decapod, isn't it?
Oh, it's not that fast.
Yeah, we don't need to stop.
Yeah, we're okay.
Yep, keep going. Sorry.
No, it's fine, Dave. I was doing that the whole time.
You can do that. As a rule, if there's anything that's, you know, moving slower than the smaller particles that's coming out near the top of the screen in the middle, so it's easier to stop on, and you can slow down a little bit and drive up to it. And if it's nothing, you just drive through it, and we get some, and you know, one frame in focus while we're doing it.
Yeah.
Pteropod,
Missed it.
wasn't it?
I
Looked
didn't,
like it was
I
swimming
didn't
one.
hit capture in time.
Yeah, yeah. It's okay.
A little bit more forward movement. That's it, that's it.
Perfect.
Velocidade da luz agora. Star Wars.
Oikaplur.
Chair down and just slide it. I find that's just in too tight. Much better. Yeah, well, the reason it wasn't seeing is because I can't see anything from that chair. And that's too far away on the other side.
I don't think the 350 was on the equipment.
Perfect. Now I'm happy. Well, happy.
You feeling positively gay?
Oh, you're putting that into that
It's this feeling of like at any moment it could be something really, really cool.
Works on it. I have a feeling.
I don't think when we come back and focus.
As long as it's not like something like, you know,
Assim, trilha sonora pra isso, vamos colocar uma trilha sonora.
because when you see it, it's too late.
Não tô vendo nada.
You'd see it somehow on a radars.
They're stealthy, didn't you know?
They are stealthy.
They're undetectable. I've heard that from high sources.
I want to see a Phyceta.
What's a phyceta?
Sperm whale.
Oh, physetter.
Phyceta.
I was like, wait a second. We already talked about that.
Oh, we did?
So Nina and I have decided we're going to see a sperm whale and a squid interaction on this video.
On this
tonight.
video.
Yes, tonight.
Ah,
Right,
tonight.
Serena? We had, we
Getting
had predicted.
Getting reasonably specific. Can you tell us the depth? at which this-
785 meters.
And the time?
12
Okay.
ish.
Ish.
We'll have to get
Okay,
our
now we're
artist
gonna
at
get-
sea
we're
to
gonna get
make
odds.
one
Farther
and
away you get from that, the more
merge it into the feed.
Yep, chance it's going to happen. That's right.
Okay.
The whale's going to come in from the left,
We
and he's
could-
going to have an arm and a tentacle in his mouth.
Yeah,
could still
and scars from a previous encounter. 200-year-old
Funny. It's like being a 16-year-old driver. Everything seems coming so fast.
Fish. Amphipod.
Didn't you say something about hyperids? If there are a lot of hyperids.
I'm gonna grab your phone.
Karen one down.
Yeah, so if we see a lot in a
When we get up near the surface,
Yeah, I
we
would say
leave the
that's-
light on.
I expect we're seeing on the top.
Yeah, I saw you hold your hand. Kayak plan, we're slow and
Slowly heading up. Yeah.
Yeah.
Looking for targets.
You want more food? I'll take. No, not faster. Right, we don't want she wants more food. So is the idea we're just moving up and stopping on meters? Yes, that was what I was told. Going slow up. That's all. Stopping
Let
on
me know when
meters.
you need to
We
make
have a
some
bunch.
adjustments. I need you to just do exactly what you know is the right thing to do.
You either have to transept it or slow it down.
It's just extraordinary to see all that.
You can make any transept you want. Hey, you guys want to do transept? Karen said no.
So what is the shutter speed on this?
I asked about
Is there any
transect
of this?
first.
Is that none of this is blur?
It's just
It's
too exploratory.
We could slow.
straight
we could slow it
organisms.
900
That's a
meters
size.
and hover for an hour.
Most things with this data set are presence of
We have a dedicated
Wow.
If we're going too fast, we can slow things down. Or we'll just spend a lot of time at the end of the episode.
Yeah, I'm
cables, right?
not
Copper,
really
and a bundle of about 8 to 10.
We're up at 940.
Right, that's what I'm saying.
And each fiber can carry a lot of data, right? So there's one.
Dave, can we have a
Whoa! Slow down.
Yeah,
Stop.
so, sorry.
You just go through it.
It's okay. We'll just
Full of dirt.
bits
Wow.
of siphonophore?
Was it a
maybe?
You're disturbed.
It could have been like up
The
to the left, there
space-time
might have been-
continuum has been upset.
Yeah.
I think we all had the same reaction. Because we knew it was going to be too
Wow.
Yeah.
late. Maybe?
Yeah. The bits of mucus and stuff that.
Dave, anything you see it far away, just hit him toe.
Ghost
Yeah.
tail will-
I
That one
didn't
did
see
come
it.
up quick, so not much we could have done about that.
So maybe a little
Slower?
a little slower ascent and a little bit more forward as we go.
Hit what you need right
So we have our own Ethernet network.
So
It's all these open
the goal is to have the particles that are coming towards you look like you're just entering hyperspace but not streaking.
So,
And then you'll have at least one frame that'll be in focus as you're driving through things. And then you hit the camera,
This
hit them
is
on the
great.
camera.
This is great.
This is
Yeah,
good.
this is perfect.
Hopefully that will be it. And then they returbine.
So pretty.
A little bit shorter.
It's really a shame we can't have music on in here.
What would your tune of choice be for this particular video sequence?
Stop.
It's a Keating
Keating. Okay. Sometimes it could be a stealthy squid. Just saying. Just saying. My squid looked like this too.
We know which you got eyes for.
It's a beautiful Keating.
Oh, up on the left, top corner.
Really?
No, there's something else.
Manu saw something else top left corner.
Top left corner. So
Far away, small, be kind of specific.
very reddish, orangish.
Close, small, or far away? Need to drive forward to
No.
lose it?
Close. It was
Close.
well in
I
focus.
think it's
Alright,
probably
so we're
gone
done
then.
for it this way. Yeah, it's probably too far.
Let's go forward again.
Yeah.
yeah, forward.
Nice.
Oh,
Oh, radiolarian.
Or forearm, yeah, forearm maybe.
Was it fair?
Well,
Stop.
oh.
Sealed
I
area,
can't see.
bottom left. Got it. There'll be two forearm guys holding hands.
Wow.
In public?
There. that him?
There he is, right in front of us.
Yeah, right in the middle of the screen.
Okay, we want to film them and suck them
Doesn't
up.
make any sense.
No, they do. They're always like this. Quite common.
Holding hands.
Yeah.
I mean, wouldn't you?
What's it called?
This is a Coelodendridae. It's a single-celled rhizarian. There's two of them together. They reproduced and they're still holding on to each other. Outro protozoário aqui, Rhizaria, protozoário colodário. Unless somehow they can find each other and hold on to
Uma
each
célula
other.
bem grande, para os padrões de um tamanho de uma célula normal. Um animal que tem essas extensões ali da membrana plasmática, como se fosse uma ameba com pseudópodes bem longos, né? Então é muito interessante que uma célula consiga ter uma estrutura dessas aí, assim, muito interessante.
Going up off the screen.
Right up front here.
Suck it when we need to. I've never
My
seen.
intuition was that it was
Yeah. That has crashed twice very recently.
Normally the testing side is a lot more like spiny and it's not normally like that.
This picture is beat up.
I had it put on ROV the first time and it crashed pretty straight away. I think John ran out on Odin as well.
Low, bottom, middle.
Odin was just unable
There's a heap of these on the Oceanic Explorer video as well.
I think the thing that I'm saying is that
Just
from a point
leave it
of
like
view
that.
of search,
Look at the data.
they're not dense enough. We don't assume.
Aí novamente
There we
o
go.
protozoário,
Finding is not. So this
como eu
is-
falei, né? Ele tem essas extensões da parede, aliás, da membrana plasmática, que são como pseudópodes. E no centro dele você tem uma proteção, né? Um animal que tem um esqueleto, uma proteção de... Geralmente os protozoários têm um esqueleto de sílica ao redor do corpo deles. E por orifícios desse esqueleto que saem essas extensões da membrana plasmática.
I'm not zooming, I'm just
But I've
letting you.
never seen four.
Yeah, you ready?
No, when you have a mineral structure, you can't modify it.
Slower. I'm letting you fly though, I'm not gonna mess with your camera.
It would be nice
see what I mean?
to see if it's.
Yeah, we can check if it's fused.
Yeah,
Or is
because
it
I think the
contact?
spines
That's
are
it.
just
Yeah.
holding each other.
Yeah, and that we see in a lot of other radiolarians.
See how the central tests are actually
Fused. Oh, it could be during the swarming time. They didn't separate.
Here we go.
They both grew a test separately. That would work.
There's a
Hey Manu, wants to talk here in the microphone?
there's a glass tube joining the tube.
That's what I'm saying.
Please.
Visualization.
Eu vou deixar aqui o professor Manu Prakash,
Between those two
professor
white
Manu
spots,
Prakash,
is
que
there a
é
glass
indiano,
tube
Universidade
joining the two of them together or not?
So if we could focus.
Universidade de Stanford, e vai falar um pouquinho em inglês, né, obviamente, sobre esses organismos.
Hi, everybody. Nice to be back. We were just looking at two rhizarians
Here he comes.
that
Maybe?
seem to be almost holding each other. But this is quite puzzling because their bodies are made of these silicaceous structures. If some of you remember paintings and drawings from Haeckel, many people since him have been obsessed about these architecture. And some of these are the largest rhizarians that we know. These are all single cells.
Right.
And this is Phaeodarians. But the puzzle is that how can you have two of them holding hands? And because it's a mineralized structure, you have to grow them. And the probability of both of them finding is very, very low. As far as we know, they're not active swimmers. They're passively floating, although they do have buoyancy control.
And I had absolutely no idea anything. And I
It's incredible to see both of them together. So we have no idea how most of the radiolarians shape these silicaceous structures. And you can see some of the tentacles are broken.
Isn't this the reproduction organ, or it's not
It's
possible
not
to see?
known. For most of them, the reproductive cycle is not known. I think only for acantharians we have what is called swarming. They have a flagellate structure that will generate a swimming swarm. But because it's mineralized, it's not so trivial to divide a mineralized structure.
I see.
Do you see? Unlike other microtubule structures, you can rearrange them.
Okay.
How do you dissolve a mineral and still grow?
And then, yeah,
So diatoms do it, but they do it with this girdle. So only these symmetric geometries are possible. So you cannot get an asymmetric geometry in mineralized structures.
Yeah.
Because you are living in this shell, how do you break it and make two shells?
That's amazing.
So what they do is they just abandon that shell that sinks to the bottom, and most of them have fossil records, so you can go back in time and tell. Actually, the petrochemical industry uses radial ions as a marker for identifying a lot of historic, because they're very well characterized in the sediments.
Can I speak what
Yeah,
you say in
yeah.
Portuguese? O professor Manu Prakash, da Universidade de Stanford, estava me explicando aqui que é um animal que tem uma armadura de sílica ao redor do corpo dele, então não é fácil que um animal desse cresça, né? Então esse animal, quando ele fica grande, ele basicamente abandona essa armadura de sílica e produz uma outra um pouco maior. Então, estão tentando fazer a amostragem, a coleta desse animal. Os pilotos estão direcionando o ROV. E o professor Manu Prakash estava mencionando que esses animais, eles são muito importantes para a indústria petroquímica, né? Porque eles sedimentam no fundo dos oceanos e essas carapaças de sílica permanecem por muitos anos lá, e o estudo de diferentes espécies desses organismos aí ajuda as pessoas que estão prospectando petróleo a encontrarem os locais onde tem petróleo, né? Então há espécies desses organismos que indicam onde que tem uma maior chance de se encontrar petróleo, né?
Oh, it's just off to your right.
No, that's something else. It's the tentacle. That's that's the. Oh, maybe it gets stuck in those jelly tentacles, right?
Yeah, from the former sampling, yeah.
It's jellies versus Feodarians right now. I think it's stuck in this suction tube. I don't know how many of you have noticed that suction cap is made out of a flask that's bottom has been cut. It's right on the tip, I think. I'm gonna try ramping up the suction to see if that grows.
Mandar um abraço ali pro pessoal de Rio Grande, tá? O professor Érico Muchagata, Nilvia, Mauri, Hiro, Max, todos acompanhando aqui a transmissão, né? Um abraço a todos aí.
Yeah.
Sending hugs to my wife is there. my son is there.
They're single-celled animals, so pretty darn small.
They're pretty big for being single cells.
Well, yeah. Well.
Yeah, no.
Compared to my 60-meter-long jellyfish.
Yeah, I know what you're doing, Dougal. I know what you're doing.
Mansplaining.
If we didn't have the single cells, we wouldn't have the animals. No metazoans.
It's a necessary stage.
Yeah.
Whoa, down. Keep going.
Alguma coisa.
I think that excitement can only be for a squid. Let's find out.
Oh.
It looked like a viper fish at the first instant, but it was a key. It looked like a viper fish or something. Very good. They always sit in an angle like that, right?
Não vimos o que passou, mas dependendo da empolgação e de quem que ficou mais empolgado, é possível ter uma noção de que animal que passou ali. Provavelmente era um peixe, era um peixe. Um quetognato também, é. Ó, o peixe. Ou um tentáculo... não.
It's gonna happen.
Yeah.
I know if you want Russ to sit in there.
Oh, right there, yeah.
Mais aqui.
Oh, there we go.
Opa, que lindo isso.
This is a beautiful forearm.
Olha lá, o professor Manu pra caixa.
We'll get video, is that enough?
Yeah, can we get a close-up
If we get
video though?
close-up video and images, we're good.
I would like to do a scan.
Oh, yeah.
Yeah, let's do a scan on this one. Yeah.
Apparently can't
Because I see these two lobes as if on the two sides.
Vamos fazer o escaneamento desse animal agora, que não é um animal na verdade, né? É um protozoário. É uma única célula muito grande que vai ser agora escaneada pelo equipamento chamado IRIS. É um radiolar.
No, this is a foraminifera.
Foraminifera.
And
Oh, okay.
this most likely is Hastidurine.
Esse é um foraminífero, também um protozoário, muito grande para ser uma célula só.
Hastidurine pelagica. Yeah.
That's what I would have pronounced.
I don't know. Hastidurine.
In our mystery thing for the
Oh.
other day.
Oh, just passing through there. Yeah.
So these are forams, and you can see that the actual core of the cell, this is again a single cell, is at that white dark spot. And then you see a little layer of an ectoplasm around it. And all of these shiny little spines are possibly silicaceous. This is what we're going to try to figure out. Forams make what are called exopods, but these structures are significantly larger than the size of the cell itself, almost 10 times larger. And we believe that they feed using these tentacles. And what's quite surprising is that you can see if these structures traditionally are made of what are called microtubules, you know, the exact same polymers that separate our DNA when cells divide. But in this case, they seem to be making structures significantly larger than what we are used to seeing. So we're going to figure out whether they are actually silicaceous and possibly because we have one captured live in one of the containers before. And in the pelagic waters, this would be one of the largest single cells by size.
Professor Manu Prakash nos contando, né, que trata-se de um foraminífero que tem reticulópodes, né, que são como se fosse uma ameba com pseudópodes. Esse animal tem extensões também da membrana plasmática, que são mais ou menos 10 vezes mais longos do que o tamanho da célula, né, e ele usa essas extensões, essas projeções para capturar partículas, né, lembrando a todos que nós estamos em uma região que é pobre em nutrientes, que não seria como um deserto, um oceano. Nessas áreas tem muito pouco alimento, e esses organismos usam essa trama, né, de projeções para capturar pequenas partículas. É uma estratégia que é muito comum entre os animais planctônicos, né, de terem essas projeções, essas extensões, como se fossem tentáculos, que são usados para filtrar partículas pequenas que são capturadas. Is your brother? Your brother. Vem traduzir em espanhol aqui. Ah, é? Vem traduzir em espanhol aqui. Agora eu vou passar aqui a palavra para a Julieta Sebuhar, que vai falar em espanhol para vocês. Doutora Julieta Sebuhar.
Sí, meu momento de fama. Hola, buenas noches a todos, otra vez. Como pueden ver, siempre variamos para que todos puedan entender y estén todas las lenguas representadas en el streaming. Ahora estamos yendo atrás de este radiolario.
Oh, Dougal looks like the Rhizaria ate
Oh.
the jelly.
Essas extensões, essas projeções do corpo para filtrar, para capturar alimentos e construir e fazer uma filtragem dessas partículas.
El profesor Manu estaba contando que estos foraminíferos son unas de las células. Oh, apareció. Ah, un agua viva. Ah, qué linda. Que es una de las células unicelulares más grandes. Vamos a tener conflicto de interés ahora. Porque tenemos algunos que solo quieren agua viva y otros que solo quieren radiolarios unicelulares. Vamos a ver qué hacemos después. Bueno, como les comentaba, estos radiolarios usan esas extensiones para filtrar alimento.
But you can see the little sheets. It's definitely had a meal. Passive.
Oh, really?
They don't. But there's a lot of fade marine snow. Every little dot.
Del otro lado del chat también tenemos al profesor Eric Muchagata, que también es de la Universidad Federal de Río Grande. Obviamente puede complementar con todo su conocimiento. Es profesor de zooplancton de la FURGI.
Yeah. Great job.
Closer though. Yeah. Well, I mean,
Voltando aqui, português, professor Renato Nagata. Esse equipamento que está sendo usado agora, ele está fazendo escaneamento do corpo desse organismo, né? Então são diversos equipamentos que tem, não é novo, ele já tem alguns anos de uso, mas sempre tem alguma inovação, né? E então estava conversando com o pessoal aqui do Monterey Bay Aquarium. É um equipamento que tem uma capacidade de fazer imagens de alta resolução, então uma quantidade enorme de dados que é transmitido através dos cabos e que tem que ter uma capacidade de processamento e de transmissão desses dados, né? Ele capta 60 imagens por segundo de alta definição e faz uma análise depois, né? Uma sobreposição dessas imagens em várias distâncias focais, né? Então ele tem uma capacidade de analisar não só dentro de um foco, mas em várias profundidades focais, formando então uma imagem tridimensional do objeto que é passado ali pelo feixe do laser, né?
Traduciendo un poco lo que el profesor Renato estaba contando, esta cámara consigue sacar 60 fotos seguidas de altísima resolución por segundo y con una computadora de alta resolución permite después formar y sobreponer todas las imágenes para poder tener toda la vista de los animales que fuesen escaneados. Este iris no solo permite saber o calcular la biometría de los animales, sino o también medir el volumen, sino que también consigue medir las corrientes que pasan por estos tentáculos para poder estudiar mejor los movimientos.
Okay, so that's now ready, right? Thank you.
I'm gonna change my most recent annotation.
We're pulling this guy up, or
We have a set. Wow.
La transmisión del ROV empezó a eso de las 2 de la tarde más o menos y vamos a ir directo 12 horas transmitiendo hasta las 2 de la mañana. Después de eso, ahora estamos subiendo, llegamos hasta los 1200 metros de profundidad. A medida que vamos subiendo también vamos colectando agua en botellas Niskin para después hacer filtraciones cuando suben hasta el barco. Y posteriormente vamos a lanzar las redes de plancton y redes Tucker para poder colectar organismos también de esas profundidades, entre los 200 y 1200 metros de profundidad. Entonces hoy vamos a tener una larga noche de trabajo, no sé cuántos vamos a poder dormir, me parece que será más un día durmiendo 2 o 3 horas.
Traduzir para o português. Traduzir um pouquinho o que a Ruli comentou, né, da rotina de trabalho aqui embarcado, que é bastante pesada. Estamos fazendo a transmissão, essas descidas têm mais ou menos 12 horas, né? Todas as descidas estão programadas para começar às 2 da tarde e terminar às 2 da manhã. E nós temos um cronograma de revezamento aqui, tanto os pesquisadores que estão embarcados, como também a tripulação. E temos também marcado para 2 da manhã o início de uma coleta, um arrasto, que é feito com rede, e que vai durar mais ou menos umas 4 horas, coletando até 500 metros de profundidade, trazendo uma série de animais para a superfície que vão ser em seguida, mais ou menos lá às 6 da manhã eles vão chegar. Então é o intervalo do tempo de arrasto que nós vamos descansar, e chegando esses organismos nós vamos ter amanhã, talvez à tarde, para processar esses animais, né? Fazer experimentos, manter alguns desses organismos vivos, outros vão ser preservados em etanol para depois serem aqui mesmo extraído o DNA desses organismos e fazer estudos sobre a biodiversidade de organismos oceânicos.
Gracias, Dora. La idea es poder llevarles a todos los que están del otro lado un poco de lo que estamos haciendo acá, que la verdad estamos todos muy contentos y con mucho orgullo de que tanto las instituciones brasileras y bueno, de alguna forma también las argentinas, porque tanto Silvina como yo nos formamos en universidades argentinas, podamos estar acá.
está agora com 138 pessoas assistindo. 148 pessoas assistindo.
Ahora en este momento estamos con 140 personas mirándonos. Bastante, mucho, dice acá Marcelo.
Sábado à noite é ótimo.
Un sábado, o los sábados de noche, muchos no están saliendo, ¿no? Son las 9. No, es temprano todavía. Meu irmão.
Seu irmão?
Comenta que o pessoal tem uma equipe que faz a marcação de todas as espécies que são vistas e fica referenciada, a localização, tem várias especialidades.
A medida que estamos viendo estas imágenes, del otro lado del estudio hay un grupo de unas 2 o 3 personas. Hay un grupo de unas 2 o 3 personas que con diferentes programas están marcando, identificando los organismos que vemos. Y quedan registrados, se marca la localización, se identifica la especie. Las personas que están con esos programas, la verdad que saben un montón y con solo ver una simple imagen.
Eleitoral. Estamos de volta ao vivo do RV Falkor.
Meu amigo da Rede Globo.
Fazer voz.
Hola, creo que volvimos. Ahí nos habíamos caído por un momento. Terminaron de escanear el foraminífero y ahora seguimos subiendo.
I didn't even see it in that one, yeah. Yeah, that's good.
Go to radio.
Yeah, I'm not teaching science. I just.
You didn't change the projector, you just moved the camera? Where? Oh, you did.
Coisa do BBC.
Ah,
Ele
yeah.
falando bem devagar.
Yeah. Right. Running into it.
Como les estaba contando antes de que se caiga la conexión, esta expedición fue pensada y diagramada por varias instituciones, su mayoría de Estados Unidos. También tenemos gente que viene de universidades de Japón, tenemos investigadores de Brasil, de universidades brasileras, aunque tanto Silvina como yo somos argentinas y nos formamos en universidades argentinas. Bom, como pudieron ver ahí en el chat, ya estamos pasando por algunos problemitas de conectividad, pero en breve van a ser solucionados.
É mais legal, porque aparece a frente.
Aí começando. Mandando missão pra Marte.
Pô, essa ficou legal.
It's pretty shallow.
Yeah. I think if this is correct, right? Is it still
Tu quer falar alguma coisa? Fala da neve marinha.
The bottom of the thermocline is about
Voltando agora aqui para a navegação, subindo bem devagarzinho até a superfície agora, 800 metros de profundidade no momento. Vocês podem ver essa imagem do tipo Guerra nas Estrelas, né? Essa neve marinha que passa pela gente são partículas, né? São animais mortos, são fezes desses organismos que vão lentamente afundando e transportando todo o material orgânico que é produzido na superfície dos oceanos para camadas profundas. E é isso que é chamado de bombeamento biológico, né? Ou seja, transferência de matéria da superfície para o fundo, onde esse material, esse carbono, né? Essa matéria é armazenada por milhares de anos e colabora com o sequestro de carbono atmosférico, né? Então, esse processo ainda ele é acelerado pelos organismos, né? Que fazem a subida na superfície. Nesse momento, a maior parte dos organismos encontra-se já mais próximo das camadas superficiais, se alimentando, né? Onde há produção desse alimento, uma vez que a fotossíntese se limita às camadas superficiais dos oceanos. E depois, quando começar a ficar mais claro, né, com o nascer do dia, próximo ali das 5 da manhã, esses organismos se escondem nas profundezas dos oceanos, né? Então ocorre a migração vertical, né, que é chamada diariamente esse movimento de ascensão e descida, que colabora com o transporte vertical de matéria orgânica que é produzido na superfície dos oceanos.
more forward?
a little bit more forward, less up. Top left. Bathysaurus. Oh, not Bathysaurus, yeah, Bathychorus.
Opa!
It's probably right off to our left now, like behind us.
Agora vocês ouviram o professor Dougal Lindsay apontando, né, um animal, uma água-viva, Bathychorus. É uma água-viva que nós estamos procurando. Provavelmente pode ser uma espécie nova, uma espécie desconhecida de água-viva. É um gênero que tem algumas espécies de águas mais frias, né, no Ártico, tem registro no Ártico, mas como nós estamos aqui a 800 metros de profundidade, essa temperatura pode ser uma temperatura parecida com o que se tem em altas latitudes, né. Agora, no momento, 4 graus, né, a temperatura, embora estejamos aqui ao largo de Alagoas, né, em Maceió, mais ou menos, temperatura de 4 graus a 800 metros de profundidade. Então tem uma pequena água-viva ali que deve medir poucos centímetros e que nós vamos tentar coletar agora, porque pode se tratar de uma espécie desconhecida. É uma água-viva do grupo Narcomeduse, são águas-vivas que têm esse tentáculo virado para cima, né? Parece um helicóptero, e que pulsa, está pulsando ali, né? E que é muito curioso, né? O grupo todo dessas águas-vivas tem esses tentáculos para cima, que pode ser uma estratégia do animal para chegar desapercebido, né? Então ele vai se aproximando com esses tentáculos sem gerar uma turbulência, então ele não é detectado. E quando uma presa dessa água-viva percebe, o tentáculo já está chegando na frente e fazendo a captura do alimento, né? Então é uma estratégia que algumas águas-vivas possuem, né?
You want to scan it? It's really small.
Is it really small? Yeah.
Yep.
centimeter maybe.
Traduzir tudo. The narcomedusa.
Boticórios. Yep, same species as before. Yeah.
Did we get it?
We didn't sample it last time successfully, but we'd like to this time.
Oh yeah, that's when we tried this, because they were so tiny.
We lost it.
Wow. Bueno, voy a intentar resumir lo que dijo el profesor Renato, que es especialista en medusas. Lo que estamos viendo en pantalla es una medusa del grupo narcomedusa, posiblemente o Baticorius. Van a intentar colectarla, como pueden ver. Creen que puede ser una especie que no está sido descrita. La idea es colectarla, y acá a bordo tenemos un grupo de geneticistas que están secuenciando los genomas de la mayoría de los organismos que colectamos. Lo que contaba Renato es que estas medusas, bueno, tienen como formita de helicóptero con estos 4 tentáculos orientados hacia arriba.
Se aproximar sem gerar turbulência. Silenciosamente.
Que son para aproximarse a las presas sin generar turbulencia y poder captarlas.
I think that's what's coming, yeah. I haven't done it yet. I think, unless they're trying to just suck it up. Are we scanning this one?
No, just sampling.
Oh, okay, there we go.
It swims too much?
Yep.
It'll change shape as it goes through the scanner.
Right underneath, to the right.
So coming back upside. Sick man overboard.
É um animal muito pequeno, deve ter menos de 2 centímetros de tamanho, e é muito complicado fazer uma aproximação do ROV sem causar turbulência que afeta o organismo e espalha ele para outra região, né? Então, é uma habilidade bastante admirável que a pilotagem aqui do ROV tem que ter, né, para coordenar essa amostragem. E às vezes a gente fica bastante tempo tentando coletar e não consegue. É complicado. O animal mede poucos centímetros, contando com as correntes, né, naturais do ambiente e a turbulência gerada pelo ROV.
Como pueden ver y como contaba Renato, esta especie es súper chiquitita, entonces dificulta bastante la maniobra del ROV para poder colectarla. No solo eso, sino que también tenemos que tener en cuenta las corrientes que lo llevan de un lado para otro. Muchas veces cuando pasan estas cosas, no siempre se puede colectar, a veces perdemos algunos pedazos de los organismos durante la colecta, y otras veces se terminan escapando. Por eso cada vez que podemos colectar alguna muestra o algún organismo, es todo una fiesta acá dentro del estudio.
Yeah, right.
My arms. This is the hardest job.
I appreciate you. Dave Fields, you've got this. You've got this.
Dave, your leadership here is setting the pace.
You got it, Dave. Our
Aqui,
business is full of this. So swirly.
are we having another field day here?
It's like primitive AI right here.
Muito difícil, muito difícil. Aqui a gente está dando risada porque o professor David Fields, que está coordenando aqui a descida do drone, está apontando. Não dá para vocês verem aqui no estúdio, né? Mas tem uma pessoa que é o coordenador da descida que aponta os animais que devem ser capturados, né? E aí a pilotagem tá tentando fazer as manobras e ele com o braço estendido aqui, cansando, dizendo que a parte mais difícil é ficar com o braço estendido, porque ele aponta na tela e fica o tempo inteiro apontando para os pilotos aqui não perderem de vista o animal, né? Então é bastante complicado de fazer essa manobra.
I feel bad for not complaining about that thing.
They're gonna sell it all.
Ever. They knew what they were doing.
Except in Japan and Australia, everything's just.
It's really clear how difficult this is. When you don't have that thing now.
Okay, that was a big swing. Where is it? Is that it? Is that it?
See that boat?
Exactly. Put it in my cage.
Yeah, I think so.
Center
I
screen
think
at the
it's like a
bottom.
really big target too. So we're in a tough position.
Can extend.
Center screen at the bottom.
This is Marcelo speaking. I'm an ichthyologist.
Center screen.
And of course, we believe
Center
in
screen,
megalodon.
drive forward.
The scientific name of it,
Drive
it's
forward.
Octodus megalodon. It's well known from its fossil record.
Down a little bit.
And it was extincted like 3.6
Whoa.
million
Ignore.
years ago,
Keep going
so
forward.
it doesn't exist anymore.
He's getting
The only
he's
fossil
going
records
away
of it.
from us right now.
Is he, you see him?
I saw him up to about 5 seconds ago.
Okay, go forward. Going forward.
Whoa, low, go lower, low,
Down.
down, stop forward, go down,
Down.
down, roundabout, should be in the screen pretty soon.
Oh.
A little bit more forward,
Okay.
stop about there,
Okay.
forward. It'll be up near the top of the screen probably now somewhere.
Wow.
Agora perdemos, estamos tentando
Drive forward
reencontrar
slowly.
a água viva.
Somewhere down here. Bit more
Mal
forward
de
motion.
menos de 2 centímetros, então o propulsor pode jogar ela para longe, né? Dependendo da posição que ela passar pelo
At the top of the
ROV.
screen, I think, maybe now. Okay.
Eu acho que agora
I think you're Google nice
desistimos
track.
agora.
Nice track, Delta.
Infelizmente não foi possível e é comum, mas
See if we can find another.
seguimos em frente.
How many has that been?
Yeah, I could stand for it too.
Oh yeah.
Center everything up again so we know where we are.
How do you feel about this? Give a little narration.
This could be just like sports news.
Woo!
No? No answer?
No answer.
Yeah, okay. It could be the video feed from every ROV out there. So we have maybe
depth
one
is
or two
usually
screens
because
for
of,
Straight ahead, dead center.
Cold old water, right?
Maybe a low bait. Still quite a distance away, fairly large.
Keep going. Keep going.
Right in the center.
Yep, dead center.
Dead center.
Oh.
See anything?
I didn't see it either.
This is a trick.
Right?
Or your eyes are exceptional.
It was an AI hallucination.
Maybe we need to focus a bit further out. Yeah, it was a halo made by one of the other pilots.
Aqui a gente está numa região oceânica, muito respondendo aqui a pergunta sobre o nível de oxigênio, saturação de oxigênio nas profundidades. Essa é uma região muito oligotrófica, né, no meio do Oceano Atlântico.
Observation house.
Então, apesar de a gente estar vendo bastante matéria orgânica
Can we try
flutuando na água, que é a neve
focusing the
marinha,
main 4K camera a little bit further away, please?
a gente vai voltar assim que eu volto com a resposta assim que a gente passar essa fase de coleta.
It's an epiphany.
Like they've used up all the material.
All taxonomic keys.
Tá todo mundo meio em êxtase aqui atrás, né?
Is it a dome portal?
Tá todo mundo em êxtase aqui.
Because we don't know what it is. Can't even start.
Thank you.
Oik, Copepod,
It's
We're
that
just
everything's relevant. It's cold.
You can see that, right?
I can
It's
see
really
it.
clear when you have the things laid out in front of you.
I mean, there's
Not
It's the way that it's the tip of the dome, the way
Justo en este momento los pilotos del ROV están cambiando de turno, así que ahora vamos a empezar la búsqueda de la pequeña medusa.
the fingerprint. I don't know what's down there.
So then you
I
have
think you did
Como la perdimos de vista, ahora estamos todos súper focados en las pantallas para ver si la podemos encontrar de vuelta. Cada una de las cámaras que estamos viendo nosotros acá en el estudio tiene diferentes aumentos, lo que les permite a todos ver y buscar mejor.
But you see it correlates with the temperature, right? When the temperature starts dropping.
Search mode again.
It's just time consuming. The more you do it,
I don't get it yet.
a little bit, you kind of figure it out. It's
My
just
head
like
around
if you
seeing
put
something.
it
We're seeing like a
and it come back to you
small.
and you try and read it.
We need more. Okay.
4M.
A lot of us
yeah.
actually rely on going through the pictures and getting an idea of what we think it's likely to end up being. And then we run through the key to confirm we get there, or find a picture that when we're all done, the key says it's this. Finding a
It's okay,
picture
we
that
got
confirms
images
aqui
of
a
them.
resposta.
it doesn't.
É só
Yeah,
zona
enough
com
of
baixa
that?
produtividade e baixo
have
consumo de
images of
oxigênio.
them. Just bug screen it. Like
Enquanto ao largo da costa do Chile é uma região de altíssima produtividade, então isso implica num alto consumo de oxigênio e, consequentemente, o oxigênio nessas regiões é mínimo. Então, por isso que nessa região nossa aqui tem a saturação é maior.
A lot of it too is just having the right tools, having the taxonomic keys. I had most of the keys I didn't realize I had. I went looking after we identified the first maps. And we got a lot of
A little bit more forward, less up, please.
But some groups are organized better
forward,
than others
less up.
in terms of having a universal or a complete key. And then others are pretty much, you have to pull a whole bunch of stuff together.
Center left, a little bit lower than center.
You need to look at a bunch of different keys.
Bright white swimming thing.
There's no way to look at it all.
About 8/10 of the way down the screen now, heading to go out. Bring it down a bit. Keep going forward. Bring it down a little bit. It's at about 11:30 now, at bottom of the screen.
Yeah,
Almost
I'm not
going
sure.
off the screen. Go down. Whoa, off the screen now. Come into the center, still off the screen. Going down. Oh,
Did we collect some
get
fish?
back up again. There you go.
¿Colectamos fish? No.
Lost it.
Ah,
Sorry. Back in search mode.
menos mal que está o Marcos para organizar os pilotos.
Oh, there was a bit more forward. Yeah, forward is fine. Still searching. That's just mung. It's just mung.
Like, two?
Snot.
Move along.
Nothing to see.
Yeah, we're trying to play Tetris, getting all that stuff back in. But I've inventoried it all anyway.
Top right.
Yeah.
Come into center. Single cell.
Little Clovarian.
No, look at the members.
okay.
Get in close.
Area. Oh, yeah, this is another
Fidarian?
Yeah.
You sure?
Here is the
You sure it's not the
species.
forehand with
No.
everything pulled in?
It's too stiff.
Oh, this is the one with the lattice shell.
It's right here.
It's like the monkey ball shell.
No, I think it's the forehand with everything pulled in.
zoom in?
Yeah, zoom in.
I think it's the forehand with everything pulled in. It's not, the sphere is, all the dots are not completely in a line. You see that, like,
Yeah, but they're all on the end of each of the spines. They're just short.
that's a forehand. You can see, look,
Yeah,
look,
it's a
look
forehand,
the center, yes.
100%. It's just a different species. Yeah, it's different.
That's
That's a
Yeah.
forehand.
Lo
You
que
can
estamos
tell
viendo
from the
ahora en
core,
pantalla
you see the lobes.
es un foraminífero parecido con el otro que estábamos viendo antes y pudimos escanear. Así que el profesor Manu está como loco pegado a la pantalla. Ahora lo podemos traer para que explique un poco más sobre estos.
You can see the center region is made of several chambers.
Están explicando acá que este foraminífero está formado por diferentes cámaras,
Pretty small. Ooh.
Yeah.
por lo menos unas 4.
Ooh. Okay. That was cool. So it's not a single test.
Yeah. Right. So you guys don't want that?
Okay.
You don't want that?
I love that.
So you
Yeah,
want that?
can we sample it?
I don't. The mesh size on the suction sample.
La idea es
that we have
intentar
looks
también
very
escanearlo ahora
No?
y no sé si lo van a poder colectar o no porque por el tamaño es
Which
minúsculo.
chemical? One of the ones that's left.
On sample 3, we have 100 micron mesh.
And there's nothing in 3 now?
To choose.
There's nothing in 3 now? Anyone know if there's something in 3?
On the current one, this is
Elio or
person.
whoever has
Gonna rotate
2,
back
3,
and
4 are
forth.
empty.
3 and 4 are empty?
2, 3, 4.
Well, two's empty as well.
So you,
We try,
you could index to 3.
so we try indexing to 3
Yeah.
Oh, but that's
and then
that fine
try
mesh.
sampling.
Is that why?
Yes,
This is
because it's a fine mesh, yeah.
You could zoom in more if you want.
Real hard to believe that's a 4-ounce.
Yeah, I think. Did you
Yes.
guys say? Yep,
Yes,
throw it in
3.
In 3.
Okay, let's index to 3 and suck him up, please.
Okay.
I don't know, where will the tuna fall?
Can we rotate the canister backward? Did I see you do that?
Not sure.
Hey Marco,
Yeah.
can you guys index, I mean, don't do this now, but I'm curious, can you index back to 2?
Yeah.
Okay, good.
Oh, so we can go back and
We can
reuse
go
the-
back.
You can go back and forth.
You could go either direction.
Okay.
Because we'll be able to see if this is on the bottom when we rotate.
Yep.
get it, we'll rotate that to the other one.
These are small targets. Oh, got him.
Holy smokes.
Nice work.
Way to go.
This is 3.
It comes in the cans. Are you getting images or it's too late?
I already got one.
Oh, you got your in the tube, so now we're gonna watch it.
Ah, the shadow cam? Hopefully.
Bueno, pudieron colectar este foraminífero. Ahora estamos viendo si llega hasta el tubo 3. Sí, llegó.
It went back up to the top.
Manu va a tener mucho trabajo esta noche.
See if he sinks.
Section's off. It's not sinking. It's gonna be like the other guy staying up at the top.
Yeah.
Yeah, he's got all
Don't
those
you think,
spikes
Dougal?
that'll probably
Yeah,
go through
like
the
that
mesh.
other really small one.
It's 150 micron mesh.
So we wanna go to 2.
Can you unzoom over here?
Okay, cocked on 2. Thank you. Back in search mode, and we've got 1 canister left to fill, right?
2.
Two.
Yeah,
Two.
about a second.
2 left to fill.
Much more smaller. Right.
No more throwing away samples if we can help it.
Yeah.
Just 2 high.
Estaban diciendo que este foraminífero puede llegar a medir como mucho 1 centímetro, o sea, pueden tener más o menos noción del tamaño y de cómo es difícil intentar capturarlo con el succionador del ROV, con las corrientes moviéndose de un lado para el otro.
Big piece of mucus straight ahead.
I'll be fine. So the set man, you could use the fact job on to focus however you want. Okay. Sometimes whichever we get, you could zoom on focus. You could also use the pan. So
That narrative
it's not
just said
fun,
like
but it's set
3 or 4 more hours.
And then, when is Maya's birthday party? Tomorrow.
And then my,
So you don't want to do it tomorrow?
if I'm blind, doesn't mean to come to meet Father up or down.
And was Grandma
To help
able
you
to
out.
come? She was able to come? Can okay. I just sent a message to Emmy, and she said that she loves the cephalophore, the one with the green head.
Oh, great.
Oop.
Yep,
there.
there we go.
Yeah.
Oh,
You should
More
you should
coelodendrons.
ask your friends. Oh,
There you go.
another Feudarian.
Oh, there you go.
Yeah.
They're stuck together again.
They are together. That is unusual.
It's the
Apparently
same ones
a-
from before.
It's not
No.
that
I
unusual.
told you we come back and find them.
It's not that
Yeah.
unusual.
You want to go right to collecting it after?
We already have one, and we've only got 2 canisters
Oh, I
left.
thought we missed it.
Oh, didn't we catch
Not
it?
even for 5
No, we missed it.
We
You
missed it?
We
Oh.
missed it.
No problem.
And you said, we'll find
You
others.
enjoy-
And I
Okay,
said, oh, we'll find
alright.
some.
So we
We'll
get
be
this,
back
we
up.
get a bad tortoise.
Hey,
and
talk to
tortoise
Emmy.
and
Talk to Emmy about something.
And these guys are not joined together at the
Emmy's
center.
mom, the photo of a cephalophore.
Can get one in
it
focus
has a
zoom
green
before
head,
we
and
try
he was
collecting.
super excited.
Nice.
So you should talk to her.
What are they called?
Love you.
Coelodendron, the
Talk tomorrow.
Rhizaria.
Bye.
Okay.
Submenu Rhizaria. Coelodendrida is COE.
Did we collect this, the first one? No.
We tried.
We tried, yeah.
So these are not joined together, look at that.
Yeah, I'm really confused by this. They cannot find each other. That doesn't make any sense.
But they do remain attached. The ones I've
That's
I've
what's
mostly
happening, that
seen
in
them
the
going
swarming
like this.
stage,
And
they are
most of them-
growing together.
Well, it's possible that the non-shelled stage would at least.
That's what I'm saying.
Yeah,
Yeah.
it remains attached,
The
but of
cytoplasmic
course-
thread doesn't break.
Right.
That's it. That's
So-
the
so, but
only
it
way
has to
you
size
can get
the size
more.
a new skeleton.
Exactly. Yeah, because you cannot split a skeleton.
Right.
that doesn't add up.
Yeah, I mean, diatoms are the same problem.
The person who's gonna look at this, they're gonna have
Well,
glass.
yeah, they make it work by each keeping half
Exactly.
until the shell gets too small.
Oh, that's so beautiful.
Wow.
Oh my
Oh my
goodness. You can see the
God. What a good
spine.
shot.
Nice job.
This is silication.
Even for these guys, it's good. Dougal, common name or group for these guys again?
It's a rhisarian, it's Coelodendridae.
And you can actually
Coelo
see the shell.
Dendridae.
Dendride.
Got it. It's rosarian.
I guess that what translates into coelom, so a space
I think
with
it's-
dendrite.
Este organismo que estamos viendo acá, no sé si lo voy a poder pronunciar bien, pero se llama rhisarian. Y lo estábamos viendo cuando estábamos bajando con el ROV y ahora lo volvimos a encontrar subiendo porque lo habíamos perdido. Obviamente, tal vez no sea el mismo, pero bueno, pudieron por lo menos hacer unas buenas imágenes.
11:30.
Right. Just try to focus it in there.
We've got beforehand, we've just got 2 or 3 others to give me a plate. Usually it's Dougal gets small.
Posiblemente no lo escaneen, pero van a tratar de colectarlo.
You know, they're entangled. They're not actually attached.
Yeah.
I think this is the same thing we see in the marine snow.
Ahora, por suerte, el profesor Manu vino para explicarnos un poco sobre estos organismos.
So we'll switch to English for a second. You're again seeing some Feodarians. You can see the round shell, and inside that is the cytoplasm and the nucleate material. Feodarians also are called because they make what is called feodeo, which is kind of, you can think of it as a fecal material of a cell, and that's the very dark center. You can see the silicaceous spines, and they're entangled together.
Yeah, we're collecting.
And
is there a particular chamber
you can
to go
see
in?
the 4 tentacles on the 2 sides. And what we were discussing a few minutes ago is for an organism that's planktonic, has no known capability to maneuver in the water column, how would 2 of them find each other? And so most likely it was in the swarming stage when they were born, the 2 cells remained together and ended up growing this mineral structure. So if some of you compare this to another planktonic organism that builds architectures out of silica, which is glass typically, is diatoms. Diatoms solve this problem that they can still divide by making a mirror replica, and one plate goes to one cell and then the other plate goes to the other cell. Kind of like a petri dish. I think they're stuck on just on the periphery. The Feodarians are fighting the jellies, and we know who wins. They're stuck, I think. Did we get it?
No.
No. Where? Oh, there. Yeah. We missed it. So it's quite unusual to have these biomineralized structures. We see a lot of them in marine snow, so thousands of species of radiolarin with mineralized structures are found in marine snow, and we believe that they act as a ballast to bring a lot of marine snow down. Oh, almost, and it's... we missed it. I should shut up. I think I'm jinxing it. The other thing about these structures is if some of you start digging into the history of radiolarans, you can start with Haeckel's drawings. Of course, a few of them were described, but he described some of the largest rhisarian groups. And till today, his drawings stand as a testament to the details, and many of the species are identified some of his drawings from the Challenger expeditions. But of course, he didn't have access to such deep sea organisms, and deep sea radiolarians are not characterized at all. We have very few descriptions from in situ imaging. Oh, it's stuck outside. You see it? It's outside the cone, right under it. Huh? Yeah. Right there, no? They're dandling, huh? No, it's not the tentacle. I think it's the... Yeah, it's the two phales. You think so? Okay, we got it. Okay, got it?
I think it went in. It's just probably stuck inside.
I don't know. We'll keep watching the canister. It does look like two small cells.
That's what I'm thinking. The two things that are stuck under look very similar to the what we're trying to do. Those.
No, that's a loop.
No, those are the cells. Those are the cells. You can tell. Oh no. Okay. Yeah. Yes, thank you. Okay. That looks suspiciously similar. Yeah. So, something about radiolarans was when Haeckel started studying it, he wrote a letter to Darwin describing what he was seeing, and he almost transitioned from his career as a painter to a scientist because he fell in love with these shapes. And one of the most famous remarks by Darwin when Haeckel wrote to him was because everybody was still pushing back on the theory of evolution. And Darwin was convinced at that time that the ocean would be the place to study evolution because, just the breadth and diversity of organisms. So when you think about these organisms, and at that time it played an important role that this idea of evolution applies at a single cell stage, that single cells and diversity of single cells have the same sets of criterias that apply to what we understood at that time in animal evolution. And that's when Haeckel finished his first large monographs describing the thousands of species. Most of them are still found in fossils, so it's very rare to find most of them alive. And as yet, as far as we know, nobody's been able to culture them ever in a lab setting. So we're stuck by just collecting and studying them for a few hours before they die. Dougal, we need you.
Set up for view. Small inside.
Now?
Same again?
Yeah, I thought I saw that.
Oh, we did get a good shot. We did run through it. What's the section? What number counts? So you think we got it? Because we haven't advanced the canvas. I think someplace else.
Oh, we didn't get it.
I like the electronic spot. So do we think if we think we got it,
Yeah.
Did you think we got this?
I saw it go into the funnel, but that's as far as.
Marcelo and I never saw it go in the canister.
The canister?
We looked, but we didn't see the canister.
It's
But
pretty
they're
hard.
pretty small to see
They're
it.
pretty solid though.
Yeah, they wouldn't survive if we got one of it. Do you have the
Yeah.
shower still running?
The pump, is the pump off now?
No, it's still pumping.
Still pumping? Could you turn it off to see whether it's hit the mesh, please?
Sure. So,
Was it number 2?
Yeah. Yes.
Can you fall down?
We haven't seen anything.
Maybe the silicaceous branch is wider than
It's caught somewhere.
I think it got
It's
caught in
rooted.
the shadow graph. I mean, it's mineral. It's
Maybe
gonna
it's
stay
caught the
there.
tubes.
This big.
Yeah, but if
Shadow
you turn, it won't make a turn. It's rigid.
Keep it on this canister
Let's try and
maybe.
get a little bit more
Yeah.
forward motion.
Yeah, turn ahead.
Wonder if they still have the thrust master on board.
Not gonna lie, I don't miss that.
You don't miss it.
That was really
What time
fun.
we dived in 2? What time we started the pool?
Why did we, did something break?
Say again?
What time do we start the pool?
Had to go with the thrust
Did,
master.
did we start the?
No, what time do we start the pool? What time do we need to be in there?
What?
Do you know what time we need to be in there? When does the dive end?
Oh,
At two-
at two, no.
Yeah.
It was 1:30.
And also
Yeah,
you
it
can
was like
explore.
1-to-1.
From about
Was originally,
200
right?
years ago, I
Sorry.
Ei, Marcel, põe a gente como colaborador.
And this pair thing seems to be.
Yeah, it's fairly common.
This pair is a strange.
I've only seen one time with 3 attached to each other. It's normally only 2. But I've never seen them like this, where like one is within the
House?
other. Normally they're spread out wide.
Right center, just so
on
the pool's up.
ends.
No, I think what's happening probably.
Like a fluid.
Headlights.
Yeah, it's just a host.
Yeah. God, how do you see that?
I thought it might have been a jelly for a second.
Moving along.
Yeah. Move along.
Yep.
Okay.
Nobody home.
Nobody cares about this.
Yep.
Put it on Zillow.
Put the lights back on.
Not those, but not those.
Be up and forward again.
Switch off with you on the hour, dude?
Yeah.
Half hour?
On the half hour. You can have it. I can't see like 80% of what you guys see. I can't see half of what everybody else can see on this.
Is the cylinder
Like,
gray?
what?
Okay.
You saw
Is that the
something?
question?
Kidding me? Where the hell's that?
Just a second.
Oh, there you go.
Small jelly.
Oh, how to create it?
I was gonna say.
Saw that.
Zoom in close. Possibly Heliscara bigeloi. Going for a look.
Oh yeah.
Oh, nice.
Olha, voltando aqui agora em português,
I heard that.
avistamos aqui uma água-viva, provavelmente Alicera bigeloi, um animal que tem 2 tipos de tentáculos, 4 que são mais compridos e o outro são mais curtos. Animal do grupo de Traquimedusae. Dá pra ver os 8 canais radiais e 8 gônadas ali. Essas bolinhas brancas que são possíveis de enxergar. Um animal bem interessante, pulsando, muito legal.
No.
Oh yeah, we're gonna have to catch this one then.
Oh.
Oh. Oh.
Equal rights to host it. Okay. Suck him off.
Stay off?
Yeah, we can.
Are we advancing too, or are we?
É um animal que o professor Dougal Lindsay comenta que pode ser um complexo de várias espécies que são chamadas desse nome. E vamos tentar coletar esse animal. Here we go.
Yeah, basically. One with a very rounded, proximal part of the gonad, the radial canal, the gonad, and another one that's quite narrow. And we know there are two clades of DNA, but we don't have a match of DNA yet.
Parece ser a Lycera bigelowi. Animal muito pequeno. Oh. Vamos ver se ela vai entrar na bomba de sucção.
It's in. That's okay.
We're watching
Opa.
Suction sampler 2.
Canister watch.
See it?
Entrou e saiu.
He went in and then he went out.
No, he went up to the top.
That clearly
It did look like it went out, yeah.
He looked like he went out.
Oh, voltou.
Suction off.
Okay, VAR says a go.
Isso aqui.
Thank you.
Pronto.
That was 2.
Okay, so
So
four
is
is the
4
last
empty?
one.
Is 4 empty? Dave,
4 should be
Dave
empty.
Field should know. Dave, which one's still empty?
4 is still empty.
Thank you.
2 has
Good.
2 in it. Maybe.
Maybe 2.
Wait, you guys, that means we're gonna have one more.
Hey Dougal.
Yeah, we're at
Someone asking whether it's Alicreas. No Alicreas.
It's the same family as Helicreas. It's a Helicreatus.
Helicreatus.
But it's Heliscera bigelowi.
I mean, I can do measurements all day, but like for the purpose of these.
Earthmoon
You're at 700 meters, so likelihood of finding.
It seems not to be answering to Explorer. It seems not to be Alicreas, but Alistera bigelowi. Same family, different genus.
We still got 3 out.
According to Professor Dougal Lindsay from Jamstack, Japan.
I think she's gonna want her video. I think she wants video.
Well, I'm happy to take more time doing imaging.
We should stay below the thermocline if we do
I
that.
think if we could sit right.
Yeah, that was the mistake they made.
There's you go.
Oh, there we go, Sidipid.
Yep.
Let's go take a look at this.
No, I think it's
Great, you guys get another gelatinous organism. Awesome.
It could be a squid with tentacles hanging down. Come on, you don't know.
It's a squid. It's a squid.
Some
All right, it is
Martensa.
beautiful, fine.
It does look pretty.
It is pretty.
It's the same one we left behind earlier.
Oh, all right. All right. I take back my
Kitenóforo. Oh.
There it goes.
It's not going to be an easy one to catch.
Very small.
And
Any cephalopod.
I think let's bring it on now. From now on, this is how it's gonna be.
Entering
Yeah.
to Explorer 1. Yes, Alicera bigelowy. Actually, probably a complex of species. And then we got some, and we will analyze its DNA and try to compare with other sequences published.
Yep. We can follow him really slowly, and then when he's calmed down a bit, approach him again. He'll be swimming, changing direction all over the place until he calms down.
That trout with that.
Fish, cyclophone. There's a jelly coming down from the top. The other side. I'm finding I'm picking better stuff up in that camera. You just have to spot it and trying to rotate. Did he escape us? Set up for search again?
Don't hold this notch to it, and I'm gonna make sure that I kept it in my head so I couldn't hide it. You can't put look, it's hidden in the I have it in the note. It's on my data. Can you wait for a second? I'll be on here about 10 minutes.
Oh, is that it? Is that a new one?
The 2 dots.
It's the 2 dots. No, it's not a tuniform, but why is it got 2 dots? We zoom in and take a look. Weird shaped snow. Yeah, it's just mung. It's a weird shape though. Keep tilting down, there was another Medusa about the same distance.
It's structure, but not, you know?
Yeah, it looks like a corpse of something. Yep. Thank you.
Yeah.
Oik.
Journey.
Oh, yeah. Zoom in on this guy. Is that the Solmissus from yesterday?
One from
No, I
yesterday
think
was-
it's in our freezer.
was a little farther away.
Oh.
Grab it.
I can't see any stomach pouches. Solmissus alarm.
I've never seen one with the stomach pouch, so.
Yeah, it looks more like a Solmaris, doesn't it?
Solmaris.
Zoom in, yeah.
Não, é iridescence.
Does it
Now,
have
is
no,
that stomach or is that where the gonads are?
it's kind of one and the same thing in a lot of the species.
That does look like the one we saw yesterday.
distinct stomach pouches if there are any.
Eu acho que é um pedaço de um sifonófilo. Foi coisa, não lembro, garrafa 5.
I've never noticed this on the umbrella.
What is that red pigment? Is that
That's
actually
it's just refraction, yeah.
Nice.
Muscle tissue refringence, I think it's called. Yeah, I'm not seeing stomach pouches.
I think it's got a plastic washer stuck inside.
Oh, that's nice. Oh no, it's got pouches.
Yeah?
Yeah.
Little lamp spot on the one side, little white dot.
Could be, or could just be the tip of the injured tentacle.
Somares, Dougal.
So, Mrs. Yeah. Can we see if there are stinging cells on the outside of the umbrella there? I think it's the same one we got yesterday. Much longer tentacles.
Something in the back.
Oh, big Tomopterid.
Big
Okay,
Tomopterid.
follow
Go
that
after
for
the
Karen. Yeah,
Tomopterid.
go after that. Yeah. Oh.
Wow.
Big
That's cool.
She'll love it.
Where are we at? 694?
What's it called, Dougal?
Tomopteris.
TT.
Tomopteris.
Agora poliqueto. Poliqueto pelágico, tomopterídeo. É um animal que é próximo das minhocas, né? Uma minhoca marinha, basicamente. Animal que tem um corpo segmentado e em cada segmento tem parapódios que ele usa para natação. Vocês conseguem ver o batimento metacronal, né, dos parapódios nadando, né? São como nadadeiras. Ele realiza esse batimento que é muito bonito, animal fantástico, antenas, longas antenas. Vocês conseguem ver ali um animal lindo, lindo, lindo.
What's that?
Is it suctionable?
It's huge.
It'd be a one-off like try for it because it's really fast. I
Estão
keep our
decidindo
distance and zoom
se
as long
é
as we
possível
can.
capturar esse organismo ou não.
It might not be
Once
Uau.
it takes over, it goes.
Agora acelerou para fugir. Uau, e deu um show aqui.
Yeah.
Tomopteris.
That was a nice
Came back again.
Once we go video, we may as well try and suck it. I mean, it's going to be hard.
Yeah. It's gonna scooch right out.
Well, you might get lucky if once you get the head started in, you're good.
Yeah, I've never seen him swim backwards at any speed.
You probably can't. We don't have enough suction to really get too much of a fighting chance. We can
He's
try.
taunting you right now.
He's like PIV me, PIV me.
Yeah, are you?
You want us to do some particle measurements?
Sure.
Just kidding.
That's
Ooh, that's good angle though.
I said that won't work.
I'm recording all of that, Yost. Does it have eggs in the tips?
Any babies on the tips of the tail?
Just look at that.
Nice, dude. Thanks, you guys.
Oh,
Ooh, I don't think so.
Oh, wow. That's some kick-ass footage.
That was gorgeous. Nicely done. You're right. I know who you're going with.
I think they're gonna be-
I know where you're going.
Yost is a hater. We have a paper on these guys.
What, scanning them?
Doing a bunch of
that's
swimming
great.
measurements.
Mm.
Okay, we just need to write a paper on the biomechanics of that sinusoidal motion.
We did.
7 phones.
Yeah, we got the high def going on.
So are we not suctioning this?
We'll try, but they're very fast swimmers. You'll need to get the head in, and it'll need to be sucking the moment the head gets close, because it'll feel the current if you go on it with the pump on.
The problem is once the setae are damaged, it doesn't survive that long. I think we should
4.
do it. I've been imaging the immune system in these guys for some time.
Okay, zoom out
Yeah.
on your side.
Have
Turn the
the
lights back
immune
on.
cells travel through, and then you damage. They actually show up at the hair sites. It's really
Oh, we were able to get one in a
Actually,
suction
we
sample
could,
or something.
Yeah,
It can be done.
Yeah.
wait, so do you guys wanna catch it, or could we size it real quick?
Anything you want.
Anything I want, careful. I will, if I could do an IRS, shot, just to get some sizing.
Okay.
Sounds good.
Marco, could you bring the iris over the? Yeah. You could take the side camera. I'll fly over the sets. You
Yeah.
take the side camera and do whatever you need to do.
It's starting to get
Nice and easy.
frisky.
So for this, you're gonna want.
Yeah.
Maybe we just follow in that.
Get good images.
How's that camera?
Does he know the 7-hour pattern or? Like 7 o'clock?
Let's put it on.
I don't think there's any of the ocean.
We fished them all out. The Japanese fished them all out because they get tired of doing.
I think there's plenty around. They're just too smart for these wacky cameras.
Yeah, they're all hanging out.
Yeah, behind it.
Yeah.
They're behind the arrow.
Waiting for us to
Saying,
reverse.
nice try, kid. That does look pretty large.
Hit the vehicle.
Yeah. Can I get a copy of the video you took?
Yeah. Hit the vehicle again. Now it's.
So you're all looking at an annelid worm, Dimopterus.
So not
They're
curling in a ball.
incredibly
Oh, yeah, it's the same. It's not
transparent,
too much.
usually. Have the sensory setae, the two giant
Thank you.
mustaches sticking out.
What was that?
Well, there's always a siphonus.
And they're so transparent that under the microscope you can image individual muscles.
There's a cyclophony to scale.
Hey Jimbo,
Open the lights.
Oh, yeah.
could you give me the
And
iris?
one of the remarkable
increase
things
the-
about
or
these
Open the
organisms
lights.
is that they produce a rare color of bioluminescence.
Could you?
So people have been looking for new forms of bioluminescent
the,
reactions. The most common one that we all know is the luciferin-luciferase reaction. But these guys produce a yellow
Sorry, put back you one day.
bioluminescent, and it turned out to be actually a new compound.
Yeah.
We use all kinds of luciferase assays in biomedical sciences in many ways of making measurements. So it's incredibly remarkable to have a new color, and that's thanks to Dimopterus. It's very rare to see them bioluminesce, but there is one or two papers showing that at the tip of the legs is where these light organs are.
You're zoomed in over
You're lucky note on that.
This just looks expensive. I mean, but it looks that cool. You know? Some serious money.
So should I release
So you
that
wanna go
one?
right to sucking them
I
up
can keep
now?
it at 5. That one shot was 39 bigger
Tiny. Thank goodness.
Did we turn the suction now? This reactor.
I don't think the suction's on yet. It's getting frisky. It's about to come in our first. Except it's flat, so with the 3D amazingness is kind of when it comes under that angle like that.
Well, how flat is flat?
We can't what it moves.
Oh, so we're using the
There we go.
I don't know, I don't trust it. Like that didn't trigger. Like that was
There was
not
no
in
death knell.
it.
Are you happy yet?
Can you do one more nice one for me, please?
Fine.
So the answer to your question was no. Not happy.
He said he's trying. I will take that.
It's moving too much.
5 seconds before it shows.
All right, let's collect it if we can. Thank you. I think we have some good stuff there.
Oh, it's far out now. Straight. There.
You could take these high camera if you want.
Oh, it's right there.
Oh, that was a nice producer.
Mm. About to scale it. Got it? Get it?
Yeah.
Got it?
Where is it? It's in?
It's on its way down.
Is it?
Rotate. Is that 4?
Yeah.
That is
I
4.
don't see it.
I think it went down the side. I didn't think it could do it.
Yeah, because the seats here are pretty stiff. I think it got caught.
They're not
Do
enough
you
to
see
jam
it
it.
go in?
They're not enough to jam it.
Some gremlin somewhere.
Where could it be?
Got the legs up against the wall, resisting the flow.
Like, I don't be still holding on right there, isn't it? Turn the flow off either side.
Oh, it's stuck right there.
Yeah,
It's
stuck
in the
in the
tube.
tackle.
See?
Oh, yeah.
Yeah, the keytae are stuck. Yeah. You
Oh,
see
you know what it might be, is it might be caught on the,
Oh, those tentacles are holding
the
everything.
parts of the jellyfish.
Or it's just swimming.
Oh man, it's trying to.
Because this jellyfish piece is still all over the inside of the tube.
No, the keytae are pretty stiff. Oh my god. That is tough. It's
Oh.
done.
No, don't. Yeah.
Yeah, it's
Tentáculo maldito, hein, cara? Tá? Não, não tá não. Ah, tá só embaralhado. Tipo o cabelo quando bate no vento.
This is the very last. This is the very last one.
yeah,
That's a different
Scott. It's dead. There's something for the DNA. Yeah. It's stuff.
Yeah, great catch.
It's not tricky. You can't rip it apart. It's very strong. You may be able to recover it when it gets easier. Yeah.
Wait,
It's
the top, all it takes is like
It's not gonna recover. That's it. Huh? It's stuck in the. It's setae or stuck.
No, I think it's held by the tentacles. I think it's tentacles. Yeah, it's been held by the tentacles.
Either way, it's stuck.
Yeah, it's collected. Yeah.
No, it's. It'd be nice to put it in the box at least if for genome.
That's what I'm most curious about. If there are more.
Here we go,
That's amazing.
Oh, they're a lot more solid than we thought.
Distance with Devon's
Ah, there he goes.
Busy. It's in.
Well, he got released from the front.
You got released from the front?
Yeah.
Oh, I didn't.
Yeah, so it's in.
Hey!
What is it? Oh,
wait,
no.
One up. Is it up against the top or go back out the hole?
No, just.
Yeah,
He's
on the top, I think.
okay.
Yeah.
Yeah. Close.
Sheer tenacity.
So that's our last sampler, by the way.
Yeah.
Great. That's full.
That's great.
Can't tell. Did you get some fluff or you get that?
I thought it was. But I saw some weird stuff on the other side.
You're assuming this one?
Oh, wait. But I'll. The hole. Okay. That's just try to set it down into the chain. I think it's going to back up. Yeah.
No,
Okay.
that worked.
Looks like a little more up on the lid of it.
Yeah. Maybe they got stuck up.
Okay.
Oh, could you see the chamber?
Yeah.
No, look at. Oh. No, I was just wondering how you knew.
Yeah, everything else.
Say it again.
It's not inside.
It's.
It's not
He went out.
He's in.
went out.
He went out
He on the
again.
roof of the lid.
What?
He was gone when you rotated the sampler.
He was gone. Yeah.
So it's part.
It's not in the
So
sampler.
he's still in the tube.
Oh, it's back.
Oh.
No, pieces are back.
No, that's whatever was in the tube originally.
Yeah.
So that's his.
You think it's right in there?
Normally when we suck in, it's very like
Yeah, there's something up against the top there.
It's either that or he got into
That
the
corner
gap
right
at the
there.
side.
That's where he was. Or something. That thing's pretty big. Yeah. Like, damn, yeah, that's your
Maybe a tissue sample.
Yeah, but that's still good.
Right? I mean, we're not-
We might get them in the trawls.
Yeah. I just
We
mean-
get the small animals all the time.
Get a behavior quality animal.
No, we do in the trawls.
No, I mean, just.
Not that large. Oh,
No matter what.
No, this is ghost.
that is done.
The probability of getting it in a trawl is very, very small. Compared to the volume.
We got the one yesterday. I isolated one. I mean, big. That big. Yeah, not that animal.
Safe on the floor?
All right. Well, we've still got a few more hours left in this dive, but we are at time for restarting the stream for this particular dive. So I'm going to be restarting the stream in the next few minutes. You should get kicked over to part 2, but if you don't, and the stream just ends, keep an eye out for part 2, and we'll see you over there.
No, but you know.
I mean, this is for you?
I think I pretty much know.
Got it?
One of them has two things. Presumably.
Press some buttons.
Now we're
Yeah,
just
so-
imaging samples.
Well, good thing is that
What?
Come up.
We gotta,
But let's find some giant
Let's-
larvae.
We've seen so few tentacles.
We haven't
It's about
seen
time they came out.
that many.
We're gonna collect them anyway.
But that's
Come to Jelly Papa.
Oh, what's that? On the left.
Yep. Cheating now?
Okay, okay. Right, let's do that. Annotate.
Oh, a doliolid. You see that?
Where?
I thought.
Flying around up here.
Okay.
Yeah.
Oh, I have to do biological observation.
No,
That's just a Keating now.
I've
You
got
want
pressure housing.
a little jelly here.
Maybe solder some wires. I'm not sure yet. That's the problem still. Yeah. On my lab. System.
Yeah, zoom in.
Oh, another hala created.
How do we do that if I want to switch
Maybe
This
to
thing?
another and come back?
Okay, can zoom in.
hala scara, it looks like.
Mascara. Hella.
HALISCERA.
Mascara conica or Bigelow mascara?
Maybe, well, I'm not sure yet.
John is here. Let's call it Bigelow.
Which group does it belong to in the
Trachy Medusa.
Thank you. Whoops.
Trachy Medusae.
Yeah, zoom out.
Created a
Oh, too far for you to get a focus on now.
Maybe conica. Yeah.
Yeah.
That's right.
Need a better look, Dougal?
I haven't quite got a look at the radial canal. See those rounded bits there?
Yep.
Need that in zoomed in and in focus.
Spell that right.
Well, I'm looking at
Ooh,
echoes.
are those tentacles in a single row or in groups?